we all are aware of it, as this is an annual event, and the professors of the School of Crop Improvement will present before you the significant highlights of the educational achievements of the different divisions across the School of Crop Improvement. So again, with the permission of the chair today, we may be allowed to start that event. Thank you very much.
Thank you, ma'am. May I now request our director, sir, for his kind introduction of the chairperson to the August gathering.
A very good morning to each one of you, and it's my pleasure to welcome you to this presentation by the professors of the significant research achievement by the students. Those who did not have the opportunity to present yesterday, their work will be presented by their professors. I have pleasure of welcoming Professor Malvika Dadlani on this occasion as chairperson of this program. As you all know, that she is one of us. And distinguished alumnus of IARI, and served as scientist, senior scientist, principal scientist in the Division of Seed Science and Technology. Also spearheaded the Division of Seed Science and Technology to a greater height. Her contribution, particularly in policy issues, be it protection of plant varieties and Farmers' Rights Act, or it is seed bill, and various other issues, International Seed Testing Association standards, norms, taking seed science and technology to international standard. She has played immense role in doing all that, and also as a great educationist, as a teacher par excellence, working in the area of seed physiology. Her contribution is enormous, and by training she is a physiologist, but she took up rather than plant physiology, seed physiology, because seed health is very important. If seed health is not good, then plant health will not be good. So that's a very specialized area where she made immense contribution, and she also served this institute as joint director research. And in that capacity, again, she guided the institute program, farmers participatory seed production. A new dimension was provided altogether to this activity, which was taken again to a greater height. Currently, she is serving the National Academy of Agricultural Sciences as editor. We are extremely pleased to have you, madam. She is also a fellow of National Academy of Agricultural Sciences. And whenever there is a discussion, particularly organized by the National Academy of Agricultural Sciences, her intellectual input on different aspects is very, very valuable. We have pleasure in welcoming you, madam, to this program, and thank you very much for sparing time. Thank you very much. Thank you, one and all. And please maintain this number throughout the day. We are going to have excellent sessions, excellent chairpersons of the subsequent session as well. And you will have wisdom of listening each one of them. Thank you so much.
on genetics and plant breeding. Thank you.
Honorable Chairperson, Madam Malvika Dadlani, former from all the disciplines and the viewers online. A very good evening to all of you. I'm Anju Mahendra Singh. I stand before you to present the significant educational achievements in the discipline of genetics and plant breeding. You can see here the statue of Gregor John Mendel, whom we call the father of genetics, and genetics division celebrated the 200th birth centenary in the year 2022. So the basic mandate of our division is the same as that of the institute, that is on basic and strategic research for genetic enhancement and applied research in varietal development, transfer of technologies, postgraduate teaching and research, and the development of human resources, development and strengthening of the collaborative linkages and initiating new ones. Now, in the year 2022, we had 71 faculty members, of which 21 are now from the associated institutes. We had 104 total students on roll, of which 28 have qualified for their degree. And from these, 18 are the MSc students and 10 are the PhD students. And I will be presenting in very, very brief only the salient findings of the work of these students. I have divided my work into 4 major themes, and a gamut of crops have been covered in the presentations of these students. So coming to the first theme, which is on abiotic stress tolerance, 7 theses were completed in this theme. P. Dharma Teja, the PhD student of Dr. Rajveer Yadav, carried out the molecular mapping of key traits influencing phosphorus uptake and utilization efficiency in wheat. He characterized in depth 182 wheat genotypes, carried out their gene-to-E interaction studies, and also identified the molecular markers. So these are the exhaustive experiments that were carried out. And out of the 15 traits, 7 traits were found to be the maximum contributing traits, and 4 genotypes were identified as the most efficient genotypes for the phosphorus uptake and use efficiency related traits. Out of 7 QTLs that they identified with different models, 18 QTLs with pleiotropic effects were identified as the common. Ravi Kiran, PhD student of Dr. Gopala Krishnan, worked on mapping of quantitative trait loci governing heat stress tolerance at reproductive stage in rice. He worked with a large number of germplasm and basmati lines, as well as the 127 rills, in which NL44-127 is the donor parent for the heat stress tolerance at reproductive stage. So this is the exhaustive phenotyping that has been carried out in the basmati and other genotypes. Also, the rill was phenotyped in 2 different locations, and the GWAS analysis was carried out. And based on this, the most salient findings are that all the basmati genotypes showed sensitivity to the heat stress, and 10 quantitative trait nucleotides for grain yield and 14 for spikelet fertility were identified. And from this study, 5 QTLs were found to be novel. Abhishek, an MSc student again of Dr. Gopala Krishnan as the chairperson, worked on genetic and molecular characterization of tetra QTL pyramids of PUSA 44 with tolerance to reproductive stage drought stress. And he worked with 49 of these QTL pyramided lines and worked with 5 molecular markers, and also carried out the background genotyping with SNPs. So the salient finding from his work is that 4 tetra QTL nils were identified that were significantly superior over PUSA 44 in both the irrigated and the drought conditions. So these have a applied value and can be further done further multi-location trials to see their worth. Ashvi Teja, MSc student of Dr. Vijay Pal Badhana, worked on marker-assisted selection of rice F2 recombinants with phosphorus uptake and QTLs for drought. From this cross, Swarna into IR4064 DRT1, he worked with 300 populations, and out of that, using the molecular markers, identified 22 plants which had the presence of all the 3 QTLs, and these plants resulted in higher grain yield. Medha, MSc student of Dr. Jyoti Kumari, worked on the genetic variation for terminal heat tolerance in emmer wheat and diversity analysis using the SSR markers. Heat during the terminal stages in wheat is a very devastating factor, which is coming to light in the later years. So, though she found significant variation for agromorphological traits in both timely zone and late zone conditions, but when structure analysis was performed after molecular characterization, only all the 96 genotypes were clustered only into 2 groups. And this data suggests that Indian emmer wheats have less diversity, and we need to bring in the diversity from other ecogeographic regions. We know the MSc student of Dr. Mallikarjuna worked on sweet gene family in maize to find identification, evolutionary analysis, and functional characterization under abiotic stress. This is more of an in silico and bioinformatics study. So mining and molecular docking of these genes was done, and also the functional analysis was carried out. So the salient study from this work is that genome-wide mining revealed 20 sweet genes, and these are directly or indirectly regulated via transcription factors and microRNAs. And the co-expression network work also showed that there is an interlink between abiotic stress tolerance and sugar metabolism at the expression level. Shubham Sachan, MSc student of Dr. Patnaik, worked on the assessment of horse gram genotypes for phosphorus deficiency tolerance. He used 16 traits, and he studied 3 different inorganic phosphorus conditions in the hydroponics. So from his work, he identified a cultivar which is the most susceptible and the other which is the most tolerant to phosphate availability. This can be used for the making of mapping population. He also studied the expression of 8 genes encoding the phosphate transporters, but this study is preliminary and more validation is needed as the next step. So this was about the first theme. Coming to the second theme on biotic stress tolerance, where 8 theses have been submitted and successfully qualified. Jordan, a PhD student of Dr. C. Bharadwaj, worked on marker-assisted pyramiding of FOC genes for developing the wilt-resistant elite chickpea lines in the background of PUSA 372 and also another one that is JG16. So this is the systematic marker-assisted breeding program, and the numbers are given here. So finally, the markers were used for 5 markers were used for foreground selection and 48 for background selection, and the phenotyping was done with the help of specific races of the wilt pathotype, and also the phenotyping was carried out. So the salient finding from this work is that the nils of parent PUSA 372 were found to be superior for all the 7 characters. Moreover, the backcross lines of the parent JG16 were the top performance in the multi-location trial with yield advantage of above 20% compared to the national check, which is also the recurrent parent here. So Subhash Chand, PhD student of Dr. D.K. Yadava, worked on this genetics of white rust resistance and molecular mapping of the underlying genes in the Indian mustard. He used 2 isolates to do the disease screening under the controlled conditions in the 25 genotypes, and also carried out the bulk segregant analysis for the inheritance. So the inheritance studies highlighted that white rust resistance is governed by a single dominant gene in the 3 genotypes, and it was mapped to chromosome 5A. But in another genotype, Donskaya, more than 1 dominant gene could be seen as controlling the white rust resistance. Vishal Dinkar, PhD student of the erstwhile professor Dr. Vinod, worked on the mapping of leaf rust resistance in Triticum spelta and monococcum derived lines in wheat. So the spelta and the monococcum derived lines were screened for bulk segregant analysis to find out the inheritance. And the salient finding is that a single recessive gene for leaf rust resistance was identified in spelta on chromosome 1DS, and in Einkorn, a dominant gene for leaf rust resistance was identified, mapped to the chromosome 2AS. And the beauty of this work is that both of these genes appear to be novel when based on the leaf rust pathotyping test and the genomic location based on linked markers. Another student, Raghunandan, worked for his PhD with Dr. Shailinder Jha on the molecular characterization and Jiva studies for leaf rust resistance again, but in this case in Triticum dicoccum. And he used the 173 genotypes as well as a single genotype NP201. So 3 different models were used for Jiva's analysis, and the F2 derived F3 population was used for the molecular mapping. Based on this, significant MTAs for resistance to leaf rust pathotypes were found on different chromosomes for the specific Lovely Arya, another MSc student, worked with Dr. Niranjana on genetic analysis of yellow rust resistance in wheat primary gene pool derivatives. So she used two introgression lines, one in the spelta background and the other in the turgidum background, having the immune reaction to yellow rust, and found out very clearly that yellow rust resistance in spelta introgression line is due to a single dominant gene, while that in the turgidum is due to a single recessive gene. Prema Kumar, MSc student of Dr. Ranjit K. Allur, worked on the identification. Yeah, so he worked on the identification of differentially expressed genes associated with rice root knot nematode resistance, and he carried out the transcriptome analysis and used the resistant and susceptible RILs from as bulks, and he was able to find out 83 differentially expressed genes, and their upregulation and downregulation was also studied. So 83 genes were found to be associated with root knot nematode resistance, and also the genes involved in ethylene biosynthesis, secondary metabolite biosynthesis, etc. are associated with rice root knot nematode resistance. Abhiroop, an MSc student with Dr. Rama Prasad G, worked on the molecular characterization of Fusarium wilt, responsive host genes in the contrasting genotypes of pigeon pea. And he carried out a systematic infection study in the 30 genotypes. And from this study, he was able to, and also carried out the molecular characterization with the RT-PCR and for the H genes, and also the enzymatic and non-enzymatic activity in these genotypes. So he was able to find a most wilt-resistant cultivar and another most susceptible cultivar for making the mapping, biparental mapping population in future, and also found out 5 genes which were observed to have higher rate in fold gene expression in the tolerant genotype. So this was about the second theme on biotic stress. Now coming to the third theme on the improvement of quality traits, 7 theses have been successfully completed. Rahul Kumar, PhD student of Dr. Talukdar, worked on the inheritance and molecular mapping of seed traits in soybean. Madam Chairperson, he was a student who also got the Best Student of the Year award. Congratulations, Rahul, and Chairperson Dr. Talukdar. And he studied the inheritance of seed coat color and other seed-related traits in the F2 derived F3 population, and also the QTL analysis for seed size and shape traits in the F2 population, from which he found out 17 seed-related QTLs, including the 9 major and 8 minor QTLs. And he then carried out the validation also of these mapped QTLs. So from his salient findings, seed coat color is governed by interaction of 2 genes which act in the supplementary interaction fashion. 18 QTLs and in F2 and 25 in F2 derived F3 were mapped for 10 seed-related traits, and of these, 6 were validated in the soybean germplasm. Mohammad Zahirul Talukdar, a Bangladeshi in-service candidate, carried out his PhD work with Dr. Firoz Hussain on enhancement of grain amylopectin in maize through marker-aided introgression of VAXI1 gene into the elite genotypes. A very systematic study was carried out. First, the putative variations were identified between the VAXI and the wild genotypes, and the markers were developed, and these were then verified or validated in the germplasm, and of these, one was used for integration of the VAXI1 allele into the elite genetic background. As you can see here with the stars, this is shown that the VAXI genotypes can be identified. The hybrids were developed, and in these, amylopectin content of the original and mass-derived hybrids was compared. So the salient findings are that DEL24FR was identified as the functional marker for VAXI gene, and the genotypes holding double recessive gene had more amylopectin than the genotypes carrying the single recessive gene or the wild types. Vijay Kamal Meena, PhD student, again Dr. D.K. Yadava, his second student, worked on the genetic and molecular analysis of alpha-tocopherol content in the Indian mustard. So he carried out screening in the 96 genotypes and identified the contrasting ones, developed the breeding populations from out of those, and carried out the inheritance studies. So this is the first study in the Indian mustard about the alpha-tocopherol in which the inheritance has been studied. It was found that this high alpha-tocopherol content is governed by recessive genes which show duplicate gene action, and the markers developed in this study would also be helpful in future for the mass. Swarnadeep Ghosh, MSc student of Dr. Harita Bolinedi, carried out genetic characterization and molecular mapping for gamma oryzanol and its components in rice. Another bright student from our division who received the merit medal yesterday in the presentations. Congratulations, Swarnadeep, and chairperson Dr. Harita Bolinedi. And he worked out the genetic variation and the spatio-temporal variation for the gamma oryzanol and its components, and identified the genomic regions associated with this trait or the compound. And his salient findings are that this is the first report on the compositional dynamics of this compound. He found wide genetic variation in Indian germplasm and identified 4 potential donors and also robust MGAs and haplotypes from this study. Hargopal Datta, MSc student of Dr. Gyan P. Mishra, worked on identification of markers, biochemical parameters, genes, and pathways regulating the seed size in lentil. He carried out, he used 2 techniques, RNA-seq analysis, wherein 375 million high-quality reads were obtained, and the linkage analysis using the bulk segregant analysis. Again, a first comprehensive study to identify the key genes and the pathways. And the bulk segregant analysis showed that PBALC449 is linked with small seed size, which is present on chromosome number 3. Mayank Sinha, MSc student of Dr. Muralidhar, worked on association mapping for grain micronutrients and anti-nutritional traits in mung bean using the SNP markers. So he carried out marker trait association analysis and analyzed it with 3 different models. And this work resulted in the identification of 35 SNP markers and candidate genes, and also a lot of phenotyping revealed genotypes which were higher in grain iron and zinc concentration, along with their identification of their linkage groups. Deepak, MSc student of Dr. H.K. Dikshit, carried out genetic diversity and stage-specific micronutrient concentration and antioxidant analysis among different mung bean microgreens. So the 40 mung bean genotypes were used in this study and were grown for 6 days and were harvested after 6th day or after 10th day. So after the antioxidant analysis, it was found that this and the micronutrient concentration were relatively more for mung bean microgreens at the 6th day rather than over the 10th day stage. And 5 genotypes were identified with high antioxidant activity, which could be very useful in the further mapping and other genetic work. So this was all about the quality improvement work. Coming to the last theme, which is on the improvement of other economic traits, 6 theses have been successfully qualified. Ranjit Sarosh, PhD student of Dr. D.K. Yadava, his 3rd student this year, carried out genetic diversity analysis in Brassica gentiana germplasm using phenotypic differences and molecular markers. He went on to do an in-depth analysis of the yield and yield contributing traits in as many as 289 diverse genotypes, and from his work he showed a high genetic variability and association between seed yield and yield related traits, and also found a marker associated with seed yield per plant was detected. So it needs to be validated now and then can be used as a component marker for the component trait. Mahek Anwar, MSc student of Dr. Avinash Pandey, worked on morphological and molecular characterization of rice germplasm and screening for the presence of fertility restorer genes. So she worked with 65 genotypes, and she was able to identify 5 genotypes which had the in which all the 6 molecular markers used by her could be amplified, and therefore they were designated as the dominant homozygous genotypes. Therefore, they can be used as potential parents in the rice hybrid development program. Now, Vadla Chandrika, MSc student of Dr. Pralay Bhumik, carried out marker-assisted identification of restorers and assessment of heterotic potential in white-compatible rice genotypes. And 121 such white-compatible lines were used, and these were based on the molecular markers for fertility restoration, grouped into 3 categories. And their fertility restorer potential was identified by using the line into tester testing. And from this, the panel of white compatible lines with high level of fertility restoration was identified and can be then utilized for the development of inter-subspecific hybrids for better exploitation of heterosis. Abhijit Mundale, MSc student of Dr. Somnath Roy, worked on assessment of genetic diversity and polymorphism associated with key domestication genes in the land races from Assam. And he worked on the, and his heteromorphological and genetic diversity in land races is shown here. And he also did the molecular markers screening of the 3 domestication genes. So this data revealed a considerable variability for as many as 36 traits, and the landraces could be identified into different groups of indica and os, and the presence of the different alleles of these 3 domestication genes studied by him could also be deciphered in these genotypes. Ankit Dawar, MSc student of Dr. Gaikwad, worked on the assessment of genetic variability and molecular marker profiling in the Indian dwarf beet, which we know as the Triticum sphaerococcum. He worked on 117 accessions collected from different parts of the country and the world. And this diversity analysis has found 7 different clusters of these genotypes. In addition, a lot of other phenotyping was done. So significant variation was observed for these genotypes with grain iron and zinc in these sphaerococcums. More than that, in the bread wheat lines were identified, and for other traits also specific genotypes were identified in this study. The last presentation is for the student, last but not the least, from Yashaswini, MSc student of Dr. Sudhir Kumar, who worked on the assessment of genetic diversity in horse gram germplasm for agromorphological and nutritional attributes. So she analyzed 34 genotypes in a systematic fashion and identified genotypes for higher grain yield, protein content, carbohydrates, iron and zinc content, and 1 genotype for low phytic acid content, which could be very useful because horse gram is very less screened species. So this study is very useful. So Madam Chairperson, with this last slide on the thematic presentation, I move on. The research work from all of these students' work has resulted into 22 research publications, and out of these, the ones with RAS rating between 6 to 7 are 4, but 18 are with the RAS rating higher than 7, reaching up to 15. And the work has been published in various international and national journals of repute like Frontiers in Genetics, Plant Science, Scientific Reports, Plants, Plant Breeding, PLOS ONE, Journal of Cellular Science, and so on. The Faculty of Genetics has also made 141 research articles publications in national and international journals. Most of these are with the RAS rating higher than 7, and the book and book chapters are 39. 85 research papers have been presented in different conferences and symposia, and 81 popular articles and technical bulletins have been published by our faculty members. Honors and awards. This is a privilege to present this before you, madam. Every year, this year also, our faculty has backed very prestigious awards. Professor A.K. Singh has led the team of RISE, and they have received the Nanaji Deshmukh Award for Outstanding Interdisciplinary Team Research in Agricultural Sciences. Professor A.K. Singh was also honored for the Sri Ram Chandra Rao Memorial National Award by Angarao and also received the Nasi Fellowship. Dr. Rajendra Kumar received Honorary Fellowship of the Society for Recent Development in Agriculture. Dr. Rajveer Yadav became NAS Fellow. Dr. G.P. Singh received INSA and Nasi Fellowships. Dr. H.K. Dikshit became NAS Fellow and bagged the A.B. Joshi Memorial Award. Dr. C. Bharadwaj became Adjunct Associate Professor at the UWA Institute of Agriculture in Perth, Australia, and also Eminent Scientist Award from AgriVision. Dr. Firoz Hussain bagged the prestigious Dr. B.P. Pal Medal of IRI. Dr. Gyan P. Mishra received Dr. Harbhajan Singh Memorial Award from IS, Indian Society of Genetics and Plant Breeding. Our younger faculty is also not behind, madam. Dr. Vignesh Muthuswamy became NAS Associate, and he additionally received 2 more awards, NAS Young Scientist and NASI's Platinum Jubilee Young Scientist Award. Dr. Harita Bulinedi also received NASI's Platinum Jubilee Young Scientist Award. Dr. Pralay Bhaumik became NAS Associate and also received Outlook, Agritech Summit and Swaraj Awards. Dr. Vikas became Fellow of National Academy of Biological Sciences. Dr. Rajkumar Junchare became ISGPB Fellow and received S. Ramanujam Memorial Award. Dr. Muralidhar Asti received Excellence in Research Award from SSD Merit. Dr. Ganapati Mukri, Dr. N. Singh, Young Scientist Award, NJI India. The list doesn't seem to coming to an end. Dr. Niranjana, DB2 Bicare Fellowship, as well as the Early Career Award of the Burlock Global Rust Initiative. And our students are not behind. Many of our students, they received MSc Thesis Award, IRI Merit Medals, Best Posters, Best Oral Awards, Oral Presentation Awards, Young Plant Breeder Awards, Rahul, Ravi Kiran, Swarnadeep, Aragopal Datta, just to name a few here. not able to complete all of them, but we are proud of the achievements of all our students. 9 of our 18 MSc students have come back to us for their PhD at IRI, and some others have got admissions in other universities. Out of the 10 students who are going to get the degree this time, 9 are already well settled, 4 in their job already, and the other 6 in the ARS or assistant professor, etc. And madam, for your kind information, 6 of the ARS vacancies this time in genetics and plant breeding have been bagged by all our IRI students. This is all about my presentation, but it will not be complete without acknowledging the great support and guidance of our Honorable Director and Vice Chancellor IRI, the support and the facilitation by Madam Dean and Joint Director Education and Joint Director Research and Extension for facilitating the research and extension work of our faculty members and students. To all the divisional heads and professors, thanks are due for sharing their facilities and instrumentation and other facilities with our faculty and students. To Associate Dean, PG School, Dr. Atul, for assistance in miscellaneous activities, to the research guides and outgoing students for providing the information included in this presentation. Special thanks to Dr. Vignesh Muthuswamy and Dr. Rama Prasad and SRF Sumit in slide preparation and editing, and to all the faculty members of discipline of genetics and plant breeding for providing the information as sought from time to time. To Mr. Ishwar, the senior technical assistant in professor's unit, for daily administrative assistance, and lastly to all the PG school staff for daily and also during this convocation all the assistance that they have provided. So with this, I thank you, Madam Chairperson, for giving me this opportunity to present, and I thank you one and all. Thank you.
Thank you, Professor Anju, for making a wonderful presentation, well within time. Actually, you have saved a couple of minutes for us. Thank you for that also. And more than anything else, once again, the division of genetics and plant breeding really stands out in all aspects, whether it is students' work, the recognition and awards, number of papers in good reputed journals, and so on and so forth, for which I congratulate and compliment the whole faculty of the Division of Genetics and Plant Breeding, and to you, Professor Madam. Thank you. Shall I? So we were just discussing that I will reserve my remarks to the end in my concluding remarks so that we can save time here. And therefore, without wasting any more minute, may I invite the Professor of Plant Genetic Resources, Dr. Veena Gupta, please. And you have 12 minutes to make your presentation. And I think Professor Anju has set the standard, and I'm sure you will also follow the same. Thank you.
Hello, yeah. Good morning to everyone, respected Chairman ma'am, Dean ma'am, and Monica, and all other faculty, students sitting in this hall. I am going to present you the significant achievements of the plant genetic resources discipline. So the mandate of the plant genetic resources education, as you know, is the assessment of genetic diversity, which is unique to India, and to know about the management and systematic utilization and conservation of the plant genetic resources. So this year we have 5 MSc students who have submitted their thesis and will be getting
Madam,
degree.
just a minute. Dr. Veena, just a minute. Let it be presented on the screen. It's not coming on the screen. Just a minute.
Oh. क्या हुआ? अभी भी नहीं आया। Yes. We can start now. Let's start. Okay, so this is the mandate of the Plant Genetic Resources Division. And this year, 5 of our MSc students are getting degree, and 4 of the PhD students will be getting degree. One student, Miss Chao Shu Hathaway, she has submitted last year, but later on, so she will be getting degree this time only. So the first student, Lateef Bashir, worked with the able guidance of Dr. Sandeep Kumar on the genome-wide association studies to identify QTNs for quality traits in bread wheat under heat stress conditions. And he has selected 126 genotypes of this and grown at 3 different locations under heat stress and normal conditions. And the major findings for different yield-related parameters and biochemical parameters have resulted that there are 8 accessions which were high yielding, 5 for protein, 4 for amylose, 4 for iron, and 3 for zinc. And second student, Miss Thandral, she worked with the able guidance of Dr. D.P. Sanwal with habitat prediction and mapping of crop wild relatives for Vigna species in India using bioclim model, because the climate change is there. So we have to predict how the climate is going to affect our biodiversity, especially the crop wide relatives. So she has selected all the germplasm which was collected and conserved in the National Gene Bank, and has shown that using the different diversity maps for all these species and the bioclim method, the conclusions are that their future areas for exploration and collection should be the Western Ghats, and the various species for different tolerance to biotic and abiotic stresses she has identified on the basis of the data which was analyzed by her using different parameters. Then the third one is Rinki Reshma Panda, who worked with Dr. Sandhya Gupta for the development of cryopreservation protocol for Heterocarpus lechuza, an indigenous underutilized fruit crop. And the main objectives were that to identify the taxonomic key for field characters and for standardization of the cryopreservation protocols. So she has ably identified several characters based on the fruits and seeds for identification of the sample as they are received at the gene bank, as well as in the field for the collectors. And for the cryopreservation protocol, she has used different method, the V-cryoplate method. And this V-cryoplate method based on the different shoot media for shoot multiplication. And she has concluded the taxonomic key and then cryopreservation of shoot tips isolated from cultures, cold hardened for 1 in BOD condition, they gave the highest growth rate. And V-cryoplate method can be used successfully for the cryopreservation of the Heterocarpus lechuza. Then fourth student, Shashank SG, worked with Dr. Sherry Jacob for genome-wide association studies to identify MTAs for better seedling establishment. And he has used 145 accessions which were conserved in the gene bank from 19 states, and have shown that there were 2 different significant MTAs along with the major candidate genes for seed weight, and they were on 7D chromosome. Then for root angle, the MTA was identified on chromosome number 6A. And like this, he has, based on all the parameters, 1 accession IC 252790, he has identified as a promising accession for higher CL and lower plant height, along with other desirable traits for seedling establishment. And the 5th student, Sridhar, he worked with Dr. K.K. Gangopadhyay for the taxonomic delineation of Solanum melanogena var in Canum from the cultivated type Solanum melanogena var in Sanum. This is the species complex through morphological and
using molecular taxonomy. And he has used 89 different accessions, 80 of the Solanum melanogena var incanum and 9 of the other species. But because of the high gene flow in between the nature, he cannot this relationship between the wild and cultivated types, so that gene flow was very high and there was no clear-cut demarcation which can be used to delineate the two different species. Now coming to the PhD student, Chaoshu Hutwe, she worked with Dr. Anuradha Agarwal for changes in transcriptomes during cryopreservation stages of banana meristem. She has used two different genotypes of banana for these studies, Williams and Burharji, and have used 3 different cryopreservation techniques: droplet vitrification, V-cryoplate, and D-cryoplate. And based on these 2 different cultivars, they all gave good results with all the 3 methodologies. So it can be concluded that any of the 3 cryopreservation approaches can be used for cryobanking of the proliferating meristems of Musa. And these cultivars, they have given good shoot tip, good results with the molecular characterization also. Then second student, Deepak DA, he has also worked with Dr. Anuradha Agarwal for in vitro clonal propagation and cryopreservation protocols, development in Piper species for germplasm conservation. And he has also used the nodal segments for explant and have shown that basal media supplemented with 0.5 mg per liter of BAP was most appropriate for producing the highest number of nodes and shoot length in Piper nigrum, and a ratio of 1.6.5 number of nodes shoots were obtained using this material, and it can be used for the further cryopreservation. M. Shankar, again, he worked with Dr. Anuradha Agarwal, and he worked on the pollen cryopreservation and cross-compatibility studies to overcome wild hybridization barriers in selected legumes. He has selected 2 different legumes, Vigna and Lathyrus, 2 different species of Vigna and 2 different species of Lathyrus, and have studied the different pollen stigma interactions. And based on his study, it was shown that the interspecific crosses and pollen pistil interaction reveals that cross combinations of Vexillata and Unguiculata shows the pre-fertilization barrier, while in other 3, they were the post-fertilization barrier. So this is a review of his work in the Vigna Unguiculata cross Vexillata and reverse cross. And this is in the Lathyrus sativus and Ciceria. So he has first reported for the cryopreservation of pollen in wild species, and it was successful as the more than 90% of the pollen viability was observed. And using this technique, once the pollens are viable, they can be used for further crop improvement programs. And then embryo rescue attempts were also made, and they showed that the regrowths were shown, but later on they started to dry on different media when tested. And Rakesh Kumar Bhairava, an in-service candidate, he worked with Dr. MC Yadav with assessment of genetic variation and allele mining for quality traits in aromatic rice. He has used different 92 rice land races and with 4 checks like Badshah Bhog, Kala Namak, P1176 and Tarauri Basmati. And he has studied the morphological characters in the different traits and grouping of aromatic rice using 14 qualitative traits, and then population structure of the rice land races. And then finally, he has concludes that there is a positive correlation between the grain length and 1000 seed weight, and positive correlation for culm length and plant height. And in the present study, GS3 gene, which was dominated allele A, was found only in 2 aromatic rice land races. So this was all about the research achievements. Now our faculty, 42 faculty, 20 research guides, and total 25 PhD students and 7 MSc students are pursuing their studies regarding honors and award. 3 of our students have got Dr. K.L. Mehra Memorial Award by Indian Society of Plant Genetic Resources. Dr. Monica Singh, our faculty, has got Young Women Scientist Award from the in Agricultural Biotechnology by Royal Association of Science-led Socio-Cultural Advancements. And then one of our student, Anil Patidar, in last year he has submitted. This year he has got Best Paper Award from the ISPGR. Then in different seminar symposias, faculty has presented their results and best oral presentation for two faculty, Dr. Shari Jacob and Ira Vaidya Malhotra, they have got best oral presentation. And all our students, they who have presented best poster, 7 students have got the best poster presentation award at different seminar and symposias. Then regarding the sports meet at IRI, our students have also played very well and have got different different events have got awarded. Then the students were also exposed to the exhibition which was organized on the occasion of Journal Body Meeting of International Treaty on Plant Genetic Resources, which was held in Pullman Hotel last year in September 19 to 26. So all the students, they have visited and they were apprised of the variable diversity available in India, and they have got a chance to meet the international people who are working on the plant genetic resources. So regarding the student placement, we have 3 of our students have been placed, one in ARS and one in agricultural officer, and Shubham Dev has got in bank. Research publication, total 64 publication from the students research, and 5 main publication from the students and 2 practical manuals. And now I thank from all my students and from my all faculty to the Director IRI, Dean Madam, and Joint Director Research, Joint Director Education for supporting the PG education in the plant genetic resources discipline. Thank you.
Thank you. Thank you very much, Dr. Veena. And in time, may I now request Nika to make a presentation of seed science and technology, discipline of science and technology. Dr. Monica.
A very good morning to everyone. So a very good morning to all present here. Thank you, Dadlani ma'am, for inviting me for the presentation from Seed Science and Technology discipline. So this is a brief mandate of the discipline. We have a total of 22 faculty well represented through outreach institutes as well. And out of these 22, 14 are madam eligible for MSc guidance and 11 for PhD guidance now. For the Seed Science and Technology students, we have on roll 65 students, out of which 13 are the degree recipients for this convocation. This is a list of the 8 MSc degree recipients. And these are the 5 degree recipients. So I will begin the salient findings of these students theme-wise. So the first theme on seed production technology has 3 PhD and 2 MSc recipients. Beginning with the first one on PhD. So Ms. Ashwini worked on comparative analysis of planting value of conventional and Indian quality mustard produced under heat stress conditions. She worked on a total of 19 mustard varieties comprising of conventional, single zero and double zero genotypes. And the trial was conducted in 4 sets of temperature across various sowing dates. Very beautifully, she has recorded all the characters ranging from phenotypic, yield attributing, taking the seed quality parameters, and successfully she was able to group them into different categories from susceptible up to the tolerant genotypes. And she reported that the susceptible genotypes were majority of the double zero genotypes, which showed less protective mechanism under high temperature treatments. And the most appropriate sowing date for desired yield and quality was reported to be mid-October for the experimental region. The second PhD under this theme area was from Mr. Debashish Paul, Physico-chemical basis of hard seededness in mung bean, wherein he studied the seed coat permeability pattern in hard and normal seed genotypes and worked out all the biochemical and the molecular markers associated with the same. So this is a list of the material and methods which he has worked on very elaboratively. And this is a brief overview of the results reported by Mr. Debashish. The results were very clear when it was taken under the scanning electron microscopy. It has very clearly indicated the differences between the hard and the non-hard genotypes. And so he reported that imbibition time was higher in the Kharif produced seeds. And finally, ultimate reporting was that the summer season was the desired season for production of quality seeds of identified hard-seeded mung bean varieties. The third PhD student under this particular theme area was Mr. Jorigal Lakshman. He studied the feasibility studies on tomato hybrid seed production under Nadran Plains, wherein he took the 3 hybrids along with the respective male and the female parents, evaluated them under various growing seasons and 3 growing conditions: polyhouse, shade house, and open field. And he recorded a range of observations and could successfully develop the marker profiles for all the parental lines along with the hybrids using various SSS primers. So he reported that for achieving higher fruit set, seed set, and better seed quality, fresh pollen should be used for pollination until the 2nd day of emasculation. Rabi season was the most favorable for seed production, and polyhouse conditions were found to be most suitable for hybrid seed production under Northern Indian plains. Coming to the 2 MSc students under this particular theme area, ma'am, these both students MSc are from IRI Jharkhand, who have been guided by Dr. Priya Ranjan Kumar from the Outreach Institute. So first is Miss Bhavani Kumari. She compared the hard and the soft wheat genotypes for seed traits and their susceptibility to storage pests, wherein she took some soft wheat lines also for the evaluation, and she used in various instruments to measure the seed parameters. And for insect infestation studies and oviposition studies, she used the facilities which are available at the entomology division of our institute. So she reported that well-defined differences between the hard and the soft wheat varieties as expected, and number of egg plugs were reported to be higher in the soft varieties than the hard varieties. So also she reported that grain hardness is negatively correlated with zinc content and positively correlated with the iron content of the grain, and that soft varieties of wheat had higher seed germination and seedling vigor. And as expected, the percent seed damage, percent wheat loss, and the number of adult weevils after 60 days of infestation were higher in the soft varieties than the hard varieties. The second MSc student from our Outreach Institute, Mr. Priyabrata Sahu, he worked on the effectiveness of plant growth regulators in improving lodging resistance and their effect on seed traits in wheat. Here I would like to acknowledge the help given from the genetics division for the evaluation of the study. So he used various plant growth regulators, and different time of application was used for 3 wheat varieties which the student had used. Various observations were recorded for the purpose, and this is his salient findings. After applying the various plant growth regulators, he reported that the application of ethifone at the rate of 600 ppm at 60 days after sowing performed best. Ma'am, coming to the second theme area, seed quality enhancement, we have 2 degree recipients under this particular theme area. The first is the PhD, Miss Dhanya. She worked on enhancing zinc use efficiency in chickpea through seed treatments with the broad objectives for standardizing the seed priming and coating protocol and evaluating the effect of zinc application. For the material, she studied both the Desi as well as the Kabuli genotypes, and different seed coating parameters were taken up by Ms. Dhanya. So this is an overview of the results which she has presented both for the Desi and the Kabuli genotypes using different treatments. And the final finding reported by her was that hydropriming for 3 hours in Desi and 4 hours in Kabuli types of chickpea proved to be efficient treatment. And moreover, she also reported that seed coating using zinc nanoclay polymer composite could be done at a rate of 1:2. For the second thesis under this particular theme area is our MSc student, Mr. Pumani, who also very well competed for the gold medal. So his participation is well acknowledged, Pumani. The title of the thesis is Effective Priming Techniques on Seed Quality Enhancement of Lentil and Their Efficacy Under Heat Stress. So with the broad objective to standardize the protocols for chemical priming and thermopriming using 3 lentil varieties and then using humic acid priming as well as thermopriming. So this is an overview of the results reported by Mr. Pumani under various normal as well as heat stress condition for various seed quality parameters. And his ultimate finding was that priming with humic acid at the rate of 600 ppm for 18 hours and thermopriming at 32 degrees Celsius for 24 hours was found to be the most effective technique to improve the seed quality in lentil. And also he reported that under heat stress, membrane integrity was disturbed, which was rectified by seed priming. Ma'am, we come to the third theme area. This is seed pathology. Under this, we have 3 degree recipients. One is PhD and 2 are MSc. So the first on the PhD is Mr. Balachandran Gowda. He worked on the effect of chili fruit rot on seed quality and development of integrated eco-friendly management strategies. For the material and methods, he used a very good comparison between both the resistant as well as the susceptible variety. This is the pictorial representation very clearly indicating the differences between the healthy and the diseased seeds. So this shows the validation of the various markers which was done by Mr. Balachandran. And he reported that in dual culture technique, Trichoderma viridi and Trichoderma hertzianum were having better antagonistic effect due to their aggressiveness and fast approach, and that a combination of systemic and contact gave good results. Moreover, seed treatment and seedling dip of carbendazim and mancozeb, and among biological treatments, seed treatment, seedling dip, and one spray of Triticum viridi was found to be very effective. Coming to the next 2 theses under the seed pathology, we have 2 MSc students guided by our young faculty, Dr. Nagmani Sandra. Here, Mr. Shrikant worked on the effect of soybean yellow mottled virus on yield and seed quality parameters of soybean. So the student evaluated various soybean varieties and then also regularly reported all the symptoms, and the disease scoring was done, and the confirmation of virus was also done through ELISA and PCR. This is a broad overview of the results which Shrikant had done under the supervision of Madam Nagmani. So this is the salient findings. Most common type of symptoms which he observed were mild chlorotic spots, mosaic, and mild mottling. And very clearly, he could group the soybean varieties into different categories based on disease scoring against the virus. The second study, MSc, again under Dr. Nagmani, was by Mr. Barla Madhusudan. He worked on the effect of mosaic and mottled disease on yield and seed quality of parameters of soybean. Mr. Barla will be receiving his degree in absentia because of certain other personal reasons. So he, with the broad objectives to identify the virus associated with both the diseases and to evaluate the effect on disease and seed quality parameters, he worked on very elaborative 34 soybean varieties using different methodologies. And this is a broad overview of the results he reported. And the salient findings were he was able to successfully identify the major viruses to be associated with mosaic and mottling disease. And multiple viral infection was also reported at detectable levels and predominantly observed in 6 cultivars. And he could also detect the presence of viruses and confirm them through PCR and RT-PCR from the positive leaf samples. Coming to the 4th theme area, very interesting studies coming from the seed science, the varietal identification and characterization. So in addition to the basic morphological and biochemical parameters, ma'am, now the students are also using image analysis and machine learning methods. Coming to the first one, that is the characterization of tomato genotypes using morphological and biochemical parameters by Mr. Rajan Vishal Praveen Kumar with the broad objectives. For this, he took the experimental material comprising total of 32 genotypes of tomato. And for this, the help rendered by the Division of Vegetable Science of our institute is duly acknowledged. So this is the 32 tomato genotypes which he characterized using various morphological markers. And he found that 39 traits showed significant variation among the genotypes and could also discover the novel type of trichomes in the study. The second MSc is the identification of rice varieties using image analysis and machine learning methods by Mr. Abhik Roy. So he attempted to distinguish varieties based on morphological and then tried to complement them with the new characters and certain images from machine learning methods. So the experimental material comprised of 16 basmati and non-basmati type of rice varieties. So this is a broad overview of the results wherein he used certain cluster analysis and different stages for field photography is also depicted. And the salient findings reported that among the morphometric characteristics, maximum variance was observed in the area and volume of seeds. And he could successfully identify new characteristics, and also reported that for genetically close varieties, we can use specific wavelength sensors for the study. So it is reported that the image analysis and machine learning methods can be a viable alternative approach for the identification of rice varieties. Continuing the trend, Mr. Hari Prasad SK also worked on using multispectral image analysis to evaluate seed morphological and quality traits in diverse rice accessions, again under one of our young faculty, Mr. Manjunath Prasad, wherein Mr. Hari Prasad evaluated 886 diverse accessions from the Rice Genome Project and the Rice Diversity Panel. So he used various methods like multispectral imaging using Videometer Lab instrument and analyze the phenotypic data using various softwares. And this is how he could well report the variation in the seed morphological traits, also the variation in the concentration of the color compounds. And his salient finding was that MSI is a potential method for measuring seed morphological traits, since significant variation was observed for all the seed morphological traits. So ma'am, I'm happy to share that now we are trying to complement the morphological dust testing with the new upcoming technologies, for which we have certain projects also in our division now. This comes to the end of the research achievements of the students. With respect to publication, we are also trying to publish in more than 10 NAS rating journals. This is a list of student publications. Now they are working diligently to get the papers acceptance in due course of time. Certain book chapters are also reported. With respect to student placement, ma'am, very proudly we share with you that all the 4 seats for the ARS discipline in seed science and technology were backed this year by all our 4 divisional students. So hearty congratulations to all the students. Apart from that, we have students in various civil services exam, as well as in as technical officers, managers in various organizations across the country. So the presence is being felt everywhere. So this is a list of training and workshop organized in our division. One startup like a user meet national workshop was organized, and training program, which is a regular feature. This is a list of PG activities. Ma'am, the faculty has really worked very hard to design a diploma course. Okay, ma'am. So this is the diploma course and the BSMA extension activities reported from Pusa, Bihar.
Okay, you may conclude, Dr.
Okay, ma'am. So last slide, ma'am, honors and awards by the students, by the faculty, and the glimpses of PG activities in the division. And this is the last slide, which I also consider to be important. Happy faces are always welcome. Thank you, ma'am. Thank you.
Thank you very much. Thank you very much, Dr. Monica. We have to maintain time.
Thank you, ma'am.
In fact, maintain time as a. So I'm just. Okay. Now, 15 minutes, right? 10 minutes. Right. Thank you all. Thank you very much. Particularly the 3 professors, Professor Anju, Professor Veena, and Professor Monica, for making excellent presentations of very good work done by the PhD and MSc students for the thesis work. I can start, rather not do that. I am actually looking at it that the students who are getting degree this year or one of these years are our future researchers. So the purpose of this thesis programs or thesis work at MSc and PhD level is primarily to prepare the students for taking means planning, executing, and analyzing and concluding the research programs on their own in future when they take up some research or scientific positions. And from that point of view, I mean, there cannot be any better place than IARI. My compliments once again for overseeing the so many faculties and such able faculty of IARI PG school. One thing that I was looking for, because I'm revisiting the research programs of this institute after a gap. So I am more to look for new areas, new dimensions. So I would have felt happier if there was a little more focus on the crops that we are at national level, we are trying to promote, that is the millets. I actually didn't find any work on any of the millets, any of the coarse grain crops in this crop improvement program. So that is one little, not dissatisfaction, but I was really actually looking for that. But then I found that there were some students who have worked on a less studied crop like horse ground in the crop improvement program in the division of genetics plant breeding, which is a very positive sign and very promising sign, which I appreciate. Similarly, in seed science and technology, I found that besides the routine work, there is quite a bit of routine work actually, but besides those, there were also some new dimensions, whether that is in terms of seed coating to manage minor or major element deficiencies, for example, zinc utilization or zinc deficiencies, or using multispectral image analysis and machine learning for variety identification. I was very happy to see two students. Actually, it was my good fortune to be examiner of one of the students. And I found that there are two theses. One was on rice and in tomato. Very good beginning. I compliment you, Madam Professor, for that. IRI should be the place where new and difficult problems will be taken, and the students will be prepared for that. My congratulations to the two students who have won the gold medals yesterday from the School of Crop Improvement, and my compliments to their advisory committee and the chairpersons for guiding them to undertake very meaningful programs very successfully, and also preparing those students to make effective presentation. That is an art. It requires a skill. And when Madam Professor Anju Mahindru made presentation for the genetics division, which had 7 theses in abiotic stress tolerance, 8 in bio, 7 on quality traits, 6 on other economic traits, and she could beautifully make presentation of each one of the theses without losing the main essence. For that, my special compliments to you. Very good. It's no wonder that 2 students from genetics division got gold medal, because these are all part of their training. It is not just thesis, of course, that is the main focus, but also to communicate in science, scientific communication without any unnecessary things, just to be to the point and in time is also a skill which needs to be imparted to them. And you have done that very effectively. That my compliments for that. There are also many awards and recognition won by your faculty as well as your students. That also recognitions were won by the students of other 2 disciplines. For that, my compliments to Professor Veena Gupta as well as Professor Monica Joshi. For NBPGR, I was also happy to see that you also encourage your students to take active part in the sports activities, and they've done very well. That's all. A student's development, all-round development. And it is good that you are encouraging them to take parts in the sports activities as well. And you are igniting that on Convocation Day presentation. My compliments for that. Some of the things that I found very important is that in case, for example, if I come to a little bit on the discipline wise, I'll just take 3 minutes for 3 disciplines. I was very happy that the focus of variety development in the division of genetics plant breeding is very rightly focused on abiotic and biotic stress tolerance. And secondly, on quality improvement. These two are really the need of the hour, not just yield. That was our first focus. In the initial years, when the availability of food grain was the main problem, that was the main focus of research as well. And now that the nutrition security and meeting the challenges of climate change is our main focus, therefore, I'm very happy to see that the large number of programs of the Division of Genetics and Plant Breeding are focused on these. Also, complementing that are the theses coming from the discipline of plant genetic resources, plant genetic resources, where they have taken a large number of genotypes for characterization, because the weakness that we have a wealth of plant genetic resources, but these are not fully characterized. So I'm very happy, madam, that through your research programs as well as through the student research, you are taking it up. That you also mentioned find from where you are getting for terminal heat drop and all, which is quite expected. In fact, maybe if the selection of germplasm in the beginning is done, keeping that in mind, that will make it even more focused, narrow it down. Similarly, I compliment you, madam, for giving the research programs to students on every aspect of seed science and technology, because it's a multidisciplinary subject where you have focused on production as well as seed health, incorporating the variety identification, and I was very happy to see 2 MSc theses coming from IRI Jharkhand campus. My special compliments to Dr. Priyaranjan, who has guided these students, because I can understand the many constraints that these students must have faced. And in spite of that, they have done some work, which may not be in terms of very high scientific output, may not be that impressive, but those are from the titles, what I could see, and a little bit in the abstract, very important in terms of the local problems. For example, the storage insect pest infestation in the soft grain wheat is a problem in the eastern part of the country. And that was the purpose of having these campuses in different regions, so that IRI can also address, besides the national problems, which is the first mandate of IRI, but it should also expand its responsibilities and take up the problems of different regions. So I'm very happy to see that in the Division of Seed Science and Technology. Overall, a very satisfying morning for me to see that IRI continues to do excellent work, and the students of IRI had always been the backbone of IRI's research programs, and they still continue to be so. I'm very happy to see that, and my compliments and congratulations to all the students who are going to receive degree this year, and also to all those students who are pursuing their studies at IRI at present, and to all the faculty members who are supporting this. And thank you very much, Madam Dean, Dr. Anupama, Professor Anupama, for giving me this opportunity to be with you this morning. Thank you all.
Thank you, madam, for a very comprehensive evaluation of all the three presentations. We are indeed honored to listen to your words, and we seek your guidance in all the years to come. Ma'am, now it's time to felicitate the chairperson. So may I request Dean Madam for kind felicitation of our dear Dr. Dadlani. So as convener of the present professor's presentation, it is my humble privilege to propose a formal vote of thanks. So on behalf of the director and dean, we all are thankful to the chairperson, Dr. Dadlani, for her kind acceptance of the invitation and conducting the proceedings of the session in a very comprehensive manner. Ma'am, the suggestions given by you will be duly taken care of, and we always look forward to your guidance in all the years to come. My sincere thanks are also due to our director, sir, and indeed our Dean, Madam Dr. Anupama Singh, for her constant support and guidance throughout and taking care of every minute detail for the success of the program. Thank you, ma'am. I also wish to place on record my thanks to all the committees which were constituted for the convocation for their kind cooperation and coordination throughout. Thanks are also due to the Associate Dean along with his dedicated team from the PG School and Dean's Office for providing all logistic support. Most importantly, I profusely thank the professors from the School of Crop Improvement for their very elaborative, still crisp presentations for the PG students for the respective disciplines. Thank you one and all. I now invite you all for a cup of tea, and we'll assemble at, ma'am, what time? Please assemble back at 11 a.m. for the next School of Crop Protection. I request the professors of crop protection to kindly upload if they haven't done so. Thank you, ma'am. ये किसका है? ये पेन ड्राइव है कि ये क्या है? पेन ड्राइव है किसी का? अच्छा,
मैम एक मिनट रुको 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 रुको। ये पॉइंटर इस साइड लगा। एक मिनट मुझे जगह नहीं।
मुझे थोड़ा सा देखना है कि खुल रहा है कि नहीं, क्योंकि प्रॉब्लम हो रहा था।
Hello, check, hello.
you all had. So now we will move on to the session 2 of today, that is the School of Crop Protection. The chairperson for today's second session on crop protection, Professor Appa Rao Podail, Senior Professor, Department of Plant Science, University of Hyderabad, is amongst us today. It's an honor, sir, for us to have you here. May I kindly request you, sir, for gracing the dais along with our Dean, Madam Dr. Anupama Singh, please. A round of applause, please. Thank you, sir. May I now request Dean ma'am to kindly welcome Dr. Appa Rao with a bouquet of flowers. I now invite Dean ma'am for her welcome address and a brief introduction of the chairperson to the August gathering.
Thank you, Dr. Monica, and thank you very much, my respected professor colleagues, the faculty IRI, and the students who have gathered here. Although I, through this podium, again request each one of you on behalf of director and on my own behalf, those who may be online also, to kindly, find time for this convocation week out of your busy schedules and to grace the occasions, because as I already stated yesterday also, that whatever we are, the way we are responding, that shows our respect to the system, to the organizers, to the event, and to those who are gracing the dais. So I again request on behalf of director and on my own behalf to find time and join us in auditorium. Thank you very much. So as Monica has given me this duty, so I again welcome you all. Respected Professor Dr. Appa Rao Podail ji, the chairperson of this session, we are, and the faculty are my dear students. We are here to have, after this first session of crop improvement, now the important session of the School of Crop Protection. So we will have here the presentations by the professors of the disciplines under School of Crop Protections from agricultural chemicals, entomology, nematology, and plant pathology. The presentations are based on 64 theses comprising 32 MSc and 32 PhDs. Although, sir, this will be representing a major chunk of the research programs which are going on in the institute and across our mandated areas. But I would like to, seek an excuse on that count, that it will not represent the entire spectrum actually in these few minutes. So we will try to actually make it more holistic as much as possible, based on your advice. So, I welcome our Chairman Academic Council and Director IRI on this dais. Thank you very much, sir, for joining us. So, without saying much now that, because we have to start our session, thank you very much. And now may I request Director, sir, Dr. A.K. Singh ji, to kindly introduce Dr. Appa Rao to the audience. Thank you, sir.
Very good morning once again, and I would like to extend a warm welcome to Professor Appara Kodli, a renowned plant molecular biologist, and he has served as the Vice Chancellor of the University of Hyderabad, one of the most prestigious universities. And his area of work is particularly in the plant microbe interaction, and a special reference to legumes. And that's the area where Dr. Rao has made enormous contribution. He's elected fellow of all the scientific academies of India, which is a rare distinction to have the fellowship of all the academies. And also is recipient of many prestigious awards, including J.C. Bose Fellowship and Tata Innovation Fellowship, K.C. Mehta Memorial Award in Crop Protection from National Academy of Agricultural Sciences, our own academy, and also Rangaswamy Award from Agricultural Microbiologists Association of Microbiologists of India. He has over 33 years of teaching and research experience and an excellent teacher, and has been associated with guiding many students for their PhD program. We are indeed pleased to have you here, sir, and thank you for sparing your valuable time. And we look forward for the presentation. Thank you.
Thank you, sir, for your kind introduction of the chairperson. So as convener for the session and with the permission of the chair, I would wish to apprise the House regarding the modalities for the presentation for School of Crop Protection. So for session 2, as already briefed by Dean Ma'am, there are 4 presentations from the professors of the following disciplines: agricultural chemicals, entomology, nematology, and plant pathology. For all the 4 disciplines, the time allocation is based on the number of thesis abstracts for each discipline, with an additional 3 minutes each. So for the ease of the presenter, yellow light will blink 2 minutes prior to the allocated time, and a red light will blink at the end of the allotted time. So I now hand over the session to the respected chairperson for his kind remarks and to conduct the further proceedings of the session. Dr. Apparao, please.
Good morning, everybody. Especially, I would like to thank the director, Professor Singh, and the joint director, Dr. Anupama, all the faculty for thinking about me to join you on this very special occasion where you are organizing your convocation, the 61st convocation. I'm very happy to be part of this particular event, especially because it has given me an opportunity to look at the details of work that have been carried out under the School of Crop Protection. And although my area may be towards more on my plant microbe interactions, we get to interact with people in the areas of agricultural chemicals. We also get to interact with people from entomology. And in fact, I have good friends in nematology in IRI, and definitely a good number of friends in plant pathology. Like that, I am happy that all the 4 divisions, the 64 graduates, including 50-50 of masters and PhD students, work is being presented. But I know the time that you have, the presenters whosoever is going to make the presentation, the time you have with you is very limited. I'm sure it is not easy for you to even summarize those things in such a short span of time. I tried my best to understand from the abstracts that have been shared with me, and to get into the full details of it, even I would require a lot of time to understand more of what you have been doing. At a cursory look, I could appreciate the work that has been done, and I'm looking forward to listen to the presenters, I'm sure it could be the dean of the, it could be the head of the division or somebody nominated by head of the division. Oh, yeah, right. So please follow your time instructions, because next session may be there. So we'll have to complete it. And I think Monica is going to take care of that, that part of it. And I'm happy to be with all of you and listen to the work that has been carried out under the School of Crop Protection. Thank you very much.
So thank you, Dr. Apparao. With your permission, sir, may we now start the presentation. So I first call upon Professor Neera Singh from the discipline of Agriculture Chemicals to kindly make her presentation for the day.
Yes. Very good morning to all. Respected Chairman, Director Sir, Dean Ma'am, my worthy faculty and dear students. I am here to present the significant educational achievement of the students of discipline of agricultural chemicals. The division have 4 mandates: the development of agrochemicals, their formulation, safety evaluation, and human resource development. We have 15 faculty members and 45 students on roll. 4 PhD students, Prakash, Shannon, Rohan, and Namrata, and 6 MSc students, Soumyajit, Joydeep, Dharni, Brendan, Jeetra, and Atanu, will be getting their degrees during the 61st convocation. And the theme areas of research addressed by them can be grouped into 2 categories. So I will start with the first one. Prakash developed trichoderma viridi biocontrol formulation for the management of chickpea wilt disease. Initially, he isolated 4 nanocellulose using biological and alkaline methods. And the biologically method produced the good quality nanocellulose, and it was used to prepare the composites, and sugarcane bagasse composite was the best for maximum water retention capacity. Therefore, it was used to prepare the trichoderma viridi formulation, 5 powder formulation and 9 tablet formulation. Both type of formulation were better than the carbendazim and the talc-based formulation, but the seed treatment was the better formulation rather than the direct application. And these results were further confirmed by in vivo field efficacy studies. So the formulation have the promising in managing the disease. Shannon developed the pyrazole derivative for the management of maize disease, the fungicide. She developed 54 compounds and characterized them and evaluated them in vitro as well as in vivo. And some of the compounds, they were promising, and especially for the control of Bacillus bifoliaris mairis. We can see it showed 58% control. However, these compounds were very good nitrification inhibitor, both in vitro and vivo studies. We can say they were better than the DCD, and they significantly lowered the nitrous oxide and methane emission. So these compounds, they are promising, more promising as a nitrification inhibitor. The Rohan Sarkar evaluated the prebiotic potential of yum bean, that is the inulin, and the citrus peel, that is the pectin. Inulin was extracted from yum bean by different methods, standardized the protocol for the extraction, and prebiotic activity was evaluated against Lactobacillus fermentus, and we can see the inulin was 58% better in promoting the growth than glucose. The glucose release from inulin was slower, around 2.6 times than the starch, And the hydrolyzed inulin with degree of polymerization 3 was optimum for prebiotic growth of these bacteria. And a human consumption, a formulation, the solid and liquid were prepared. Similarly, the citrus peel, orange, mossmi, and quinoa were used for essential oil and other bioactive compounds. The essential oil was formulation showed very good antifungal activity against a human pathogen. Carotenoid and phenolics, good antioxidant compound, and the pectin extracted from the orange peel content was more. And the pectin showed very good prebiotic activity, around 55% increase than the glucose. And similarly, the glucose release from the hydrolysis was better than that of starch. And the leftover waste was successfully used for bioethanol production, and that is a significant achievement. The Somajit Ghoshal prepared the water soluble formulation of the inulin from a marigold, which is a non-edible flower. Lutein was extracted from the marigold flower by different methods, and its extraction was optimized and it was characterized. 27 water-soluble emulsion formulations were prepared and they were evaluated physicochemically. And finally, 3 microemulsions were converted into powder formulation and they were water-soluble. These lutein formulations, they showed very good antioxidant activity and they were bioavailable in a simulated human digestive system. The Joydeep evaluated the benzamine derivative as antifungal compounds. He prepared 27 compounds and evaluated against Rhizoctonia solani and Macrophomina physiolana, and we can say that here some of the compounds, they were even better than the hexaconazole, the compound used for its control. Structure activity relationship identified the group for the optimum activity of this series of compounds. Dharini prepared the slow release formulation of a insecticide, chlorantraniliprole. The 8 polymer were prepared, characterized, and they were used for encapsulation of insecticide. And we can see that the encapsulation increased the, so the half-life of release from the control release formulation was extended by 3 to 10 times by encapsulation. So your insecticide will be available for longer period in the soil. And we can see the in vitro evaluation study suggested that one of the compound was better than the commercially available coragen formulation. Jeetram evaluated the essential oil from garlic for the control of root knot nematodes. 18 garlic selection from IRI were profiled for oil content and chemical composition. We can see the Bhima Omkar had the highest oil content. And chemically, these are the essential oil contain the dihalide, trisulfide, and disulfide as the major constituent. And the in vitro and in vivo activities suggested that though these compounds are not as good as our chemical pesticide, but certainly they can be used for the management of root knot nematode in organic agriculture. This is the second theme area, and the Namrata evaluated molecular imprinted polymers for the extraction of tricyclazole and clothionidine from environmental matrices. 25 MIPs and 25 NIPs were prepared for each of the compound, and they were characterized. The tricyclazole binding efficiency of MIP or more, 52.5 to 94%, while NIP, they are non-specific. 5 MIPs were promising in nature, and parameter for optimum absorption were standardized. Similarly, for clothianidin, the MIPs were better absorbent and then NIPs. 7 were promising in nature and parameters were standardized. And these MIPs gave acceptable recovery of tricyclazole from rice and water sample, and for clothianidin from rice, honey, and tea samples. The Atheno studied the fate of an antibiotic in soil and water system. The persistence of antibiotic in soil was mainly attributed to soil organic carbon and the moisture content and microbial population in the soil, while in water it was affected by soil pH and dissolved organic matter content. The absorption and mobility studies suggested that the antibiotic has leaching potential in the insectisol soil, so it should be further evaluated. The Brendan prepared the metal organic framework for the removal of fungicide tricyclazole from water sample. The 25 MOFs were virtually screened for tricyclazole binding. And based on the simulation studies, the 3 best MOF were prepared in the lab and they were evaluated for tricyclazole absorption. And one MOF MIL 53 was the best for removal of this tricyclazole from water sample. And the interaction studies between tricyclazole and MOF suggested that pi state and pi lone pair interaction were mainly responsible for its absorption behavior. And then other activities, our student took a study tour to Himachal Pradesh and visited the institution of importance. Regarding the student research, out of total 141 publications from the division, 17 originated from the student research, and they were published in high impact journal. We can see more than 10 now stating the student have published 6 research papers. Students were honored for their academic as well as extracurricular activity. Ashish, Roshi, Partha, Rajni, and Someji were the awarded student. And our students are well placed. The 2, Devdas and Sutanva, has already joined their respective jobs, while Kailash, Harshang, and Rakesh, which have 3 or the multiple offers with them, will be joining ARS once they are getting their selection letters. Finally, I congratulate and thank all the degree recipient students, their respective chairmen, my faculty, all students for helping out in this presentation. And finally, I am thankful to Director Sir, Dean Ma'am, all administrative support, all scientists of the division who contributed to the research of these students, BG School, and each one of you for listening to my presentation. Thank you.
Thank you, Dr. Neera. Sir, with your permission, may I now call upon the next presenter, Dr. Devjani Dev, to present for entomology discipline. dignitaries on the dais, my dear colleagues, students, ladies and gentlemen. It is my proud privilege to present the significant achievements of the students of entomology who will be getting their degree this year. The mandate of our division is to conduct basic and strategic research to develop as a center of excellence in research, teaching, and human resources, and to provide services in insect science and technology. This year, 17 of the students will be receiving their degrees, 10 in MSc and 7 in PhD. And these are the various themes under which their work has been different areas of entomology under which the students have conducted their research. First of all, insect systematics. As you all know, Karthik Reddy worked under Dr. P.R. Shashank, and he received IRI Merit Medal yesterday for his MSc work on micro moths of southern Karnataka. His work had two objectives, and he made his collections from 11 different areas of southern Karnataka, collected over 800 specimens, out of which 650 belonged to his group, and he could identify 6 species new to science, established 6 new records for India, 2 genus and 6 species. In addition, he has contributed 28 new records to the fauna of Karnataka. This is how his studies were conducted. A total of 50 species were studied. This is how illustrations were made. More than 800, 900 illustrations were there in his thesis. And he submitted 25 accessions to the GenBank. 21 of them were first records to the GenBank. The second student, Mr. Mowgli Ramaiah, he worked under my guidance on leafhopper species associated with bamboo from India. He also received IRI Merit Medal yesterday for his PhD work. First of all, he compiled a checklist of species which are associated with bamboo. So now currently 35 genera with 91 species have been recorded to be on bamboo. And this includes 10 new species and 12 new geographical records. And of course, 2 first records for India. These are the species, 4 new genera, 10 new species have been described, and 2 new records established for India. And bamboo was also recorded as a new host for off-season survival of mango hoppers. These are the illustrations of each of these species. More than 60 sequences were submitted to NCBI. Targeted genes were MTCO1, histone gene, and 28S rDNA gene. So based on this, phylogenetic tree is based on the MTCO1. And here you can see the new species have been clearly demarcated in this tree. Then integrative taxonomy was used for diagnosing important problems or complexities in identification. And these are very small leafhoppers. So if frequently misidentification is there due to its color polymorphism, and we could solve this problem as well. The third student, Monica Jat, she also worked under me on a very less known subfamily Evacanthini. She also, due to COVID, limited surveys could be made, and she collected around 300 specimens belonging to her group from 11 localities of India, and established 11 new records. These are the species, 29 species in all, which she studied. 1 new genus and 3 new species were described, and all of them have been published in high impact journals. More than 50 sequences were submitted to NCBI, and phylogenetic tree showed that the tribes of this subfamily are paraphyletic in nature. Coming to insect pest management, Pooja Kumari from our Jharkhand campus under the guidance of Jaipal Chaudhary worked on temporal abundance and species diversity of pollinators associated with lychee flowering. Around 20 pollinator species belonging to Hymenoptera, Diptera, and Lepidoptera were identified to be visiting in lychee during flowering. And Pollinator activity was found to be negatively correlated with maximum temperature and positively with relative humidity. The path analysis demonstrated that interrelated factors like temperature, RH, wind velocity had the greatest influence directly and indirectly on the flight activity of the bees. Another student, Sai Padmini, again from our Jharkhand campus, worked under Dr. Jaipal Chaudhary on the population dynamics and bio-intensive management of insect pest complex. She recorded lychee stink bug, gandhi bug, flower weber, and leaf weber as the key pests at flowering stage of lychee. 2 effective management modules were also devised, and after evaluating the benefit cost ratio, the highest returns were observed in case of imidacloprid, imamectin benzoate, thiacloroprid, and fipronil plus fipronil combination. Another intervention she suggested was tree trunk banding with polythene, which was found effective against the crawling nymphs. Kirti MC, an in-service candidate ARS scientist, worked under the guidance of Dr. Sachin Suresho, evaluated eco-friendly pest management techniques for promoting organic cultivation of cauliflower. The various growth parameters were studied for Lipophis erythemi and Plutella xylostella on 5 different cole crops: broccoli, cabbage, cauliflower, nolcol, and mustard. And it was found that intrinsic rate of increase of Lipophis erythemi was lowest on broccoli and highest on mustard. Similarly, for Plutella xylostella, it was highest on mustard and lowest on nolcol. The expected longevity of both the pests were also studied, and in the ascending order it was listed. Now the GGE bioplot graphs showed that cabbage was preferred host for both the pests, while broccoli was least preferred by Lipophis erythemi, and nolcol was found least preferred by Plutella xylostella. He also evaluated certain intercrops. He also listed the various cropping systems for lowering the pest and increasing natural enemies. These are the combinations for each of these pests. And based on highest benefit cost ratio, he found that calendula intercropping system was the best, and it can be recommended for the farmers as well. Priyanshu Pravar worked again from our off campus at Raipur, worked under the guidance of Dr. Murali Bhaskaran. He studied the effect of Caromone gel formulations on biocontrol potential of Trichogramma chelonis on 2 crops, Lepidopteran pests of wheat and chickpea. So application of Octadane gel formulation at 500 ppm reduced the pink stem borer induced dead heart. And also there was a reduction in the white ear symptoms in case of wheat. While in case of chickpea, again, the population of chickpea pod borer larvae was reduced. And salient findings of the laboratory trials was that parasitism by Trichogramma chelonis in Octadane 500 ppm treated eggs ranged from 85 to 85, almost 85%. And similarly, the other Trichogramma japonicum also, there was quite high parasitism because of this Caromone application. Rakshit, he worked under Dr. Sachin Suresho on molecular marker-based assessment of population structure and dispersal potential of Chilomenus sex maculata. He developed and validated various Indel and SSR markers, and he found that 9 SSR and 5 Indel polymorphic markers significantly could differentiate the various populations of Chilomenus, and genetic diversity analysis revealed that north, west, and central zone populations were highly diverse, while moderate level of genetic differentiation was observed in the southern populations and also the northeast population from Barapani. And therefore, it was opined that due to these subpopulations need to be conserved by augmentative release of other area populations in these areas. Migration studies were also conducted. Dispersal pattern of C maculators was also seen, and diffusion coefficient and relative departure rate was again recorded highest for the North Zone populations, followed by West and Central Zone, and lowest in South and East Zone populations. 4 of the students, 2 MSc and 2 PhD, they use certain novel approaches for IPM, that is the recent RNAi technology. V. Rajesh worked under the guidance of Dr. Amalindu Ghosh and worked on silencing key genes for TRPS-PAMI for novel management options. The genes targeted was BTK29A and COL3A1. And these were synthesized in vitro transcription and delivered through oral feeding by incorporating in the artificial diet, and significant mortality was seen. Although significant mortality was there, but no morphological deformities were observed, and silencing of these genes, BTK29A reduced the viral load, but silencing of COL3A increased the viral load, supporting its presumed function in innate immune response. And both the genes can be targeted to induce mortality and impair virus transmission. And this could be a suitable novel genetic target to manage TRPS. Another girl, Thesneen P., she also worked under Dr. Ghosh for her MSc degree and worked on the functional role of key genes of whitefly Bemisia tabaci in begomovirus transmission. Target genes here was B tabaci, TLR3, and TOP1, and silencing of both these genes were seen to induce significant mortality, but again, no morphological deformities. And silencing down-regulated the expression as well as virus copy number. And this also is a novel approach for managing B tabaci and restrict virus transmission. Saurabh Sarkar, for his PhD work under the guidance of Dr. Vinay Kalia, and worked on the characterization of juvenile hormone epoxy hydrolase and related genes through RNAi in Spodoptera litura. This is the basic plan of his work, and developmental abnormalities in final instar larvae were observed after ingesting the dsRNA. Larval mortality was there, intermold deformities were observed, longer larval duration, and reduction in the pupil size was observed after dsRNA ingestion. GS Uma also worked under Dr. Vinay Kalia and screened putative RNAi targets for cotton Aphis gossypi and their functional validation. Again, this is the scheme of her experiments, and the conclusions of her work were silencing of AG-SUC1 and AG-TREC1 genes reduced mortality and also reduced fecundity. And production of dsRNA by bacterial expression is a cost-effective method and equally efficient in production of in vitro synthesis, and it has the potential to be used as a biopesticide. Insect physiology, one student, Reshma R., worked under Dr. Sagar on the effect of heat stress in progeny of Spodoptera frugiperda. 2 hot events, that is 42-degree and 45-degree temperature stress, was applied to the early 4th instar larvae, and then rearing was continued till F3 generation. The points mentioned in red indicates which all factors having interactive effect of heat treatment and generation. So significant reduction and carryover effect on reproductive parameters across generations was observed. And then biochemical response in S. frugiperda exposed to heat stress was also studied. And across generation, there was depletion or accumulation of each metabolites, and impact of parental generation as well as carryover effect on subsequent generation was linear to the frequency of exposure and the temperature. So these are some of the important conclusions. Sublethal temperature administered to larval stage in parental generation induced developmental attributes and reproductive fitness. Then heat stress, impact was increased in a linear trend in relation to temperature and frequency of exposure. The present investigations give a holistic information on recent and evolutionary heat stress history of a given population of an invasive pest, which can be used to predict outcome of management practices under changing climate. One student, Sandeep Kumar, he worked under the guidance of Dr. G.K. Mahapatra he generated susceptibility data of various populations of fall against the commonly used insecticides. And the new insecticides molecules, imamectin benzoate, spinotorum, chloramphenicol, novaluron, were found to be highly effective against fall compared to thiodicarb and lambda cylohydrin. Another aspect he worked on was the molecular studies to target the site mutations in different populations of test insect, Spodoptera frugiperda, and he found the 2 amino acid substitutions, A201S and F290V, were detected in the acetylcholinesterase gene of FOR in the Indian populations. And this confirmed the presence of resistance gene for organophosphates and carbamates in FOR. Impact of temperature on the toxicity was also studied. And the PCA biplot showed that positive temperature coefficient insecticides work better in the Kharif season, that is imamectin benzoate, chloramphenicol, novaluron, and thiocarb, and vice versa. Storage pest management was also worked on. Parveen Pranda, again from Raipur, our off-campus, worked under KC Sharma, and he screened 12 varieties and recorded data on the various parameters. And significant findings was Mahamaya showed maximum tolerance against rice weevil, and various inert dusts were also studied. Fly ash was found to be most effective, followed by diatomaceous earth. And compared to all plant products, custard apple seed powder had maximum efficacy. And no adverse effect of all these inert dust and plant products were there on the germination of rice. Neelkantha Rajarushi worked under Suresh M. Nebapure. Again, toxicity evaluation of contact insecticides against rice weevil. He took 3 different surfaces on which the toxicity evaluation was done. And the order of toxicity among the various insecticides have been listed here. And profiling of activity of the detoxifying enzymes vis-a-vis insecticides was also done. And it was seen that in carboxylesterase and CYP450 enzyme activity was higher in deltametrin treated insects and lower in the case of the other insecticides. All the above prove that role of these enzymes in detoxification. These are some of the conclusions. Monica J. also worked under Suresh Nebapure, and factors of resistance in pigeon pea. 50 genotypes were screened, and these are the observations which were recorded. And based on that, 2 varieties, Pusa RR16 and IPA-B, were found to be resistant. Physical parameters and biochemical parameters were also taken into consideration, and it was found that resistance of pigeon pea genotypes against C. chanensis was mainly associated with the anti-nutritional factors. Our faculty strength is 28, division 15, other disciplines, other divisions 2, NCIPM, and from Jharkhand and Raipur. And we have 18 research guides. 2 more faculty have been inducted at NIBSM Raipur, superannuation none. And at present, we have 86 students, including the 17 who will be receiving their degrees. These are some of the honors and awards of the faculty members. We had 5 training workshops conducted in the division, and we have manuals for each of them in place. These are some of the significant student awards. One girl, she received IRI Merit Medal. Then Ipshita Samal received Guru Prasad Pradhan Memorial Medal. Varun Saini Best Thesis Award. And also one of my student, Pin, received the National Overseas Scholarship for ST students to work in Cambridge. And 8 out of 11 seats of ARS were backed by entomology division students.
More than 15 awards were for oral and poster awards. Extracurricular activities also, our students did not lag behind, and students fellowship and awards and publications. 53 out of the 85 published papers of entomology were students. 7 above 10 and 8 to 10, 2 and 6 to 8 were maximum 31. I acknowledge the help received from Director, JDRs, PG School Administration, students and other staff members of entomology. Thank you.
Thank you, Dr. Devjani, for a very comprehensive presentation. So before I invite upon the next speaker, sir, I would also like to welcome our former Dean, Dr. Rashmi Agarwal is here. Welcome, ma'am. May I now call upon the next presenter for the day, Professor Pankaj from Nematology discipline to make his presentation, please.
Good morning to everybody, because still 4 minutes are left and for the afternoon. I will be going for the significant educational achievements in the field of nematology during the period 22-23. This is basically the total faculty members which are present in the division, total 19. And we have in total 22 PhD students on roll and 8 MSc students on roll. So total 30 students on roll. The list of students who are taking degree this time, we have 3 MSc students and 8 PhD students. Total 11. This is basically the theme-wise significant achievements of the students. And 5 themes are there. The first student, Abid Hussain Sheikh, he worked on the characterization of some of the population of Pratylincus in India. And he identified the Pratylincus species, that was Hipsteri. That is the first report of its occurrence from India for the first time. And that too from apple orchards, with the morphological as well as morphometric differentiation he has taken. The molecular characterization was conducted for 4 species, those who are present in IRI form. So the lesion nematode was basically to give a clear-cut picture of that particular nematode and particular field. So we can design the nematode suppressive crop sequence for their management. Naveen Kumar, who worked on the survey of root knot nematodes in guava orchards, as it is one of the major problems in the area of Uttarakhand, Uttar Pradesh and Haryana. He did survey as well as he has characterized those populations with regard to the morphometrically and morphologically, and later on preliminary identification confirmed with the perennial pattern. So what he has done, he has taken the molecular confirmation with the most of the all these species identified. Later on, the Maya Patil, she has worked on characterization and pathogenic potential of root knot nematodes infecting guava in India, because this nematode is one of the major problems. And she has collected different population. She has identified and morphometrically which was present or similar, found to be similar with regard to the Melanogain enterolobi. She conducted an experiment with regard to its pathogenic potential. What the nematode, the 3 nematodes which were present, that was the enterolobi, Melanogain incognita and javanica. What she found that the Melanogain enterolobi was highly pathogenic compared to other 2 species. She also seen the guava genotypes reaction with regard to enterolobi, so that we can have a good confirmation of those reactions, basically with regard to the guava genotypes. Chandramani Wagmare, he did the diversity of fig fruit associated nematodes from in and around Delhi. So this fruit is one of the most important and highly valuable. So he collected the samples from Delhi and in and around the Ghaziabad, the Modinagar area, where he found abundant of this nematode. Only basically what he was planning that the ficus was this Cystoncus sensulata. It was highly diverse. So the species was not identified so far. What he did that the 5 already described species were described and found in India, and later on 3 species he has given the ficus, Phycophagus morphospecies. What he has done with regard to the phylogeographical pattern of fig nematode species, that all the 7 they were or 8 species they were present in India. Rest of the world they have already reported those species. Soumya worked on effect of long-term rice wheat crop rotation on soil nematode abundance and diversity. She did the experiment with regard to the rice wheat rotation along with the moonbeam as well as crotalaria. What she found that the rice wheat cropping system had abundance of nematode problem, that is your Meladogyn gramicola. But what we have seen, what she has seen that the Meladogyn gramicola population reduced with the application of moonbeam as well as crotalaria, and there was increase in the bacteriovorous nematodes. So these bacteriovorous nematodes were reported and they were characterized molecularly, and they were seen whether these species can be effective for the management of nematodes. So field experiments were conducted for 2 years continuously, and they have reported the subtilis as well as megatherium. These were found to be effective. Now, Devraj worked on understanding the effect of temperature on rice root knot nematode interaction. Basically, this nematode is really a major problem, and the temperature increase has reduced the resistance capability of the plants. So, in the we have received around more than 1,000 specimens, the accessions where we have tested for resistance against Meladogyn gramicola. We narrowed down to 717, then later on 70, then later on only 3 accessions were found to be effective and had very less nematode infection. Even the penetration was less. So cumulative hatch percentage was also observed because NW1 and MW11 as well as 17, they were reported to be highly resistant or even immune. Only 2 or 3 nematodes penetrated at a later stage up to 7 days of inoculation. This was the pattern which was observed with regard to the development. The expression profiling was also done with regard to the key genes and metabolomics studied, and we have already given to Dr. Ranjit with regard to the metabolomics studies. Anamika worked on RNA effect of Meladogyn incognita development genes on its development and multiplication. She worked on RNA effect, and she has observed that the RNA effect on number of nematode or the number of eggs per egg mass were found to be less as compared to even the hatching percentage was less. Even the variation, there was variation in the egg size of the nematode. So that particular gene was expressed fully, and positive fold changes was observed with regard to this URPCR. Abhishek Gowda worked on eco-friendly management of root knot nematode and associated bacterial wilt, that is the Ralstonia solanacearum. So wilt problem is there. So we have identified and isolated the main compound of from the Mentha longifolia. What we observed that they were found to be effective at in vitro condition as well as in vivo under the polyhouse conditions. Even the bioefficacy of VGPRs were also conducted. What we found that the lemongrass as well as clove, they were found to be effective in reducing the wilts incidence also with regard to the nematode infection. The reproduction factor of that particular nematode was reduced.
Manjunath Gowda, he worked on relationship between root knot nematode and microbial community from the rhizosphere of tomato grown under protected cultivation. He has compared the rhizobacteria with regard to the infected as well as non-infected area. He identified and he has observed the microbial community and went for metagenomics as well as profiling of viable microbial community was seen. What they found that the healthy root as well as healthy rhizospheric soil, they have been reported with high population of the bacterial this bacterial population was basically 3 in healthy rhizosphere and 3 in infested rhizosphere. They found that all the 3 or the 6, they were effective for control of Meladogyn incognita, and it was reduced the reproduction of Meladogyn incognita in tomato plants under pot conditions. Devendrappa worked on exploration and evaluation of root knot nematode antagonist bacteria from tomato rhizosphere in polyhouse cultivation. What he did that he took the infested soil and infested and uninfested area where he isolated the bacteria, and these bacteria were taken for the control of Meladogyn incognita. So phylogenetically, they have seen the rhizobacterial isolates, and these potential rhizobacterial isolates were taken into consideration for the management of Meladogyn incognita in tomato. Enhanced expression was also observed with regard to the defense genes. Last is the Chaitra Bhatt. What she got the IRI medal this time yesterday only. Congratulations to Chaitra. She has done the whole genome sequencing of Heterorhabditis indica, which basically has the final assembly size that was 91.26 MB, and the gene predicted were 10,494. So she also elucidated the genes involved in the nematode bacterium symbiosis, because she has already seen at the early stages of Heterorhabditis differentially expressed transcripts 754 and 12,151 symbiotic nematode specific transcripts were identified. So they predicted that the particular symbiosis model can be seen so that we can have a study on the transmission of that particular bacteria from a host. She also standardized the RNA gene silencing in Heterorhabditis nematode, so that we can have a protocol for RNA-based functional validation. With regard to the publication of we have total of 27. Out of 27, 13 were from the student thesis, with more than 6 or 6 to 8, 3, 8 to 10, 5, 10 to 12, 1, and more than 12, 1. Conference papers or souvenirs and manuals, we have 2. These are the 13 publications in total of the students who are receiving degrees from the student placements. They are being placed in private companies. Even the assistant scientist, Chaitra, she was selected as an assistant scientist in HAU-ISAR, assistant plant protection officer, and assistant professor TNAU Trichy. Then Abhishek Gowda, Devendrappa, and they were back with the best poster presentation award. Tushar Kanti Datta got the associateship in NAS from NAS. These are the training programs which they have received, as well as Chaitra Bhatt, she got the Fulbright doctoral research fellowship. I want to thank Director Sir, Dean, Chairpersons, Advisory Committee members, and students for help. Thank you very much.
Thank you, Professor Pankaj, for making a presentation with the time limit. Thank you. So with the permission of the chair, may I now request the next presenter for the day, Professor Robin Gogoi, to kindly make his presentation from the discipline of plant pathology.
ये मैं डाल दी पहले। तो मैं बड़ा कर रहा हूँ ना, हो नहीं रहा।
Good afternoon. Respected dignitaries on the dais, scientists, professors and heads of the various disciplines and divisions and students. So now I am going to present the achievement made by our divisional students for the year 2022-23. So as per the mandate fixed for our discipline is plant pathology, that is to conduct basic and applied research and to serve as the center of academic excellence in the area of postgraduate education and to provide national leadership in plant pathological research. So with respect to the faculty, so as a whole in our division, the strength of scientists is 30, and out of 30, now 24 scientists are in position, and with respect to the faculty, so 13 as a principal scientist from IRI, one from NBSM Raipur, one principal scientist, and in the cadre of senior scientists, total 6, out of which 5 senior scientists from IRI Delhi and 1 from IRI Jharkhand, and 3 scientists are also faculty, total 23, and total students in MSc on roll is 26, out of which 13 students will be receiving degree in this convocation, and in PhD total 57 students are on roll and 12 students will be receiving degree in this convocation. And here the list of MSc students who are going to receive the degree in this convocation: 13 MSc students and 12 PhD students. And here from the top you see the second one, Rahul Kumar Tiwari. Yesterday he bagged the IR Merit Medal. One more, this PhD student, Wajdani Dauda, he is from Nigeria. He also completed his PhD program under the guidance of V. Shanmugam this year. And in these 4 thematic areas, I am going to present the research achievement of our student. And the first themes, disease diagnosis and pathogen characterization. Total 5 MSc and 5 PhD students, these achievements are summarized here. Very briefly, I am going to tell about those achievements. Venkata Dinesh, under the guidance of Dr. M.S. Saharan, with the title, he did work, that is the characterization of Fusarium graminearum isolates for mycotoxins and an identification of Fusarium head blight resistance in durum wheat. And total 22 Fusarium graminearum isolates showed. So no DON, that is deoxynivalenol toxin production, but showed presence of 15-adone, 3-adone, and nivalenol. And that was proved by using this toxin-specific primers. And also for survey of the resistance to Fusarium head blight, 3 genotypes exhibited resistance, while 6 genotypes showed moderately resistant to Fusarium head blight. And the other student, Nisit Reddy, under the guidance of Dr. Malkhan Singh Gujar, so he studied with the title Morphological and Genetic Diversity Analysis of Bipolar Sorokiniana, that is the causal pathogen of this spots blots of wheat, and identification of resistant sources for this spot blots disease. And Nisit collected different isolates and disease samples from wheat growing areas of different states. And after purification of the isolate, he confirmed about the pathogen as bipolar Sorokiniana using ITS primer, and the sequences submitted to the gene bank, and also accession numbers were obtained. And for the resistance sources survey, so reaction of 196 wheat genotypes to spot blots were studied, of which only 7 were found to have moderately resistance. None of the examined genotypes were found to be resistant. And RT Gowns, under the guidance of Dr. Kajal Kumar Biswas, she worked with the title Current Status of Cotton Leaf Curl Disease and Associated Begum Virus Complex in Cotton Growing Areas of Northwest India. And she found that cotton leaf curl disease and associated Begum virus complex in cotton growing areas of Northwest India, and that was studied and also found to be recombinant. And the cause, the lower outbreak of cotton leaf curl disease in the Northwest India under the existing situation. Sudip Adhikari, under the guidance of Basavraj, so he this investigated about the virome profiling of the viruses or virus-like pathogens from the naturally infected, especially cucurbits, most specifically pumpkin. And on the list, you can see these are the number of viromes could be detected, and next generation sequencing based virome profiling was conducted to detect known and novel viruses in naturally infected pumpkin plants and other cucurbits host. As cucurbit is the reservoir of many viral diseases, and his significant findings is the whitefly transmitted cucurbit yellow stunting disorder virus as a new crinivirus infecting pumpkin plants in India. Another report also he made a whitefly transmitted squash leaf curl China virus as a new Begum virus infecting bitter gourd plants in India. And third one is a novel aphid transmitted poliovirus tentatively named as pumpkin yellow virus. Chandu so did this is research work under the guidance of Amrita Banerjee. This is from IRI Jharkhand with the title Molecular Characterization of Rice Tumor Virus from Eastern India, and developed a rapid diagnostic assay using recombinase polymerase amplification technique. As we know that rice tumor virus disease, so it is mainly occurring in the eastern part and northeast Indian rice belt. And with that this aspect, Dr. Amrita gave the this work to Chandu, and Chandu could find a new rice tumor isolates Katak 2020 collected during the Kharif 2020 from Eastern India. And rice tumor basically from virus Katak 2020 characterized based on the complete large intergenic region, and also rice tumor spherical virus Katak 2020 characterized based on the partial CP3 gene. And further, this RPA-based protocol was also developed to detect RTBV directly from the crude shape, and also developed RPA-based assay to detect RTBV, which is not possible to detect in the simple PCR method. And Sanjeev Kumar, the PhD student under the guidance of Dr. Kalyan Kumar Mandal. So with the title, he studied, title is the study on bacterial panicle blight of rice caused by Burkholderia gloomi, with reference to genetic, pathogenic variability and toxin profiling of Indian strains. And for your information, this bacterial panicle blight disease was first reported from our division by Dr. Kalyan Mandal and his group in the year 2015. And since then, the work has been carried out for detailed study. So the multi-locus analysis using specific gene sequences of representative isolates of Burkholderia gloomi showed 99.76% and 100% homology, respectively, in the US strain of Burkholderia gloomi isolates from rice. And the REC gene sequence exhibited 100% similarity of Burkholderia gloomi and HN2 strain from China. And further, those fingerprinting data with box PCR seems to be very useful for analyzing the very genetic variation among the bacterial pathogen. Pradeep Kumar, a PhD student under the guidance of Dr. Vikas Mandal, so he undertook the study, that is serological assay for broad spectrum diagnosis and fingerprinting pattern for specific diagnostics of important begomoviruses in India. And he developed antibody. So this antibody developed against synthetic, this tomato leaf curl delivirus CP, that could not differentiate the disease and healthy plants. But in planta, expression of the CP, code protein of tomato leaf curl, this new delivirus could be developed. And also, so Pradeep developed this species-specific detection assay for important begomovirus species, and for this robust detection system, and PCR-based fingerprinting pattern was also developed for these viruses like MYMB, MYMIV, and MYVMV. And so duplex PCR for detection of the two viruses could be standardized. And Rubin Dev Barma, under the guidance of Dr. Diva Kamil, so he studied the molecular phylogenetic diversity and development of signature sequences for pathogenic species of Fusarium solani species complex. Always it is confusing. This Fusarium species of Fusarium oxysporum is always giving confusion to the plant pathologists. So with that aspect, so this to resolve that problem, Rubin Dev Barma studied, and he identified. He initially collected 85 Fusarium solani isolates from different geographical regions of India, and he could identify this 11 cryptic species, and those cryptic species also pathogenicity test was conducted, and out of these 8 cryptic species, except Fusarium solani from a species radicicola, all identified specific this cryptic species are able to infect tomato plant. And Srinath, under the guidance of Dr. G.P. Rao, so studied on the genetic diversity of phytoplasma associated with stone fruits, its natural reservoirs and development of rapid diagnosis. So this is only the phytoplasma, this line of research work going on from the Division of Plant Pathology. This phytoplasma belonging to 4 different groups and 5 subgroups were detected in the symptomatic stone fruits from Himachal Pradesh, Jammu and Kashmir, and Uttarakhand. And this Candidatus phytoplasma asteris and Candidatus phytoplasma australasiae detected in the peas are the new host records from India. An association of 16 SR1 and 16 SR4D subgroup phytoplasma was detected in a sweet cherry. And LAMP-based detection protocol was also developed, and that was validated to prove the existence or presence of this phytoplasma. And Prasant, under the guidance of Dr. Vishnu Maya Baishal, so worked on the molecular characterization and development of high throughput field-based diagnostics for Fusarium fusicora, which is the causal organism of Bakani of rice disease. So a total of 15 these petotypes were identified in Fusarium fusicuroi, and total of 15 SNPs were identified across 6 housekeeping genes of Fusarium fusicuroi. And successful amplification of and detection of the target region of Fusarium fusicuroi through this lateral flow RPA could be achieved. And the overall RPA detection assay, starting from template preparation to visual detection, took very short time, within 30 minutes that could be completed. And under the team host plant resistance, here I am going to present this 4 MSc students' work. So Ram Mohan, under the guidance of Dr. Vaibhav Kumar Singh, worked on the phenotypic and molecular characterization of leaf rust resistance in some wheat materials. So he collected, he used 86 genotypes, among which 18 promising genotypes were found to have the 3 LR genes, and 3 varieties showing resistant reaction in the phenotypic evolution, and that were found to possess so the gene LR10 and LR34. And Amjad Khan, under the able guidance of Dr. Z. Prakash, so worked on the evaluation of 2 genome-edited rice lines for response to the blast disease, which is incited by Menga oryzae. So among all the isolates evaluated, including the parental line, so here this maximum virulence was found from this isolate MONI0123, and with the parental disease index 10%, 11.33, and 12% in the one genome edited, both the genome edited lines, as well as this parental line. And on the other hand, susceptible chicks exhibited very high disease pressure, 97%. And those resistance lines were evaluated for the presence of R genes, like important R genes for blast diseases PI9, PI1, PETA, and PI2 by molecular markers, specific molecular markers. And among the R genes, the presence of PETA was detected in both the genome edited rice lines as well as in the this parental line. And Komal, with the title Phenotyping and Molecular Screening of Rice Landraces from Northeastern India for Blast Resistance, so worked under the guidance of Dr. Amrita Banerjee in IIRI Jharkhand. So her salient findings are there. 166 rice landraces were collected, and those the landraces having multiple genes and they proved actually out of 166, 89 phenotypically resistant to moderately resistant rice landraces could be recognized, and those landraces having multiple genes could serve as a valuable donor for introgression of blast resistance genes into the susceptible varieties. And further, this confirmed the resistance potential of accession having PI2 in Komal Dhan, and also PI54 in Amusu, Michigan. And however, interestingly, the accession having PETA2 in Malbhog Sali appeared to be susceptible during the challenge inoculation. And Swagata Thakur, under the guidance of Dr. S.K. Jain from NIBSM Raipur, so worked with the title Phenotypic and Molecular Characterization of Resistance in Finger Millet to the blast disease caused by Paricularia grigia. So she collected total 8 accessions of finger millet core collections, and also they were tested for this resistance or this disease reaction, and this categorized as resistant to moderately resistance category for both neck blast and also finger blast under the artificial inoculation conditions. And using some molecular marker like SSR markers, 2 SSR markers, so UZEP76 and UZEP102 gave the best polymorphism having band constantly, interestingly, in the susceptible genotype. And these 2 SSR markers could be associated with the neck blast susceptibility in the finger millet. So in the third theme, the disease management here, 6 presentations, 6 reports will be made. And Belu Murugan, under the guidance of Dr. Andi Kumar, studied on the leaf endophytic bacillus mediated suppression of blast disease of rice. And so through under his study, so he determined the secreted metabolome mediated antifungal antibodies say performed in 2 bacillus species bacillus siamensis and bacillus amyloliquefacens, and showed 72% and 70% mycelial inhibition. And also it was proved that using GAR A gene marker, subspecies identity 2 bacillus subtilis, which I just now mentioned, so was established. And these bacillus species so could suppress the blast disease by 65% and 64% respectively. And after that, so the functional analysis was also done. The expression of the defense-related enzymes was also studied. So both these bacillus species were found to have the excellent candidate to suppress the blast disease of rice. And Chemi Docker, under the guidance of Dr. Amrita Das, studied on the plant-associated yeast with antagonistic potential against banana anthracnose caused by Coleotrichum houssii. And you see, interestingly, so this time, so Dr. Amrita Das, with the idea given to her student that exploitation of yeast as for controlling the pathogen. So in vivo assay, potential yeast isolates showed significant reduction of the lesion diameter. And here 3 yeast species utilized, that is this Hanseníaspora ovarum, then Oreobasidium pullulans, and Trichosporum asahi. And all the 3 were found to have very good potential to suppress Coleotrichum houssii and Coleotrichum siamensis. And Rahul Tiwari, so I congratulate Rahul Tiwari for bagging the gold medal of IRI. So he worked under the guidance of Dr. Rashmi Agarwal. So his work title was Identification, Physiology and Management of Potato Dry Rot Caused by Fusarium Species. So as you know, this dry rot disease, it is really troublesome. It is causing havoc loss during the storage as well as transportation. So he tried to just find out what are the Fusarium species are involved to cause dry rot disease. So among 106 Fusarium samples he collected, so 3 different species like Fusarium this Sambucinum, Fusarium solani, Fusarium oxysporum were found to be predominant to cause dry rot in the potato. And out of the 20 popular varieties of Indian potato cultivars, so Kufri Pukras was observed to be highly susceptible to the Kufri Chipsona 3. And after identification and confirmation of the association of pathogen to cause dry rot disease, so he attempted for proper management. So a management approach of the to mitigate this dry rot of potato disease was done, and this sodium metabisulfite, melatonin, and Chitomium globosum derived initially silver nanoparticles were synthesized and characterized and applied to suppress this dry rot disease, and he found very promising result. And Pothiraj, under the guidance of V. Shanmugam, so worked with the title development of a carrier-based biocontrol consortium to induce Fusarium wilt resistance in tomato. And he established the physiological races of 14 Fusarium oxysporum lycopersicii strain representing 4 Indian states as race 3 and race 2 strains based on the PCR profiling. And he established the candidate defense genes in host resistance induced by the biocontrol consortium. And these are some field validation of this identified this biocontrol potential of that organism, then consortia. And next, Asarani Patel, so under the guidance of Dr. Andi Kumar, worked with the title genetic and functional characterization of endophytic this Mycobacterium testacea against rice blast pathogen, this Magnaportia oryzae. And so initially the genus level identity of rice phylosphere adopted bacteria was identified as Microbacterium using this polyphasic taxonomic tools. And then whole genome sequencing was also completed. And the endophytism of Microbacterium testaceum could be established. And interestingly, so see this laid out very exhaustive field trial in 3 different states and found to be having very, very promising bioformulation from Microbacterium testaceans. And Chaitra, under the guidance of T. Pramila Devi, worked on the population diversity and metabolomics of Beauveria bassiana and their evaluation for virulence against mite. And initially, based on morphocultural basis, she characterized the different this species of Beauveria bassiana. And here, this isolates BB6, BB12, and BB15 isolates showed the lower number of this means suppression of this mite. And out of which this B6, which was showed high percent mortality of this Tetranicus species, that is mite, and also produce high sporulation on the dead insect. Prasan Patidar, under the guidance of Dr. Lakshman Prasad, worked on the morphopathogenic and genetic analysis of Parpuriosilum, Linacinum, and Pacillomyces species and their efficacy against Melodogain incognita, that is root knot nematode. And based on this investigation, so a potential biocontrol agent for Melodogain incognita was found to have this Pacillomyces, this Parpuriosilum, Linacinum 6553, which could inhibit the nematode under the egg hatching, secrete various nematicidal compound, promote various promoting or boosting the plant growth, and also can suppress root knot nematode population in soil. And the last theme is the post-pathogen interaction. Here I am going to present 4 this achievement made by our 4 students. Hamida, she was also contestant for IRI Merit Medal, but she could not beg. So under the guidance of Dr. Tushar-Kanti Bhatt, worked on the quantitative variation in fusaric acid production by different isolates of Fusarium oxysporum formicis ciceris and its correlation with pathogen virulence and the post-infectional physiology of chickpea. So she exploited the Fusarium oxysporum ciceris isolates taking from Indian type culture collections, and she could extract the fusaric acid and characterized. And after application, so she studied in the chickpea plant the difference related enzyme expression. And next, Neelam Gate, under the guidance of Dr. Dinesh Singh, worked on the expression analysis of defense-related genes in response to elicitor applied on cauliflower against black rot disease caused by Xanthomonas campestris, Petavar campestris. So out of 170 screened lines or cultivars of cauliflower against this black rot disease, so only 9 were found to have resistance. And to control this disease, black rot disease, so ASM, acetylbenzolar S-methyl, at the rate of 350 milligram per ml was found to be very, very effective to manage this black rot disease in both the resistant as well as susceptible cultivars of cauliflower. And after that, expression analysis was also investigated and found 6 putative defense-related genes expression highly. Then this under the title Defense Elucidation by Pseudomonas putida BP25 in rice against rice blast incited by Megnaporta oryzae, work by Asha Jyoti under the guidance of Dr. Kumar. And here, Pseudomonas putida BP25 genome analysis also revealed very, very potential for the synthesis of several antimicrobial compounds. And that is found to be very, very promising to use as seed priming on the rice, which can trigger the altered these transcriptional responses. And after that, Pseudomonas putida BP25 elicited defense by LPS, EPS, flagelline, and siderophore. Further, this prophylactic application of this plant endophytic Pseudomonas putida BP 25. This can be a complementary strategy for the management of rice blast disease caused by Pyracularia, Pyracularia oryzae or Megnaporte, this oryzae. And Wazdani Dauda Palnam under the guidance of Shanmugam. So he worked on the identification and characterization of candidate this pathogenicity factor of robust pathogen Rhizoctonia solani coon inciting the seed blight disease of rice. So through his investigation, so a new phytotoxic compound from the host selective toxin of a virulent Rhizoctonia solani AG1A incited this was isolated and reported, which can incite the disease, and the role of the candidate hypothetical pathogenicity gene of Rhizoctonia solani in seed blight development was also elucidated. And these are some of his this research experiment design. So coming to the last part of this presentation, so publication from our student, overall student as well as our faculty, 60 last year. Here this training and our practical manuals 3, conference and seminar souvenirs 2, and manuals and book chapters 14. And only from students publication from the division of plant pathology, 24 publication we had in different this NAS rating category. So more than 10, 6, more than 8, 7, more than 6, 5, and less than 6, 6. And here our students placement. So our very, very bright students are taking placements. Usually our MSc students, they are joining the higher studies like a PhD program, and also those completed are on the verge of completion of PhD program. So they are going from some government job like ARS, State Department, like that. And I feel very proud that this year out of 10 ARS positions, our divisional 9 students could capture this ARS positions. Out of 10, 9. Honors and awards received by our very brilliant students. So I cannot name the name of the awards, like this Asajoti, Asarani Patel, Velmurgan, Pankuri, Chaitra, Wazdani Doda, Firuz Mandal, Mehuli Sarkar, Elur Mohan. And our able faculty members also bagged many honors and awards. So I name them: V.K. Baranwal, M.S. Saharan, T.K. Bag, Andi Kumar, Kalyan Kumar Mandal, Vishnu Maya Baishal, Amalendu Ghosh, G. Prakash, Sushil Kumar Sharma, Sumaila Sahid, and Nikita Gupta. And we could organize these trainings here. So ICR funded trainings, NABARD funded trainings, and also from this professional society, I, as a secretary of Indian Phytopathological Society, we could successfully organize IPS Platinum Jubilee International Conference in the SKN University Job near Jaipur last year. So my sincere thanks and acknowledgement to the Director, Dean and Joint Director Education, Joint Director Research, Joint Director Extension, our PG school, and various these funding agencies like NAHEP, DBT, DST, and ASF, our faculty members and scientists of the division, and also staff and dear students. With this, I thanking you all.
Thank you, Dr. Gogoi, for a very elaborative and presentation within the time frame. So may I now request Dr. Akka Rao, chairperson for the session on crop protection, for his remarks to the audience.
It's working, no problem. To me, this has been a full session. Of reasons. What I have seen is coming from a conventional university where we do it in an one-day event convocation, where one after the other, the PhD students come onto the dais and then receive their degree and then go away. Of course, the teachers are there to witness that. But this is something very, very, very special, a convocation which you are holding for a week, now maybe 5-6 days, whatever. Deemed to be university of 65 years now, and maintaining this kind of a tradition, I feel is a very excellent thing. And I am very happy that I am part of this particular event, an event which we all feel proud of. The students are proud of their degrees. The faculty are proud of the training that they have imparted to the students. And the institute is proud of the turnover of the graduates in all this. So on an occasion like this, to organize a presentation on the work done by the graduate students is something very special, and I liked it. And having said that, I would like to now congratulate the students, the 64 students who are graduating this convocation, the 32 PhDs and 32 master students who are graduating this convocation, and also congratulate the supervisors for guiding them for successful completion of their thesis and then towards getting this degree. What I noticed is that most of work that has been presented by the the professors who have been entrusted with this responsibility, I find that the work is of very high in most of the cases. And the students, I must congratulate for quality of work that they have carried out, and also for the they are receiving the achievements that they have made during the process. And I have seen there are other things like awards, and some of the students have been considered and then recognized for their work, and then adjudged as the best thesis or gold medal, all that. That's a wonderful thing. And I congratulate all the students first, and also the medal winners, very particularly those special medal winners, and the faculty who have guided them. On such such kind of a proud moment, what happens is the chest expands. But at the same time, we must remember that our shoulder size also increases with the increase in responsibilities when we graduate, when we get out into the society for to shoulder our responsibilities. I would like to now thank all the four professors, Professor Neera Singh, Professor Debjani Day, Pankaj, and Robin for the nice presentations. It's not easy, I know, to make presentations of all the works that are done in your own division and to summarize in a short span of time, which all four of you have done it very systematically. And I must congratulate the office of the director and the office of the dean, who have guided in preparing this abstract book, which is looks like a conference for me. And it is a 1.5 days conference condensed into a kind of a 1.5 hour session. That's what it looks like to me. And that's a great achievement from your office and her office. And I must congratulate all of you, the professors who made presentations and the guidance you have received from them. Another important thing that I have noticed is, as expected, of course, as a premier agricultural research institute, the kind of pathogens that you have selected, or the pests that you have selected, or the kind of chemical technologies that you have chosen for crop protection, most of them seem to be contemporary, and they are very, very much relevant to the requirements of the country. And therefore, I don't want to say, put my finger into it and say that we should have done that, we should have done this. Another reason is this is only a limited work that whatever is happening in the division you have presented, because this is about the outgoing students' work that I can understand, and a lot more things I'm sure you are doing with the number of faculties that you have. And I'm sure those other works also must be covering several other important areas. Therefore, I would say that I'm quite pleased to see that the kind of work that you have done with the different pests and pathogens, and also with the different agrochemicals that you are working with. And very happy to see another important thing, that all the 4 divisions have focused both on basic aspects and also on applied aspects. And that's a very good thing, to have a blend of both. Somebody like me will be always appreciating that, because we also come from that kind of a school where we look into the basics and also a little bit of application. And maybe here it may be more of application and a little of basic, but still it is much better. I'm very happy about that sharing component of basics and applied research that you have been doing at under the School of Crop Protection. This is no surprise to me, because I have been watching the progress of some of the faculty members, at least through the activities of NASA on one side, and as a member in DST or DBT on the other side. I've been watching the progress made by the faculty at IRI, some of you, if not all of you, the work that you have been carrying out, you have used the modern techniques in biology, and that's you proved that you can lead the agricultural research from the front, and with use of these modern technologies, and then giving training to the students in the areas that are required, especially like RNAi technology or the genome editing, and the advanced characterization of materials that you have been carrying out. They're all quite impressive. And one more important point I noted in the abstracts and also during the presentations is that a deserving attention has been given by all the 3 schools, particularly 3 divisions, where the agrochemicals may not have much to do with biosystematics, whether it is entomology, nematology, or pathology. All of you have given very much a required emphasis on the systematics aspect of these pathogens and pests, and that's very much appreciated. And also the number of GenBank submissions that you have made, or NCBI submissions that you have made on the insects, nematodes, viruses, phytoplasmas, and bacteria and fungi. In all of them, you have made a good contribution, and you have submitted, you have proved that these are very important areas, and we should not be neglecting these areas for the years to come. With all these observations that I have made, these are quick observations. Let me tell you, my dear friends, I cannot be an expert in all the crop protection areas. I don't want to venture on saying giving directions on what kind of a research that should be carried out at the Center for or at the School of Crop Protection. But I have a couple of suggestions on this occasion, because it is an occasion where the students are graduating, and the students are going to be working at different places. I also must congratulate the students who have got very good placements at different places already, and some of them, one division made a claim that all the 8 ARS scientists have, all of them got ARS. That's a wonderful achievement, definitely. And the suggestion that I have is, the first and foremost suggestion that I have is on the entrepreneurship culture that as a faculty, we need to promote, and as students, they should think about it. And that is one area where we will be able to make good contributions with the knowledge that we acquire in science. I say this consciously because at least in Hyderabad, from my own School of Life Sciences, my own students, not my own PhD students, my own students, I own my master's students as well. even though they have not done a project with me. They have set up 3 different companies today. If you look at their websites, you will realize how much they are contributing to the crop protection. Interestingly, all of them are into crop protection. Okay, they are not PhD students from my lab, but all 3 of them are contributing to crop protection. One is an ATGC. It is the company name is ATGC Biotech. It is in Hyderabad, and they produce high quality pheromones, very high quality pheromones, and they say they are confident that they can not only satisfy the customer about the essentials, but they can give much beyond that. That is something that they are capable of, and they have proven. They have successfully synthesized several varieties of pheromones, which can, as you all expect, are definitely used in mating disruption and also in mass trapping. And they also are very good components of IPM. That is something that they have made, and they are very good company, globally existing. That is one good company. The second company that I would like to mention is Sri Bioaesthetics. He has been again a student from our own department of plant sciences, and he has he's a big name in himself in the area of crop protection. And several of the microbiological products that he produces for crop protection are they're all novel. The third is the KN Bioscience. The KN Bioscience is 2 women entrepreneurs. Both of them are my students, biochemistry students, and both of them are doing exceedingly well together. So what the success of these 3 companies I see is more linked to their knowledge that they have acquired and their capability to understand how to deploy a particular product, develop a product, and then they take it to the market. So these 64 graduates this year, and maybe the yesteryear graduates and the coming year graduates, with the knowledge that they are acquiring in crop protection, I can imagine the information that they have in their brain can be really used for crop protection by setting up their own companies. Some of them, at least, we can't say that all of them should set up companies, but some of them can think of doing it if their mind supports, and if their system supports, and if their ambience supports. This is something I would like to suggest to the students who are graduating, and to the teachers. I request you to encourage them to move in that direction. If every year 2-3 of them come out from this kind of institute from every school, we see so many of them doing wonderful jobs. And the kind of work that, as I said, I've already mentioned, you have done in diverse areas and stuck to the mandates and then doing high quality work. These has resulted into very good publications, about 150 publications I could see from across the 4 divisions that have made presentations. And there is no end to us, for both for faculty and for the students, to set the quality of the paper. There is no limit. So if we publish in a NAS rating of 9 today, we should aim for 10 tomorrow and maybe 11 day after tomorrow. That should be the kind of setting, the quality setting that we can make. And these things can happen only if both the faculty and the students are thinking in one direction. And if they think, if we all think that they can become a complete story and then publish in a high quality paper, high quality journal, that will help us to build a reputation as an institution, division, as a faculty, and finally to the country. That is something which I thought we can, not that you are not conscious of it, but I'm just reminded of it when I saw about 150 publications made by the 4 divisions. I thought I'll make a mention of that. And I think I'm done with it. And finally, I would like to once again congratulate the students and faculty for this great achievement that you have made. and thank the administrative officers, our director, Singh Saab, and then Dr. Anupama for thinking about me, giving me an opportunity to interact, to learn about at one glance, what the crop protection work is happening at IRI. But from this book, I also know more details about what other what other schools are also doing in at IRI, and that's a very learning experience for me. And thank you so much, and congratulations to once again.
Thank you so much, sir. Now it's time to felicitate the chairperson. May I please request our director, sir, to do the honors? Thank you so much, sir. Thank you, sir. So as convener for the session, it's my moral duty to propose a formal vote of thanks. So on behalf of our director, sir, and dean ma'am, we all are thankful to the chairperson, Professor Appa Rao Podail, for his kind acceptance of the invitation and conducting the proceedings of the session in a very comprehensive manner. Sir, the suggestions given by you have been duly taken care of, and we shall try to do our best to follow the guidelines set by you. Thank you so much, sir. My sincere thanks are also due to our director, sir, and dean madam for their constant support and guidance throughout and taking care of every minute detail for the success of the program. I also wish to place on record my thanks to the various convocation committees for their kind cooperation and coordination throughout. Special thanks goes to Dr. G. Prakash, the member from the School of Crop Protection. And I would also like to place on record my gratitude to Dr. Neelu Jain as well, principal scientist from genetics, who assisted me in the School of Crop Improvement. Thank you to both of you. Most importantly, I would also like to thank the PG school and the Dean office, as well as our Associate Dean for providing all the logistic support. And I also profusely thank all the 4 professors of the disciplines who made a very comprehensive presentation, enabling us to finish the session in the time allotted. So thank you. It's all because of you people will be able to start the next session in time. So with the permission of Director, sir, we would now like to break for lunch, sir, and we reassemble at what time? So we'll reassemble at 2 p.m. for the NRM session. Thank you, one and all. Thank you.
ये वाला सिंगल आगे ना सेटिंग और ऑडियो पे ये वाला चालू करो
Hey, one. Hey, one. Hey, one.
इसको ठीक है ना इसको यही रहने दो इसको यही रहने दो इसको इतना नजदीक रखने से तो इधर हेलो हेलो अरे हेलो हेलो एक बार वो स्पीकर देना देखा था Hello, am I audible? Am I audible? Am I audible?
Check. Mic test 1, 2, 3, 4.
Hello. Sagar. यहाँ से ऑपरेट होगा ना यहाँ पे दिखाई देगा सब इस प्रोग्राम के बाद ना अच्छा यहाँ पे
Check 1, 2, 3, 4. Mic test. Sound check. Check. Sound check. Yeah.
जैसे यहाँ से ऊपर उठेगा ना।
Check 1, 2, 3, 4. Mic testing. 1, 2, 3, 4. Hello, hello. Sound check. Sound check. 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 Sound check. 1, 2, 3, 4. Hello, mic testing. 1, 2, 3, 4. Hey, hey, hey.
फुल स्क्रीन हो गया ये फुल कितना साइज
हेलो चेक 1 2 अच्छा आवाज ही ऐसी हेलो 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 1 2 3 हेलो हेलो कैसे हो सब हेलो साउंड चेक 1 2 3 हेलो चेक 1 2 3 हेलो
करेंगे तो बड़ा नहीं होगा कितना छोटा तो दिखाई नहीं देगा
ओके
पहले में तो सुनता नहीं है ये, तू सुना कैसे?
हेलो हेलो आवाज आ रही है
दिमाग खराब कर रखा है क्या
हेलो चेक 1 2 3 साउंड चेक 1 भाग निकलो चेक 1 क्या भाई हेलो हेलो अरे मैं घंटे दे रहे हेलो चेक 1 2 3 1 2 साउंड चेक 1 हेलो चेक 1 2 हेलो 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 तो दी नहीं हूं ना मैं हेलो चेक 1 2 हेलो साउंड चेक हेलो साउंड चेक हेलो दीन दयाल उपाध्याय हेलो चेक 1 2 हेलो चेक 1 2 ये ली
क्या हो गया क्या हो गया क्या हो गया
माइक इतना अच्छा काम कर रहा था वो।
क्या हो गया क्या हो गया क्या हो गया क्या हो गया आवाज नहीं आ रहा आवाज नहीं आ रहा आवाज नहीं आ रहा। आया 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 आया। Hello, hello, hello.
A very good afternoon to all present for the post-lunch session today. As convener of the session, I heartily welcome all the dignitaries, esteemed faculties, and dear students who have joined us for the session 3. In the morning, we had heard the presentations from session 1 and session 2, highly enlightening and very excellent presentations made by all the professors. For the third session today, we have the chairperson with us today, Dr. A.K. Sikka, former DDG NRM ICR. A very warm welcome to you, sir. It's an honor to have you amongst us. So may I kindly request you, sir, for gracing the dais along with our director, sir, and Dean ma'am. Thank you, sir. May I now request our director, sir, to kindly welcome the chairperson with the bouquet of flowers. Thank you, sir. May I now request Dean Madam for her welcome address to the gathering.
Thank you, Dr. Monica. So to begin the program time. Dr. Schedule to preside over this function, this event. And sir, we will have in this particular session in natural resource management, we will have from the School of Natural Resource Management, we will have 122 thesis highlights from 122 theses comprising 69 MSc and 53 PhD. Wherein the 7 disciplines, professors from 7 disciplines, they will be making these presentations. And sir, it's a real, it will be a real marvel to the eyes and the feast to the eyes in the post-lunch session, I'm sure on that. So thank you very much, sir. Once again, I warmly welcome all of you. And through this podium, again, I request all the scientists and the students who are still to join, and they must be listening to me online also, to kindly join and grace the occasion. It's our own function. Thank you so much.
Thank you, ma'am. May I now request our director, sir, for a kind introduction of the chairperson to the gathering?
Very good afternoon. I once again request all the members who have joined online to be present physically here, and the hall should be full. I think very soon people will join. Its number is less. It's my proud privilege to welcome Professor Alok Sikka, a name who is known to each one of us, and he is a renowned figure in field of natural resource management. We have been a great beneficiary of his wisdom in the field of natural resource management, having him as a member of our research advisory committee, where his input has been phenomenal. And you will see, sir, the glimpse of whatever suggestions you have made earlier. Many of those things have been adopted through individual scientist research and also through students research. And today, the presentation of a student's work will be made by professor of respective discipline. Because from each discipline, one student yesterday competed for the gold medal, and the rest of the students' work, which is also equally significant, will be presented by the corresponding discipline professor. Professor Sikka has made a phenomenal contribution in the field of soil and water conservation, watershed management, hydrologic modeling, drought management, climate change, water productivity, and farming system. He did his PhD in civil and environmental engineering with his specialization in hydrology and water resource engineering from Utah State University. And he was also visiting professor at Arizona and a faculty at Oregon State University. He was also director of the Northeastern Research Complex at Patna and Deputy Director General Natural Resource Management, ICAR headquarters. And currently he is working with International Water Management Institute, where his contribution and taking the issues pertaining to natural resource management at global level in context of India is very, very important and remarkable. We extend a warm welcome to you, sir, for sparing your time and being with us for this presentation. Thank you very much.
Thank you, sir, for your introduction of the chairperson. Now, with the permission of the chair, sir, I would wish to apprise the House regarding
And before that, I would like to make, I miss Professor Deshmukh we have with us. He is a great well-wisher of IRI at all forum, and he's very active member on social media. He stays in US most of the time. Sometimes he comes to India also, but wherever he is, he always takes as a front runner the cause of IRI. Thank you, sir, for joining us. Thank you so much.
A very warm welcome to you, sir. Thank you. So I would like to apprise the House regarding the modalities for the presentation for School of NRM. So for session 3, as already briefed by Dean Ma'am, there are 7 presentations from the professors of the following disciplines in the order which I'll just now read out: agricultural engineering, agricultural physics, agronomy, environmental science, microbiology, soil science, and agricultural chemistry. And last will be water science and technology. So as already circulated to all the presenting professors, due to the time constraint, we will be allotting the time based on the number of thesis abstracts to be presented, plus an additional only 2 minutes. So kindly respect so that we are able to complete the session, since we have one more session to go for today. So For the ease of the presenters, a yellow light will blink 2 minutes prior to the allocated time, and a red light will blink at the end of the allotted time. So with this, I now hand over the session to the respected chairperson for his kind remarks and to conduct the further proceedings of the session. Thank you, sir. Over to Dr. Sikka, please.
Thank you very much. Thank you, Monica. And first of all, good afternoon to all of you here on the dais and off the dais. And Dr. A.K. Singh, Director IRI, Dr. Anupma Singh, Dean of PG Studies or the Joint Director Postgraduate School, because there are different nomenclatures everywhere. And Dr. Monica Joshi, Dr. Deshmukh, all the professors who are over here, all the students, professors, ladies and gentlemen. First of all, let me thank Dr. A.K. Singh and Anupma for having given me this opportunity to be over here amongst all of you this afternoon and to witness the kind of the good work which is being done in IRI. And I know it's always a privilege for me to be associated with IRI in any way because the premier institutions like this. And as Dr. A.K. Singh was telling, I had a privilege. It was in fact my privilege to be on the RSC and also on the Academic Council. And of course, it was more or less similar time. And I think, but unfortunately, 2 years we were having a regular meeting. The third year was because of the Corona. Of course, it was all online, but still it was nevertheless a wonderful experience. And it always reminds me that the kind of the good work which the institution is doing. And this is an institution, though I did not study here, but I did have my basic training for 2 months when I was a student at IIT Kharagpur. And I was in for 2 months, 6 weeks or 8 weeks training in WTC. And I still remember on the other side when there was a drip irrigation layout was there, and drip irrigation in the 77 was in kind of fancy. And I remember Dr. Michael and Dr. R.K. Kaul. And then I was working with them. And when we used to do the layout for the drip irrigation, sometimes people will come and say, "Arey, ye kya ped ko injection laga raha, drip laga raha?" So that was the kind of a thing then. But I used to tell them, look, maybe right now it looks like that, but the days are not far when. So that was the kind of a thing that now you see it everywhere. I mean, everybody is talking about that. And also the solar pump, because I remember that on the back side there was a solar pump and the windmill. So that was, and now when I see, because everybody is now talking about the solar irrigation. So what I wanted to say is that IRI has always been the front runner for all the technological advancements in India. And not only that, but also whatever IRI has been doing, even overseas people have been making best use of that. So it's not that we are simply bringing technology from elsewhere, but we are also contributing to building the new technologies and the new research in number of disciplines which we have in IRI. So I deem it as my privilege to be over here. And as Monica was telling, for the paucity of the time, I will definitely not like to dwell into making a long speech, though the subject is very near and dear to me. But I'll desist doing so. And I would be rather more on a listening mode to enrich myself. So I must thank you and Anupma that we are given with the opportunity to enrich my knowledge, what is the latest on this subject of natural resource management. And at International Water Management Institute, we are very also happy to be continuously in association with some of the professors, especially Professor D.K. Singh. And now one of his student is also with us for doing the PhD. And we wish to definitely have more students, be it for the masters or for the PhD, and even as an intern, if the intern policy exists, is there with you. So that would be a wonderful thing. And of course, we are entering into an MOU, but of course there is a umbrella MOU of ICAR and IMI. So that still holds good. So I think I'll not, as I said, take much of a time, and maybe then you would be inviting them. No, please, because, you know.
Okay, thank you, sir. The permission of the chair, we'll begin the School of NRM presentations. So may I first invite the first of the day, present the thesis abstracts for the students from the discipline of agricultural engineering. Dr. D.K. Singh, please.
Welcome to the post lunch session. Respected chairperson of the session, Dr. A.K. Sikka, our beloved director, Dr. A.K. Singh, Dean and Joint Director Education, Dr. Anupma Singh, and the convener of the session, Dr. Monica, head of the divisions, professors, faculty member, and dear students. I have to present the achievement of 39 students, and that too of 3 major sub-disciplines, which are a bit diverse. They are not much interrelated also. I know that it is going to be very, very difficult for me, but I shall try to finish in time. Division of Agricultural Engineering was established in 1945. Perhaps it was the second department anywhere in India after the Allahabad Agricultural Institute, which came in existence. The PG program in the division started in 1966 with the 2 students. And we are happy that this year we have admitted 70 students. That includes 33 students from BTech. In 1970, the degree recipients were 9. This year, the highest number is 39. Probably that is the highest for any of the divisions in IARI. And then, of course, we are happy that our division has achieved another milestone by starting BTech program from this academic session. Now, I shall be moving to my organized presentation. The mandate of the division includes research, teaching, extension, and training. Also, we have a testing center sponsored by the Ministry of Agriculture, Government of India, where the farmers of this region are tested and the manufacturers are given certificates. Also, we have a scheme under revolving fund where the prototypes are developed and distributed throughout the country on sale basis. Students' research in the division is conducted under 3 sub-disciplines, that is farm machinery and power engineering, soil and water conservation engineering, and process and food engineering. And 39 students which would be receiving degree in this convocation by and large between these sub-disciplines. We have about 28 faculty members at IARI and then 46 faculty members at CIE Bhopal, where we started teaching program in 2015. As of now, on roll, we have 186 students. That includes 49 students at CIE Bhopal and then 69 at IRI. And then this year, as I said, that the 39 students would be awarded degree in the coming convocation. Coming to the first theme, that is on resource conservation technologies. One of the students, he studied the energy demand pattern for the tillage and sowing equipments in this region, but in northern India. And then 7 equipments, which include the tillage as well as the sowing equipments, they were evaluated at the part load, that is at 1/3 of the load, 2/3 of the load, and full throttle load under 5 soil conditions. And then the salient findings of the study were that the load factor and soil condition had a significant effect on tractive efficiency. Maximum tractive efficiency was obtained of 75% was obtained at the 0.52 load factor, and then at the 2/3 speed, the back machine, most of the machines they perform best. In another studies, one of the students from Rwanda, he designed a power system, solar operated threshing thresher. Rather, the thresher was existing. He connected the solar energy with the thresher, and then it was evaluated for soybean, green gram, and sorghum. The capacity of the machine was about 100 10 kg per hectare, threshing efficiency 99.8%, and energy consumption varied from 1.66 to 1.74 kilowatt per quintal. Under theme 2, that is under precision farm equipments, one of the students, in fact, we had been trying to use the sensor technologies and artificial intelligence and machine learning also in students' research. One of them, take a student, Karisma C, evaluated different sensor for estimation of crop disease severity and developed a sensor-based device for detecting disease severity in tomato and cowpea. Device detected the disease severity with an accuracy of 94%. It was tested for tomato and cowpea, and the disease severity was detected, and the success rate was about 94 and 88%. Another students, again, he worked on a similar line. He used the RGB imaging and spectral reflectance for the detection of diseased regions in the cauliflower and brinjal. The steps and the different parts of the machine system are shown here. And it was found that the sensor-based sprayer saved about 28% of the chemical, which otherwise would have been lost in conventional or manual operated sprayer, using manual sprayer. A student at CIA Bhopal investigated, did investigation on spray characteristic and recycling of spray liquid for tunnel sprayer, which is again an upcoming technology. He developed a single row air-assisted tunnel sprayer. The tunnel sprayer resulted in 67% of more spray deposits and 41% improvement in spray deposit ratio. For the time, paucity of the time, it may not be possible for me to discuss the details of this machine, which can be discussed later on if someone is interested. Again, a student at CIA Bhopal, he developed a 4-row electronic pneumatic planter with improved seed metering unit. Was, and then the quality of heat index was 97%. I did not get much detail of this because this is probably the last student who conducted the Viva which was conducted. So, but at least we can say that the quality feed index of this machine was 97%. Then again, a student at CIE Bhopal worked under Dr. P.N. Agrawal, and he studied on a tractor mounted camera based site specific Vidicide applicator. Rather, he developed the Vidicide applicator in which the camera was mounted on the tractor itself. The applicator included camera, Raspberry Pi 4B artificial light, 24-volt solenoid valve, etc. And then the average beating efficiency of the machine was 87%. The application rate was 400 liters per hour. And the overall efficiency, overall saving in the spray was about 26%. Then again, a student at CIE Bhopal was from farm machinery. He developed a process to convert the paddy straw into micro porous carbon, something like biochar. It's not exactly biochar, but he tried some co-pyrolysis process to convert using lignite to convert paddy straw into micro porous carbon. And the steps to convert that are shown here in this device. And then a study was conducted again at CIA Bhopal, where a student designed and developed a single automatic transplanter, which has the metering efficiency of 98%, and the survival rate of the plants using this machine was about 90%, and then transplanting rate was 131 seedlings minute more than the manual transplanter, and then cost in saving the cost of about 24% compared to the manual transplanting. A student at IRI, MTech student at IRI, he studied the phototactic behavioral response of the selected insect under varying light source. In fact, he designed and developed a smart trap insect control system for trapping the insect. And the feature of this system included LDR based LED illumination system, electronically controlled height adjustment system, insect removal and water recirculation system. And then power consumption of this system was about 35.79 watt per day. Then a student, cumin is a very popular crop, particularly grown in the North India, particularly in the Rajasthan. And then harvesting of cumin has always been a problem. So this probably study was done in consultation with the Spice Research Institute in Rajasthan, where a student was allotted this problem, and he studied the physical and mechanical properties of the cumin. He determined the design parameter of the different component of the harvester, and then finally the machine was developed, and it looks like as is shown in the figure. The average field capacity of the machine was 0.06 hectare per year. If it is propelled by the battery, it can be operated by battery as well as it can be operated manually. So in case of battery operated machine, the average capacity was 0.06 per hectare, whereas in case of manual operated, it was 0.05 hectare per hour. Average field efficiency in case of battery propelled, it was 61%, and in case of manually operated, it was 51%. And then cost of operating cost of using this machine was about 2,631 rupees per hectare. And then operating cost in case of battery operated machine was 2,251 rupees per hectare. A student, spraying has always been a problem. So a student during COVID period did study in Andhra Pradesh. He was here only, but his topic was related to Andhra Pradesh, where he developed a swinging type lance sprayer, which consisted of battery, voltage regulator, microcontroller, LCD display, speed sensor, servo module, latch bridge, etc. So all the components are shown here in this figure. And then it was evaluated in Andhra Pradesh itself with the College of Agriculture, in the field of College of Agriculture in Bapatla. The machine resulted in saving of capital to the extent of 44%, whereas the saving of man hour was 75% over the tractor operated gun sprayer. And the interesting thing was in this case was that there was no exposure to harmful chemical, if spraying is done using this machine. Then another study was done by a PhD student, Gopal. It was very interesting study where he developed a canopy volume based variable rate sprayer for mango orchard using the LiDAR technology. You can see here that using this technology, more or less entire canopy of the mango is covered. And then the AI-based 3D processing algorithm embedded with this system has really done wonderful thing in operating this sprayer. And then if spraying is done using this sprayer, about 52% saving could be done in the chemical and 55% compared to the gun type orchard sprayer. The cost of operation using this sprayer was 353 rupees per hectare, and saving in operation cost was about 26% in comparison to the orchard sprayer. In theme 3, that is the climate change impact and integrated water resource management, which is related to my subdiscipline. A student did a study in Kerala, in the coastal region of the Kerala, particularly in the coast of the Kozhikode, where the seawater intrusion is a very severe problem. She did a study, she did an initial survey, and it was from initial survey it was found that the seawater intrusion is really happening there because of excessive groundwater pumping in this area, this region. And then we thought of why not to take a sample from sea as well as along the river. The interesting thing was that during high tide, the water, sea water goes up to upstream of the river up to 8 to 10 kilometers, which indirectly also causes seawater intrusion. So all this experiment was planned. The samples were collected, analyzed at the CWRDM laboratory. The Piper diagram, Gibbs ratio plot, geostatistics, and the MART flow, PMBin and MT3D were used. In addition to that, the drastic model was also used to do the vulnerability mapping arising out of the seawater intrusion, and it was all done using on the GIS platform. So we can see here that the rivers are marked here. We can see that the red color is spreading up to the 10 kilometers from the sea, and all along the river also, both sides up to 200, 300 meters, there is severe seawater intrusion. So we, after all modeling as well as the other analysis, it was found that without any management salinity without any management alternative, salinity will reach up to 7.38 kilometer by 2030. And if it has to be checked, then the reduced pumping, meeting the requirement from the river to the regulator, can help in reducing the seawater intrusion. And in fact, that has already been thought of by the Kerala government, where they are going to have regulator on this river. And a 30% reduction in pumping, and if some amount of the water is met from the river, seawater intrusion could be minimized to the greater extent. Then another student, he did determine the parameter using water and nutrient sensor for modeling water and nitrogen distribution under greenhouse, for chrysanthemum grown under greenhouse condition. He used the hydrostory model and developed fertigation schedule strategy for the chrysanthemum. Salient findings of his study included that the yield and water productivity were highest for the treatment T2, which was 100% up to ETC and 80% recommended dose of nitrogen, and irrigation scheduling with emitter discharge of 1.5 liter contained the nitrogen within the root zone. It did not allow the leaching of the nitrogen below the root zone depth. Then another study was done in the 3 districts of Eastern Uttar Pradesh. 2 of these districts are under, covered under aspirational district program. That is the Chandoli, Mirzapur, and Sonbha district. And then this is the part of Middle Indo-Gangetic Plain. The problem with this area is that the geohydrological condition is such that the large scale groundwater recharge and storage is not possible, and also undulating topography doesn't permit for rainwater harvesting the pond at the other location. So the idea was to do some kind of analysis and study where we can find out, identify the regions where groundwater recharge could be enhanced, and also the convenient location for the rainwater harvesting through surface storage. And then the technologies and the method used were the DEM, which we got from the SRTM, the soil map, which were obtained from the NBSSLUP, and then the groundwater data from India Varnis, and all these are listed here. And then SWOT and AHP were used to do the analysis. And it was found that the surface runoff potential in these districts are fairly large, but due to the undulating topography, it was not possible. It is not possible even in future also to go for large scale utilization of this available surface runoff potential. Even if you utilize 35 to 40%, that will be really a great. And then based on all this study, the area was categorized under different groundwater recharge zone as well as rainwater harvesting potential zone, which are mapped on this. And this study, the finding of this study will really be great of huge to the planners. Also for the agencies which are involved in planning the rainwater harvesting structures, because through all these analysis, he has also marked the location of the rainwater harvesting structure, which is shown in the third figure at the bottom. And then a study was done in Punjab, central Punjab, where groundwater depletion is really a problem because of rice wheat system. Student Manish Devnath, who is scientist also at Cuttack, he integrated crop, water supply, and climate model for predicting groundwater recharge under changing climate for developing groundwater management strategies in that region. The flow diagram and the models and tools which were used by him are shown in the left-hand side figure. And the groundwater must be finally, after doing all this analysis and simulation under different scenario, it was found that the adoption of DSR along with increased allocation of the canal water supply could really help in reducing or arresting the groundwater decline, though not completely. Yearly average rainfall, if nothing is done, then would decrease by 24 to 31% in that area. That is through the climate model. And then, as I said earlier, that enhancing area under DSR and augmentation of canal water supply would result in arresting the decline in water table by 3 to 11 centimeter per year. So we have to see this figure, the 3 to 11. Even if you go for DSR, nothing much is going to happen to the groundwater in the Punjab unless we go for some other measures along with this. Then another study, Prashant, he developed a decision support system for irrigation scheduling of rice and wheat in Sultanpur district of Uttar Pradesh. Most of these studies, they were planned and done during the COVID period. So we have tried to see that the students, they undertake study in their respective regions or districts or area. He estimated AET in the rice wheat crop region of Sultanpur districts and developed a decision support system for irrigation scheduling. And based on all his analysis, it was found that irrigation water productivity and crop water productivity could be of 0.83 kg per cubic meter and 0.4 kg per cubic meter could be obtained if irrigation is done using the decision given by the decision support system. And then another student at Bhopal, he did spatiotemporal multivariate drought analysis in upper Tapti river of Besan, upper Tapti river basin in Maharashtra, where he concluded that agricultural drought events are longest in duration, 38.8 months, roughly 4 months in that region during monsoon season. And then meteorological drought occur varied based on the time scale. Then a study was done at the Naisham Baramati by MTech students, where the idea was where he studied the effect of deficit irrigation strategy on water use efficiency, yield and quality of pomegranate in shallow basaltic river. He found that the irrigation at 80% evapotranspiration with plant growth regulator increased fruit yield. He used in fact the plant growth regulator to combat the water stress in the plant. And it was found that with the plant growth regulator, fruit yield could be increased by 31.9% in deficit irrigation and 40.38% in partial root drying. In water stressed plant, salicylic acid can be used as a plant growth regulator. Another student, he did study, he is the student of IRI Jharkhand, probably the first MTech student of IRI Jharkhand, where he estimated the blue, green and grey water footprints are the major vegetable crops grown in Eastern Indo-Gangetic plain. And he found that the water food under different climatic change. He in fact investigated that how the crop water footprint is going to be affected as a result of climate change. He found that the water footprint of cabbage, potato and tomato were under prevailing conditions, they were under 481, 2,689 and 435 million meter cube per year, and under the baseline scenario. And green and blue water footprint of cabbage would increase by 2.75% to 6.88% as a result of climate change. A student at IRI, she derived the R and K factors of the universal soil loss equation, which is the only equation which is used to estimate the soil loss globally or universally. She found R value for IRI as 40,452 megajoule millimeter per hectare per year, where the soil erodibility factor K was 0.18, 0.1, 0.1 and 0.15 metric ton per hectare per square meter per for sandy loam and sandy clay soil. Again, a study was done at the Naisham Baramati by a MTech student, where he quantified the water stress tolerance of wild rootstock in eggplants for improving the fruit yield, water productivity and quality in scarcity conditions. And he found that the marketable fruit yield and water productivity improved by 15.4 to 43.9% and 5.8 to 11.1% kg per meter cube in grafted treatment under the medium and severe water deficit. He used the grafting technique to combat the water scarcity. Then a sensor-based irrigation scheduling was done for spinach at the CPCT farm in IRI, where a student evaluated the yield, irrigation water use efficiency, nitrogen uptake efficiency and nitrogen use efficiency. Under the experiment was planned, she has 9 treatment and then 3 replications, and then based on the study, it was found that irrigation water use efficiency, nitrogen uptake efficiency, nitrogen use efficiency, nitrogen uptake efficiency and yield under treatment T4, which was 85, where irrigation was done up to the 85% of the field capacity and 100% of the recommended N was applied. That treatment was found to be the best. Then a student addressed the issue of groundwater in Delhi. As you know that, as we all know that the groundwater is a major problem in Delhi. In fact, not only groundwater, the water itself is a big problem in Delhi, more particularly in the summer season. So a study was planned where spatio-temporal variation in groundwater availability and demand was assessed, and then area suitable for groundwater recharge was delineated, and suitable strategies were suggested to minimize the risk to the groundwater or minimize the stress on groundwater resources in Delhi. We'll see in the next slide that the problem with the Delhi is that if you look at the graph here, the first graph at the top, the population in Delhi in 1901 was 4.05 lakh only. Now the population of Delhi is 206 lakh. That itself tells us the problem of Delhi. Whatever we do, whatever we plan, until unless we address this issue by any means, it is not the problem of the water problem of Delhi is not going to be solved. You can see from this figure how the population has increased over last 110 or 115 years. And then if you look at the bottom figure where the average rainfall by and large remain the same, that is 631 millimeters. So these two figures speaks about the problem of water in Delhi. that the rainfall, which is the main source of groundwater recharge in Delhi, remains by and large the same, whereas the population has increased from 4 lakh to 206 lakh. So that's one. Now, analysis was planned where all these tools were used, and ultimately it was found that the domestic water demand in Delhi has increased 52 times from 1901 to 221. Rainfall, the main source, remains constant. Then the groundwater recharge total groundwater recharge in Delhi is only 314 million meter cube, whereas the demand is 777.93 million meter cube. About 41, that's a very striking feature again about the Delhi, that about 41.2% of the total area in groundwater is saline. So that has to be in any case discounted, and if you discount so, you'll find that large area in Delhi is not fit for is not where you cannot use the groundwater for irrigation or even drinking water purposes. So if you do some study, there is still some scope. If you go for managed aquifer recharge, particularly in Yamuna floodplain, so we can create additional 38 million meter cube by dewatering the aquifer in pre-monsoon season, where the aquifer will be recharged during the monsoon season when the river is at the high stage. Another study was done by a student. This I was interested in. This study was interested. I had planned this study for several years. Ultimately, one of the students, he came forward, and we took did a study in the phenomics lab of IRI, where she estimated or determined the evaporation, transpiration, photosynthesis, transpiration efficiency, and water efficiency. In fact, we all know that there is the transpiration component which really helps a lot in photosynthesis, or rather, if I had to say, it's basically the transpiration which helps in the conversion of solar energy into the chemical energy. So she tried to address this issue. It was a very small study, only conducted in one season with feed variety of 3086 in 30 parts with 5 treatments, where the treatments were normal, nitrogen excess, nitrogen deficit, then get controlled, and no weight and under bare soil. Bare soil we take because that helps us in determining the evaporation, soil evaporation. It was found that the mild water stress during the early vegetative growth stage and severe water stress during the maturity stage could be considered as an optimum condition for the limited irrigation regime. Increase in nitrogen accelerated the transpiration and photosynthesis and led to the higher biomass production. A strong positive correlation was found between transpiration rate and photosynthesis rate, and increase in transpiration by limiting the evaporation loss would increase photosynthetic activity, thereby increasing the yield. Then another student, he developed a sensor-based fertigation schedule for greenhouse tomato. He designed and developed a control head for sensor-based automated fertigation system to evaluate greenhouse tomato crop parameters based on the standardized fertigation sensor. And then it was found that the sensor-based drip irrigation saved about 10 to 15% of the water tomato, and he also suggested that tomato must be irrigated to 100% of the ETC demand and 100% of the recommended dose of fertilizer under greenhouse condition, and maximum TSS was obtained in case of T1, that is with 100% ETC irrigation up to 100% of the ETC and 100% of the RDF. Under coming to the probably the last theme, that is quality storage of agricultural produce. Here a student, he developed a polypropylene reinforced jute bag for pulse storage. Though he did this study with in collaboration with the NERJAF at Calcutta, where he went there and stayed about 2.5 months there and developed the bag, by using this material. And then optimal blending ratio for the preparation of storage bag was 60% jute and 40% was the polypropylene. The developed storage bag had a higher cycling potential for use in bulk bag storage, and the risk of spoilage of the stored grain with the jute and woven polypropylene bag was minimized. Oh, there is another thing. Last is this one, the value addition of agriculture produce. That is the last theme, where a student, Sanket Samant, he did a study on ohmic heating. Ohmic heating is a very upcoming technology. It's not very new technology, but this is being used for oil extraction in process industry. He did a laboratory, developed a laboratory unit of ohmic heating for enhancement of oil yield for enzymatically hydrolyzed black cumin seed, and the steps for development of this ohmic heating system was shown here. And then he found that the electricity to heat conversion efficiency of the developed apparatus was about 63%. The optimized ohming assisted extraction gave a maximum oil recovery of 79%. Then another student at CIA Bhopal, was the student from the process and food engineering. He developed a hydrodynamic cavitation system for enhancing shelf life of sugar cane juice, and a low temperature hydrodynamic cavitation system was developed for shelf life extension of sugar cane juice, and it was found that the hydrodynamic cavitation treatment resulted in significant reduction of microbial load and enzymatic activity of the juice, and the juice could be stored for 49 days with minimum deterioration in PS, TSS, and total phenolic content. And then another student at CIA Bhopal, again from the process and food engineering, developed a sensor-based system for detection of adulteration in selected dairy products. He did it for paneer as well as for the milk. The developed sensor-based system was efficient in detecting the adulteration with vegetable oil, because it's mostly the adulteration in the milk is done by the vegetable oil. So this system was able to detect the deterioration or adulteration by vegetable oil, and then accuracy of the system to detect the various category of the paneer was significant to the extent of 80%. And then another student again at the CIA Bhopal developed a micro encapsulation technology for papaya extract, because papaya leaves are often, it's very common, it's used in viral fever. The people who are suffering from the viral fever, another thing, they use papaya leaf. The chewing of the papaya leaf is it's not that convenient or comfortable. So he tried to develop extract and encapsulate it in capsule form. And then as per this study, the micro encapsulated papaya leaf extract could be preserved in amber glass bottles under the refrigerated condition with the maximum stability of bioactive compounds. Then another student again is CIA Bhopal developed and studied the dynamics of ozone in bulk storage system for onion and its effect on shelf life, the efficacy of gaseous ozone for elimination of A. niger and E. Cortovora inoculated on onion bulbs was investigated. It was found that ozonation in the pulse mode compared to the single higher dose was found better in activating the microorganism, and then overall ozonation reduced the rotting percentage in onion bulbs during the storage with minimum alteration in the quality parameter. Then again, a student at CIE Bhopal developed a non-destructive detection of the fungal squalage in maize by reflectance spectroscopy and hypospectral imaging. The optimal wavelength for this system was 538 it went up to the 1068, even up to the 1852 level. And then a student at IRA, MTech student, she optimized the pre-treatment processes, drying and packaging condition for the production of dehydrated apple slice using infrared dryer. The apple slice are becoming very common. It's tasty also. In fact, we had tasted her product and it was really good, very crispy. So the sample was dried at 650 watt with sample to emitter distance of 20 centimeter and had a comparatively better rehydration ratio and antioxidant activity. And the aluminum laminates were better packaging material compared to the polypropylene and low density polyethylene. And then a student, again, we know that our Pusa farm sun freeze is becoming very popular, and it has been, in fact, it is being installed at various locations in North India. And the student studied the storage characteristics of the Pusa sun freeze, and with variation, and then also she tried to study the variation in temperature and the relative humidity inside the sun freeze, if it is the product is stored for longer or commodity stored for longer duration. She found that reducing sugar concentration in Pusa sun freeze was about 232 milligram per 100 gram at the end of 90 days, which is much below than the acceptable limit of 500. So that's the advantage one gets by storing the commodity in the Pusa sun freeze. And then storage of the potatoes in Pusa sun freeze enhance the shelf life and maintain the quality. Then another student at CIA Bhopal developed a lag dye-based intelligent packaging indicator for real-time freshness monitoring of paneer and mushroom. And it was found that the log dye-based indicator was effective in spoilage detection, paneer and mushroom. A color range for the indicators during storage clearly distinguished the samples as a fresh, medium, fresh and spoiled one. So that was all from the students' presentations, sir. I'll just quickly cover the publications. There was one publication which had an NAS score of more than 1. In fact, this does not include the publication from the CIA Bhopal. In fact, we could not get their publication list of publication in time. So that's only for IRI. If you add the publication of CIA Bhopal, probably it will that more than NAS rating will go much more. It could be even 8-10 because processing students they publish the paper in higher NAS rating compared to our farm machinery and soil water engineering students. The placement, we are very comfortable with the placement of our students. And then 9 of the students were placed in either in the state universities as assistant professor or in the state government department. Also, majority of our master students, they have joined the PhD program, either in IRI or in other institutes. Honors and award to the students. One of our student, Anand, he got IST National Award for the best MTech thesis. Then Dattatreya Bhalekar, he got ISA ITFA students gold medal, and then Karishma got graduate assistance for higher education in South Dakota University, USA. And then honors and award to the faculty member. Our former head, Dr. Indmani, was appointed vice chancellor in University in Maharashtra. Dr. Sarang is joined as a director at Bhubaneswar. Dr. Abhijit Kar, one of our faculty member, again joined as a director in Ranchi. And then the honors and award to other faculty member like Dr. S.P. Singh got fellow of Society for Sugar Cane. Again, Dr. S.P. Singh got fellowship for International Society of Noni Science. Dr. Rao got AIA as a scientist award. Again, he got IST award likewise. So I'm not going to read in the name of all, but most of our faculty members have got one or another award last year. Acknowledgement. I acknowledge the students whose work I presented today. Chairperson of their advisory committee, head of the divisions and faculty member of the division. They have been great source to us. And I special acknowledgement to Dr. Rao, Dr. Dilip, and Dr. Arun, and Engineer Nandalal in compilation of these work of 39 students so that I could present well in time. And then I also acknowledge Project Director WTC, Dean and Joint Director Education, Joint Director Research, Joint Director Extension, Director IRI, and all the staff and officers of the PG School. Thank you. Thank you, sir.
Very much. Let's give him a big hand. I know that you had the most onerous job because you had the maximum students' presentations to be made. And I think you did a good job of consolidating everything in
that too
a
from
very,
a
very
diversified
precise way.
It was really
So although your students have been doing a lot of precision farming, so you also followed some precision approach in trying to
thank you
do
sir.
that. So I don't think there is a time for any question or anything that's not required. At the end we will. Yeah, I'll keep my remarks.
Thank you.
All right, so thank you very much, Professor D.K. Singh. And next, I invite Professor P. Krishnan to present for the agricultural physics summary of the students' work.
Good afternoon. Respected Chairman of today's session, Dr. Sikaji, Director, Joint Director, Convener of today's session, and my senior and junior colleagues sitting off the dais. And today I'll be presenting the salient findings of our students who are graduated during the year 2022 in the discipline of agricultural physics. Agriculture physics, as you know, is a very important subject because it deals with strategic research, and our division has been given the mandate for conducting the strategic research, postgraduate research, and teaching and training for understanding the physical processes of soil, crop, and atmospheric continuum. And in the Division of Agriculture Physics, during the year 2022, about 9 students have graduated. 5 of them are in the MSc and 4 PhD students. And their area of research covers various subdisciplines like biophysics, crop pest forecasting, soil physics, crop simulation modeling, agricultural meteorology, and remote sensing. And the first student, MSc, Ms. Sudipta Basu, she worked on weather-based forewarning models for potato. Sorry, Deepthi Joshi, she worked on the weather-based forewarning model for potato leaf blight incidents. She used the NASA Power daily weather data, and she combined it with the pest incidence data for the past 15 years. And based on that, using the machine learning approaches, she characterized to forewarn the incidence of disease in a plain area, a coastal region like Kalyani, and in a hilly region like Srinagar. In her study, she found that the method using SPM performed to be the best, and also for Kalyani, the method using the lasso performed the best. In addition, she also identified the maximum minimum temperature and solar radiation to be the important parameters for forewarning the disease, and the hybrid methods like using hybrid models like lasso SPM and SMLR SPM also considered to be one of the best strategies for forecasting the disease. And student Sudipta Basu, she studied on the effect of tillage, residue, and irrigation nitrogen management on soil physical properties and root growth in wheat. She very meticulously characterized the soil properties and also the root growth in almost about 48 treatments involving the conventional and no tillage system with different residue, irrigation, and nitrogen management. In a study, this is the 7th year of this experiment, she observed that even though there is not much of changes in yield, but definitely there is a lot of change in the water use efficiency under the tillage practices, and also the effect of nitrogen and irrigation is also found to be very significant. She found a correlation between the different soil physical parameters and observed that bulk density seems to be having a very important effect on this. She did a meta-analysis, and in her study, she observed that the bulk density under the layer of about 30 to 45 seems to be more in case of no tillage compared to CT. And similarly, the water stable aggregates in the 0 to 15 layer and the mean weight diameter in 0 to 15 layer found to be having a significant change. And when it comes to correlation of different parameters, especially with root length, root parameters, she observed that the ET seems to be having a very significant effect on all the root parameters and also the nutrient use efficiency. The next student for his MSc, Devjyoti Resh, he worked on the evaluation of crop growth models to optimize the sowing dates of mustard cultivars. Devjyoti worked on 2 mustard cultivars, and also he had taken 3 cultivars with 2 different dates of sowing. And in his study, he used Aquacrop, which is mostly a water-based model, and Infocrop, which is a radiation-driven model. And he meticulously calibrated and validated the model in terms of its yield and biomass prediction. And he later used this model to identify the best date for sowing the mustard cultivars. In his study, he observed, but when the sowing is earlier than September 15, there seems to be a significant reduction in mustard yield. Similarly, when the sowing is delayed after November 15, there seems to be 30% delay, loss in yield. And any sowing after December really leads to a 60% loss in mustard yield. And both the models seems to be performing very good in understanding selecting the best date of sowing for mustard cultivars. Later, the student Avinash Goyal, he worked on mustard yield prediction by machine learning and also by crop simulation models. Mr. Avinash, he took 3 dates of sowing and also with 3 different mustard cultivars. He also studied or characterized the mustard yield growth in 5 different zones of Rajasthan. And in his study, he clearly showed that by combining the variable extraction method, using PCA SPM seems to be performing very well in predicting the mustard yield in different regions or zones of Rajasthan. And also when you combine or make a hybrid combination of models, he observed that PCA with ANN and SPM and random forest seems to be the best hybrid model for predicting the mustard yield in IRA and also in the different zones of Rajasthan. He later used this study to see the effect of simulation or predictive simulation in the farmers' condition in different regions of Rajasthan, and he could very significantly simulate the physiological maturity leaf area index and also the biomass and seed yield to the tune of about 15% NRMSE. The next student was Mr. Vimal Kumar. He studied on the spatial scale estimation of evapotranspiration, water productivity in wheat using remote sensing through surface energy balance approach. In his study, he made a field study and also on a large scale study in the IRA farm. And he used the energy fluxes and meteorological observations and also the satellite-based observations from OLI. And later he used this to predict assess the water productivity and also the water use efficiency in a regional scale for the IRA farm. And he meticulously calculated the evapotranspiration using the operational simplified surface energy model for different fortnight during both 2019-20 and also on 2021. And later he validated the prediction of this evapotranspiration with his other model like Bowen ratio energy balance and found to be having a very significant prediction. And he used this to predict or calculate the water productivity and water use efficiency for wheat crop for the IRI. The next student, Miss Bhavya, she worked on the machine learning approach for estimating the reference evapotranspiration using meteorological data. Ms. Bhavya meticulously collected the various weather parameters, almost about 20 years of data she used from IRI, Patiala, Amritsar, Ludhiana, and Hisar. She used the machine learning approach to estimate the evapotranspiration. And in her study, she also estimated the ET naught using Penman-Monteith and Ritchie and other methods. And she observed that the Penman-Monteith method to be the best. And later she used this method to validate her finding to predict using the machine learning approaches. And she observed the random forest method to be the best machine learning approach for estimating the ET naught. And not only this, using the different weather combinations, she observed that the maximum weather combination definitely will give you the best estimate, but using random forest kind of machine learning approach, even with a minimum combination like Tmax and RH1 and 2, she could estimate the reference evapotranspiration to the tune of about 0.04 error. The next student, Abhilasha, she worked for her MSc on the effect of advection on canopy energy balance in irrigated maize wheat rotation system. And in her study, she very meticulously calculated the energy balance using the net radiation, Bowman's ratio, sensible heat, and latent heat. And for this, she made use of the flux eddy covariance tower established in the maize and wheat canopy system, and where she calculated the weather parameters at different heights like 51 centimeter, 1 meter, 2 meter, 3 meter, and 4 meter, and used these to estimate the energy balance. And she meticulously observed the diurnal changes and observed that the radiation seems to be the major factor which gave a very significant change, but there seems to be a lag period for observing the maximum in case of sensible heat. In general, latent energy seems to be the major component of the total energy balance. And in addition to this, she meticulously calculated the seasonal pattern of energy flux and observed that latent energy seems to be a major contributor, but at the time of maturation and harvest, the sensible heat seems to be a very major important component for energy balance. She also calculated the seasonal profile of the daily ET and observed that during a Kharif season, 2.45 mm per day ET was observed, whereas in Rabi season, 1.51 mm per day was observed. The next student, Pragya, she worked on the crop of water use assessment based on surface energy balance using large aperture scintillometry and also using the remote sensing technique. She used the scintillometry which was established in our system with a transmitter and receiver in IRA farm, and later with the help of our remote sensing technique where spatial ET mapping interface was developed by her, and based on this she calculated the regional profile of the changes in the ET and also the yield and also the water productivity for Rohtak, Jhajjar, and even Gurgaon. The next student monitored the in-season crop nitrogen monitoring and management through hyperspectral remote sensing. She meticulously observed the hyperspectral parameters for the wheat and maize at different nitrogen and irrigation levels. Almost about 431 observations were used, and based on the machine learning approach, she observed that the random forest method to be the best method for predicting the leaf nitrogen content. And she used this method for predicting the in-season nitrogen application and evaluating the yield, and the methodology that was developed by her was found to be very significant. And the division has conducted a national seminar on agrophysics, and also the 8th Gildea lecture was delivered by Dr. Ratan Lal. And coming to the academic awards, our students they got a DAT fellowship, IRA merit medal during the last convocation, best oral and poster presentations in various symposium, and UGC NET scholarship also. And our students also got, they proved their talent in track and also in the team events of sports. And students' placement seems to be very good, because during this year, we have struggled to reintroduce the agricultural physics in ARS. And I'm very proud to say that 2 of our students got selected in the ARS in the discipline of agricultural physics. Of course, in the previous year, our students have got selected in soil science and meteorology. Apart from that, the students also got good placement as assistant professor in other universities. Even though we are very less number, but our students' publication is really very good. We got 21 publications, of which I'm very proud to say about 11 publications are more than 9 RAS rating. And our faculty also got good awards like NAS Fellow, Best Scientist Award, Excellent Service Award, and others. With this, I thank you all for giving me a wonderful opportunity. Thank you very much.
Thank you very much, Krishna. And the water waiting for you.
Thank you.
So-
I'll take it. I'm sorry for the. Thank you very much.
Thank you very much. And I know that particularly when you are talking at a speed, you need more water. So anyway, so the next I would like to invite Professor T.K. Das from the agronomy division. Yeah, you start with water. Conservation agriculture, no?
Yeah, sir. इसको खोल दो ना नेक्स्ट के लिए बड़ा करना हो हाँ पूरा स्क्रीन हाँ राइट और भी करो Respected Honorable Dr. Sikka Saab, respected Honorable Dr. A.K. Singh Saab, Director IRI and Vice Chancellor, Dr. Anukumar Singh, Dean and General Education, Dr. Monica Convener, our heads of division, professors, distinguished scientists, ladies and gentlemen, and my dear students. I welcome you all to my presentation. You all know that agronomy is one of the leading disciplines of IRI, fostering excellence in research, teaching, and extension. The mandate of agronomy is to provide leadership in crop and cropping system-based agronomic research by developing new concepts and approaches of sustainable crop production suited to farmer, to develop appropriate crop production technologies to enhance the production and productivity of major cropping system on a sustainable basis, and to provide postgraduate teaching and develop human resource. Teaching program in agronomy started way back in 1945, and then IRI associateship equivalent to MSc degree was given for the first time in 1947, which continued till 1958, when formally IRI IRI MSc and PhD degree were awarded. Till date, IRI has in agronomy has awarded 258 IRI associateship, 363 MSc, and 548 PhD degree. Presently, student faculty ratio is 5:1, and student guide ratio 6:1. And we have 4 principal scientists, 8 senior scientists, and 4 scientists. This year, 24 students receiving degree. They are from IRI New Delhi, IRI Jharkhand, IRI Assam, and AIBSM Raipur. Their 14 MSc and 10 PhD. They work in this different theme area: nutrient management, conservation agriculture, weed management, crop diversification, and irrigation management. And this year, IRI, there are 26 students got admitted to PG program in IRI New Delhi and IRI Assam, 11 MSc and 15 PhD students. And so far in IRI Assam, IRI Jharkhand and NIBSM Raipur, no student got admitted. So our first theme is nutrient management. First student Bipin Dixit worked under the guidance and supervision of Dr. Kapila Shekhavat on the effect of polyhalite on growth and productivity of wheat under rice wheat cropping system. And there the polyhalite is source of potassium, and it also contains K2O 14% and sulfur 19%. Along with this, magnesium oxide and calcium oxide 6% and 17% respectively. And they found, and they compared it with the commercial formulation of potassium fertilizer like MOP, and they found that 100% potassium through polyhalite gave significantly higher grain yield and net return and protein content and nutrient harvest index of wheat than the 100% K application through MOP. They also studied apparent sulfur balance and apparent K balance. Now, higher the K application, higher was the potassium balance, but 100% K through polyhalite resulted in significantly higher potassium sulfur uptake by wheat grain and straw, and which was as per with 100% K through MOP plus sulfur equivalent. And apparent sulfur balance was more negative with 100% K through MOP or sulfur. Then Rakesh Prajapati also studied on the potassium management in Indian Mustard and under the chairmanship of Dr. S.L. Meena. They used two sources like MOP, potassium chloride, and KNO3 as for foliar feeding and MOP for soil application. And they found that 75% MOP as basal plus 1 foliar spray of KNO3 before flowering gave significantly higher yield attributes, yield, quality parameter, and net return of Indian mustard. They also recorded higher oil content, 38%, and for protein content almost 19% in this treatment. Manjunath studied precision nutrient management under the chairmanship of Dr. Ramal Ghosh, and they studied under conventional agriculture system. They use different type of sensor like Nutrient Expert, Green Seeker, then SPAD, as well as the LCC, and they found that SPAD added nitrogen application led to significantly higher grain and soil and harvest index of wheat, but Nutrient Expert plus Green Seeker added nitrogen application gave higher protein content and yield, net return, as well as net benefit cost ratio. Krishna Babu, similar nutrient management, precision nutrient management studied under the chairmanship of Dr. Rajiv Singh in conservation agriculture, and they had CT without residue, zero tillage, and permanent raised bed with residue, and they are combined with nutrient management system. They found that permanent raised bed with residue plus Nutrient Expert plus Green Seeker based nutrient management gave significantly higher yield. Similarly, this zero tillage plus residue and permanent raised bed with residue significantly improve the bulk density of soil as well as soil organic carbon. And then this zero tillage plus residue and Nutrient Expert plus Green Seeker treatment had highest nitrogen uptake, but permanent raised bed with residue and state recommendation, soil test based recommendation had higher nitrogen uptake. OM Sarkar, under chairmanship of Dr. Tikam Singh, studied nano. In fact, 3 students studied on the nano urea, and here he studied in nano urea with the herbicide interaction. And there they recommended dose of nitrogen. They supplemented 50% and 75% RDN with nano urea tubes and 1 nano urea spray. But they found that 100% RDN gave significantly higher values of air-bearing tiller, root weight, soil microbial biomass carbon, grains per pike, and total nitrogen uptake. It also gives 41% and 18% higher grain yield over 50% RDN plus 2 nanometer spray and 75% nitrogen plus 1 nanometer spray. Similarly, this was validated by, they also found this RDN gave significantly higher agronomic efficiency as well as apparent recovery efficiency. Abhijit Mandal studied the nano urea irrigation regime on wheat in Jharkhand, Eastern India. This result again got further validated, and there they used 3 irrigation regime and combined with the nitrogen application, And they found the same thing, that 100% RDN with the 100% nitrogen gave a higher yield compared to the nano-urea spray treatment, and 5 irrigation, 3 irrigation was superior in terms of yield and other compared to 2 irrigation. So much also Mutali, again, this study nitrogen management for acid soils of Jharkhand, acid soil nitrogen efficiency is lower. For that, they combine nitrogen application with the FIM as well as nano application, and they found that 100 kg nitrogen plus FIM or 2 nano-urea gave higher wheat leaf area, yield, length, this is the spike length as well as yield, and urea plus FIM or urea 120 kg plus FIM or urea plus nano-urea led to 14%, 12% and 9% increase in wheat grain yield over urea alone. They also studied the economics break-even point and optimization nitrogen dose, and they found that economics analysis revealed that 97 kg nitrogen applied to urea plus FIM or urea plus nano-urea was as good as or was as efficient as 120 kg nitrogen recommended dose. So there was a clear saving of 23 kg nitrogen, and the response to urea plus FIM, urea plus nano-urea was 12, 14 and 15 kg grain per kg nitrogen applied. So Tarun Sharma, he presented yesterday and he received the IRI merit medal and best student of IRI medal. And he worked on 12-year-old 100% agriculture experiment pigeon pea wheat cropping system. And there was permanent narrow bed, broad bed, flat bed with and without residue compared with conventional tillage. And they found this shear-based system gave higher yield, higher net return, and as well as there was higher water productivity. Particularly broadbed system with, and then there was a clear saving of 25 kg nitrogen. After over a period of 12 years, we got this saving of 25% nitrogen. And there also that weed diversity, that CNCT, there was diversity weed has increased, but that is a shift in weed flora. In CS system, diversity is more, and that's why he also studied the energy use efficiency, which was lower under CS system because of residue energies involved. And total organic carbon also studied, and particularly CS system, they were much higher. And then active and passive carbon pool in 0 to 5 and 5 to 15 centimeter layer was found, evaluated, and that was also higher. And nitrogen and phosphorus and potassium buildup also in soil over a period of 12 years, there was a 23% increase in nitrogen and 32% in phosphorus and 25% in potassium buildup was there in the CS system. Then meanwhile time worked under chairmanship of Dr. Raj Singh, and they worked with this strip intercropping and planting method combination, and they found that wheat plus chickpea strip intercropping led to significantly higher system yield, water productivity, land equivalent ratio, as well as microbial biomass carbon, dehydrogenase activity, and fluorescein diacetyl activity in wheat over soil or wheat or mustard. The rich fermentation was superior. Then VM Raju, the nutrient wheat management, also very another important area of agronomy, and particularly for winter pulses, we don't have registered herbicide for controlling weed in this, particularly post-emergent herbicide. So that's why they tried different combination of herbicides for weed control in chickpea, and they found that pendimethalin followed by hand weeding 30 days was most effective, controlling almost all weed, but more economic was the application of pendimethalin followed by imazathapid. And imazathapid and imazamox, another combination that was very toxic, although it was very efficient against weed, but it was toxic to chickpea also. Similarly, Chinnalidas worked on integrated weed management in vegetable pea under the guidance and supervision of Dr. Ramanjit Kaur. They also studied different combination of herbicide, and they found pendimethalin plus mulch followed by ready mix of metribuzin plus chlorinafor was better and more effective, and they are as good as mechanical weeding, and gave a significantly higher weed control efficiency, lower yield loss, as well as significantly higher green pod yield. And there was no there was residual level of this herbicide in green pod of pea as well as soil was below maximum residual level. Then Poulomi at ICAR NIBSM Raipur, Poulomi Devnath worked under the chairmanship of Dr. Anil Dixit, and they studied that nutrient and herbicide interaction in wheat. Their situation little different. Their wheat problem start from the germination of the crop in wheat crop. So they need to apply pre-emergence herbicide. Otherwise, we don't recommend pre-emergence herbicide for wheat control in wheat. But here they started from the pendimethalin as pre-emergence, and they found that 100% RDN along with pendimethalin followed by clonazepam metsulfuron gave higher weed control efficiency comparable with the pendimethalin plus sulfosulfuron plus metsulfuron, which is recommended change. Then Mukesh K. Yadav, he worked under Dr. Dinesh Kumar because IRA has developed several very important high-yield varieties like HD3226. It is having some lodging tendency and it is sensitive to lodging. For that, agronomic management was sought so that agronomic management can reduce the lodging of this variety, and that's why they studied to variety, to nitrogen doses, to irrigation regime, as well as no with and without plant growth regulatory application. And they found that HD3226 with 100 kg nitrogen plus hectare with timely irrigation and without teaser application improve growth parameter as well as yield of wheat, and no lodging was noticed. And lodging parameter, particularly bottom stem and lodging score, that were lower when the varieties were treated or applied with TGI. Now, first PhD student Somnath Nayak work under the chairmanship of Dr. Y. Sibe, and they studied relative zinc loading ability of rice and wheat variety in the rice wheat cropping system, because you know zinc malnutrition is widespread in the world, and that's why zinc biofortification is being studied genetically or agronomically. Agronomic fortification is mainly due to soil application or supplemented with foliar feeding, and then they studied with the different varieties of rice. They used two zinc fortified variety of one for rice DRR 45 and one PBW 1 zinc for wheat, and then found PUSA 44 and PBW 1 zinc with both soil and foliar application of zinc. They got significantly higher grade yield, but PUSA Basmati 1509 and PBW 1 zinc gave higher net return, and higher phytic acid in grain was accumulated in PUSA 44 as well as HD 2967. Soil foliar application gave 17% and 34% higher protein content in white rice kernel and bran. Same thing, there's different grains of rice like with rice kernel, bran, husk, and straw. They found that DRR Dhan 45 with this foliar as well as soil application of zinc led to higher zinc content in this different rice part, economic part. But there was a problem of antagonism. When higher zinc content means there is a P content, there is antagonistic effect of zinc and P. So P content and accumulation was lower. Then Rajendra Prasad Meena under the chairmanship of Dr. Sivadar, they work under nutrient management baby corn based cropping system. And there are 3 baby corn based cropping systems: whole baby corn and baby corn, followed by baby corn plus fenugreek, followed by cowpea or sweet corn. And they found these 2 diversified systems. They are more profitable, more remunerative than the baby corn system, as well as that's the 75% RDN when applied with supplemented with 25% RDN through FO and gave significantly higher net return. They also studied the MDC and dehydrogenase activity and other so many soil physical chemical parameters, and they found this diversified cropping system had higher values of microbial biomass carbon, dehydrogenase activity, urease activity, alkaline phosphatase, acid phosphatase, aryl sulfatase, as well as beta glucosidase activities than soil based system. Then Ankur Bhakur under the chairmanship of Dr. Y. Singh studied on the effect of nutrient with management on productivity and soil microbial properties in aerobic rice wheat system, and they found that 100% RDA gave higher aerobic rice wheat and system productivity system yield than 75% RDA plus biofertilizer. But the pyrazole sulfuron followed by bisphenol followed by almix gave higher root control efficiency, root control index, and higher rice yield. But soil plus this sulfur based sulfuron in wheat was more effective against root control in wheat. They also studied different biological parameters, and they found that 75% RDA plus biofertilizer consortia in rice and wheat led to higher values of dehydrogenase activity, acid phosphatase, FD activities, and soil respiration. But 100% RDN had higher UDAs, nitrate reductive activities, ammonia, and nitrate oxidizing bacteria activity. Hari Singh Meena, under my chairmanship, studied under conservation agriculture, tillage received with management effect in onion wheat cropping system. This is one of the, could be a one diversified cropping system for rice wheat. And he studied also this, he found that zero-till raised bed plus residue led to significantly higher system productivity and system net benefit cost ratio. Pendimethalin followed by Quizalapur or 2 hand weeding gave significantly higher system productivity and system net benefit cost ratio. Zero-till raised bed with residue or zero-till flat bed with residue has had significantly higher workland black carbon, and mean weight diameter of soil aggregate than the city. They also studied this several biological parameters, and the CFS system was superior. And Harishankar Nayak yesterday presented for gold medal. He has been awarded with IRI merit medal, and his team of 25 enumerators surveyed around 11,000 farmers in rice food system from in Punjab, 7 districts of Punjab, Haryana, and they had questionnaire. Based on this questionnaire, they collected data, and their key findings, some of the key findings are small rice yield gap was noticed, almost 20 to 25% of the potential yield. Observed in district of Punjab, Haryana, most yield gap was due to technology yield gap. The 95th quantile regression showed a plateau response to apply nitrogen irrigation, and classification to high, low, and medium yielding farmer and average yielding farmers suggested that different groups of farms apply nearly same amount of nitrogen but different quantity of irrigation. And there some of the key findings that crop residue retention benefited at low nitrogen dose, seed rate, and disease infestation, and nitrogen response was low with the residue retention and at low nitrogen dose. So residue retention effect on yield was prominent, and then minimum temperature was the most important biophysical variable in the study. Komalgar, under chairmanship of Dr. Shivadhar, he studied this the development and evaluation of formulation of organic sources, and then there are 2 cropping systems, baby corn based and pigeon pea based cropping system. They use different organic formulation prepared by their own, and that is rice residue compost, then ready paddy husk ash, and then potato peel compost. And they found that baby corn, carbohydrate, and vegetable cowpea system had significantly higher system productivity, net return, and net BC over pigeon pea wheat system, and PH, potato paddy husk ash based formulation, 100% nitrogen gave significantly higher values of all these parameters. They also studied different DHA, soil available nitrogen, soil organic carbon, and microbial biomass carbon. And this PHA based 100% nitrogen recommendation gave significantly higher values of DHA in all the 3 seasons. And MBC then controlled, but PPB based RDN and FOM were comparable. Ankit Tiwari, under the chairmanship of Dr. K.S. Rana, worked on conservation agriculture and sulfur nutrition, on productivity and resource use efficiency of rainfed pulp millet. And they found that CFS no tillage crop residue management system led to higher growth, yield, grain quality, as well as profitability in pulp millet. And similarly, the foliar application of ammonium thiosulfate at 10 ml per liter twice, and led to higher values of this parameter, and that was very, very remunerative also. They also studied this SPAD index and DBI protein index, and they are also same almost similar report with respect to conservation agriculture based system as well as this ammonium thiosulfate application. And then they also found that NTCM led to lower soil bulk density, penetration resistance, and showed higher potential to conserve moisture, utilization of renewable energy, as well as increase the moisture use efficiency by improving conservative use of water of rainfed pulp millet. And Neeraj Vishwakarma, under the chairmanship of Dr. Vijay Punia, worked under ICM, Integrated Nutrient Management in the rice food cropping system. They had 4 ICM module that was tilled like your CT based, and 4 was zero tillage CA based, and they found that ICM's 8, which was zero till DSR with the wheat residue and mung residue and 75% RDA, and then NPK plus biofertilizer and that is MF fungi, and then ICM was similar treatment. They found ICM 8 and ICM 7 based rice food system led to significantly higher yield of rice and wheat, system productivity, system production efficiency, system monetary efficiency, as well as net return, then other ICM system. Similarly, the improvement in energy use efficiency and CFS system was lower compared to city system, but water stable aggregate, active carbon pool, dehydrogenase activity, SMDC was higher under ICM module like 7-8, ICM 7-8. Similar ICM module was studied under maize plus black gram intercropping, followed by wheat cropping system, and there they use 4 CA-based ICM and 4 city-based ICM and compared with another organic agriculture-based ICM. And there they found that CA-based ICM 7 led to significantly higher system productivity, system total water use efficiency, energy use efficiency, and net return. Similarly, maize diagram system was higher, and then they also used the licensed tomato content and system carbon budgeting. All these things were studied, and there was significant improvement in the CA-based ICM compared to this. Then Rishi Raj, he studied under my chance. He also presented yesterday for gold medal, IRI medal, and his participation is highly appreciated. And now he also in rice wheat system, this is the 8-year-old rice wheat system, and he found that there was a double zero till system, triple zero till system with or without residue. And we found that triple zero system with residue gave 13% lower rice yield, but 15 and 37% and 37 and 51% higher wheat yield and system productivity and system net return. And there is a pyrosulfuron followed by Silapa followed by Bispalvac. Then chlorinafa plus carfentrazone wheat resulted in significantly higher system yield. He also studied this root knot nematode first time in the rice. That is, one root knot nematode was observed in wheat Dineba retrofloxa. This is first time report. And then wheat management was in a CS system when Mummin was there. The nematode infestation was drastically reduced. And herbicide application that reduced the nematode infestation by almost 74% by reducing the weed infestation. So herbicide indirectly by controlling weed can lead to weed, nematode management. So if weed management is there, nematode could be managed also. And then there are several soil physical, chemical, biological parameters also studied. And there was also favorable impact of CA compared to CT was observed. Now our division last year there was ICRH training program. I was the course director, and then another ICRH training program by Dr. T. Kamsi. We had national level campaign on efficient balancing the fertilizer, village Kullan, Tehsil Assam, district Karnal. Then 1 month internship I provided to one student, second year BSc agriculture student of Punjab University on conservation agriculture. Then our research publication, there are 31 total publication, and 5 are in greater than 11 NAS rating, 2 9 to 11 NAS rating, 3 7 to 9 NAS rating, and less than there are 21 publication is 13 in 6 to 7 and 8 in 5 to 6. Our student, particularly MSc student, they are mostly pursuing PhD, and MSc PhD student, they are all well placed. Our student also got placement 28 student. There are ICR technical T6 student.
You are run out of time now.
Just one minute. Then NEBAR 2, the National Fertilizer 2, and then your State Public Service Commission 11 student got placed, and others 4. This is our student recognitions that Kiranmay Patra got Fulbright Nehru Doctoral Fellowship, University of Florida. And then these students only, so they are having, they have eminent student award, UGC GRF, UGC NET, then best poster award 7, certificate of appreciation from Director IRI, and then this is the annual sport. Almost our majority students participating IRI annual sport meet, and they have received several prizes from the IRI authority, and this is IRI cultural.
I think please now conclude, because otherwise you will be encroaching in others' time.
Award, this is the award received by the faculties, and this is all these. I thank, I acknowledge Director.
Thank you,
Dean and
Professor.
Education, Director Research, Director Education.
Thank you, Professor Das. And I know that the time is.
Thank you, sir.
Thank you.
There are 24 students. That's why.
Yeah. No, thank you very much. Let's give him a big hand. And next, my privilege to invite Professor Dinesh Kumar Sharma from the Environmental Sciences. And I would request you to be very precise, because everybody is talking about precision farming, precision irrigation, but they have to be precise also in their delivery.
Honorable Chairman of the session, Dean Ma'am, convener of the session, my senior colleague, Dr. Deshmukh, head of the division, fellow professors, scientists, and dear students, I am here to present the salient academic findings of Division of Environmental Sciences. As you know, the environmental science is a multidisciplinary science, and then we have 10 disciplines in environmental science like plant physiology, biotechnology, economic botany, agronomy, agriculture, chemicals, agroforestry, microbiology, soil science, and organic chemistry. So it is a multidisciplinary. So far 98 MSc students has been graduated from our division and 56 PhD, and students on roll MSc 27, PhD 39, and our students are placed in ARS, SAU, CPCB, State Pollution Control Board, FCI, DDA, CSIR, Ministry of Environment and Forest, Central Universities, UPSC, NABARD. And this year, all 4 seats has been occupied by our ARS seat has been occupied by all our students. So these are significant research findings I am going to discuss. So the areas of student research is climate change, waste management, ecosystem services, and environmental biotechnology. In climate change, we have 6 MSc students. Those who are going to get their degrees, 3 PhD students. In waste management, 2 MSc students are going to get their degree. In ecosystem services, 1 PhD student, and in MSc, 1 environmental biotechnology student. So coming to, I will start my presentation with Ram. So student Ram Kumar worked with Dr. Aarti Bhatia on the effect of ozone and carbon dioxide interaction on growth, yield, and greenhouse gas emission in rice. So he has used 3 rice varieties, PRS10, PUSA 44, and PUSA 1509 under different ambient CO2, ambient ozone, and different interactions. So what he observed that highest emission in both the years was observed under elevated CO2, and the least emission was observed under elevated ozone, because ozone has inhibitory effect. And PUSA 44 was having highest emission among different genotypes, and PUSA 1509 was having lowest emission. And similarly, N2 emission in 2016 ranged from 1.25 kg per hectare in PUSA 1509. And in 2007, it ranged from 1.21 kg per hectare. In both the years, the average cumulative N2 emissions was highest under elevated CO2, because CO2 favors the growth. That's why the more emissions was there, and the lowest emissions was there in elevated ozone. And then infocrop rice model satisfactorily simulated the growth, development, and yield of the rice crop. And elevated CO2 could potentially increase the methane and nitrous oxide emission. So these are the main findings of that particular study. So coming to the next students, Amita Raj worked with again Dr. Aarti Bhatia, and they try to compare what is the how this eddy covariance and closed chamber techniques are different with the following 3 objectives. I'm not going to read all these things. So they first time in the IRI, or I think in India, so they first time use eddy covariance technique, which is a rather advanced technique, and they compare with the closed chamber technique. The collection of data, flux calculation, and correction is very, very important for this particular eddy covariance. And they found that, and they also use Eddy Pro software. They found that net ecosystem exchange of CO2 was estimated to be -441 gram per meter square for rice, and for wheat it was -588.56 gram per meter square. It means that more carbon sequestration was there in the wheat, and then contribution of rice crop to the annual cumulative values for gross primary productivity and ecosystem respiration was found in the 46.02% and 46.47%. And the take home message for this particular study was the rice wheat cropping system roughly fixed 300 kg carbon per hectare per year. And then an overestimation of 14% of methane flux was recorded by the closed chamber method over the eddy covariance, because closed chamber technique, they are having this closed chamber and the temperature is more, so more methane emission was recorded. And then another study, the quantification and simulation of maize wheat green gram cropping system response to FYM, nitrogen, phosphorus, and climate change. This work was done by Alimoy Kamara. Is a Senegal student. He worked with Dr. Naresh Kumar, and they try to develop the maize wheat green gram cropping system module in the infocrop. The infocrop cropping system module used for the calibration and validation of the model for simulating climate change practices impacts in specific locations, Ludhiana, Karnal, Delhi, Lucknow, Varanasi, and Patna. So, and they found that the system sustainability yield index was highest in F10, 9, N180, P30, followed by F10, N120, and P0 treatment. The application of 10-ton FYM can significantly reduce the nitrogen and phosphorus application requirement to the system by 25% and 50% respectively. And then the infocrop maize wheat green gram cropping system model can simulate the growth and yield of the system. So this is the main finding of this particular system. The climate change impacts are projected to increase the overall productivity of the maize wheat green gram cropping system in 2020 climate scenario, but with the reduction in wheat yield and environmental trade-off. So yes, Dinesh GK worked with me on the quantification of ecosystem services in conservation agriculture maize wheat mung bean system. Yesterday he also presented for merit medal presentation. So in that he used 10 years old conservation system where the main plot was zero tillage with residue, and then subplots were different nitrogen treatment. So he tried to work out that, okay, what kind of provisioning, regulating and cultural and then supporting system ecosystem services were generated by conservation agriculture. And then he find that overall the zero tillage without residue plot can generate 5.49 lakhs of ecosystem services, whereas zero tillage with residue can generate 6.10 lakh of ecosystem services. So over and all, or you can say 60,000 rupees of ecosystem services were generated in conservation of agriculture system, but the market is not there for that activities. So therefore, the concept of payment of ecosystem services is a very, very important. And another students, Miss Apurva MS, she worked with Dr. Aarti Bhatia on interactive effect of elevated CO2, temperature, and nitrogen fertilizer on gaseous nitrogen fluxes and nitrogen use efficiency in wheat. So the student used various nitrogen combination like neem coated urea, sulfur coated urea, and liquid nano urea, and different parameters she studied under elevated temperature and ambient CO2, elevated temperature and elevated CO2, ambient temperature and elevated CO2. So different combinations were tried, and then ammonia emissions was higher after first dose of fertilizer application. All the interactions treatment emissions after nano application were non-detectable. So this is the beauty of nano fertilizer they have observed. And then elevated temperature increased both ammonia and nitrous oxide emission in the treatment. Nano reduced the ammonia and nitrous oxide loss significantly under all CO2 and temperature interactions. So again, Anushka, Anil worked with me on the impact of elevated ozone, carbon dioxide, and their interaction on growth, yield, and reproductive behavior in Indian mustard. In that study, she used two mustard variety, Pusa Bold and Pusa 30, and she wanted to study that how this elevated ozone is going to affect the reproductive biology of how these behaving differently under different ozone and interactions. So the philosophy behind this experiment is that, okay, under ozone concentration, oxidative stress is generated. Whether these oxidative stress, because another my PhD student is working in how this oxidative stress is affecting the pollinators' behavior in the mustard, like honeybees. So she studied various reproductive biology parameters like pollen viability, pollen germination, pollen tube length, and stigma receptivity. So she tried to work that under which variety is having more stigma receptivity even under elevated ozone conditions. The reproductive biology study showed that Pusa 30 mustard variety showed more receptivity of stigma and viability than Pusa Bold. The high concentration of ozone in the atmospheric showed a negative exposure to the reproductive development of the crop, and it is due to the oxidative stress by the elevated ozone. There was an intermediate response in the interaction between elevated ozone and CO2. So, and then the percent yield gain under elevated CO2 was 12.15 in Pusa Bold and 7.1% in Pusa 30, then ambient. Under elevated ozone, 8.9% yield reduction was there in Pusa Bold and 12.06% yield reduction was there in Pusa 30. So another student, Mr. Y. Sandeep Kumar, worked with Dr. Navita Jain on the greenhouse gas emission under different rice residue management practices using Pusa decomposer in succeeding wheat crop. It is a very emerging area, and in that they have used various combinations like full residue burning, residue retention, then full residue incorporation, full residue retention with Pusa decomposer and full residue incorporation with Pusa decomposer. These were the treatment, and they want to see that how much emissions is there in the form of CO2, nitrous oxide in the succeeding wheat crop. So they observed that on-farm burning of 9.3 ton of rice residue lead to the emission of 10.91 tons of CO2 equivalent and 668 kg gaseous air pollutant and 33 grams of particulate matter. Effect of Pusa decomposer on emission of GHG under different residue management practices in succeeding wheat crops. They observed that the highest grain yield was also recorded in full residue retention plus Pusa decomposer. So this is a main finding of the study. Another student, Mr. Rishabh Srivastava, worked with Dr. Navendu Gupta, and he wanted the effect of bioslurry on soil properties, yield and nutrition quality of P. To study the effect of bioslurry from admixture of cattle dung and paddy straw, they have used in various proportions, and to assess the effect of bioslurry on the growth and nutrition quality of P. So different combinations they have tried, and they have substituted nitrogen urea, chemical urea with the bioslurry. And they observed that the economic analysis turned out to be the better for split treatment over the 100% biogas slurry and recommended dose of fertilizer. All the quality parameters like total soluble sugars, crude protein, total sugar content, and reducing sugar recorded highest value in the T4, where they have substituted 25% of urea with the slurry. In another study, evaluation of antifungal potential of lacto-fermented agriculture byproduct. In this study, Mr. Azhar Mahmood worked with Dr. Anita Chaudhary, and they have isolated roughly 51 bacteria from the phyllosphere of the different plant species, and they have characterized the cell-free supernatant for the determining antifungal potential for selected lactobacteria. Methodology they have used, and they have also studied the inhibition of fungi using well diffusion assay, and the conclusion of the study is that total 51 lactic acid bacteria were isolated, and from the different wheat, chickpea, strawberry, and orange peel, and among them one strain Lactobacillus fermentum was selected and identified using 16 srRNA, which was active not only limiting the fungal growth but also different antioxidant activities. So another student, Mr. Sadashiva, we worked with Dr. Ajay Kumar Singh and Mia Sen, and the focus was to develop water stress tolerant soybean genotype with using molecular techniques, and they have screened 15 soybean genotypes. The promising soybean genotypes TGX 854-429 and TGX 539-2D7 exhibited efficient photosystem second as compared to the drought tolerant Czech varieties. I have already discussed all these things. So the next student, quantification of greenhouse gas and ammonia gas emission from different cattle manure management practices. They studied that how this indigenous breed and the exogenous breed are different from as far as methane emission is concerned from their cattle management. So these are different methods of composting, stockpiling with turning and vermicomposting, and this is the layout of the treatment. And then there was a significant difference in the quality of fresh cow dung manure in the terms of pH, EC, nitrogen, phosphorus, potassium. There was a significant difference in GHG and ammonia emission from manure of indigenous and crossbreed cattle. Crossbreed cattle manure shows significantly higher emission as compared to the manure of indigenous cattle. So this was the main finding. So coming to the another student, he quantified.
You run out of time.
Sir, I'm just.
Then we are encroaching on others' time and also next session.
Okay. So they quantify this, how much emission is there from native forest, agroforestry, agriculture upland, and agriculture lowland. So they say that, okay, the highest soil carbon and nitrogen stock was there in agroforestry system. Native forest has the lowest greenhouse gas emission from among the different land use system. The last student, sir, this work with Dr. Himanshu Pathak on to quantify the carbon sequestration potential in different fruit-based system in semi-arid environment and to assess the sink source of greenhouse gases. In this study, they have quantified how much carbon can be sequestered under degraded environmental conditions, the basaltic soil, the semi-arid regions of Baramati, and 8-year-old fruit trees were harvested and to develop biomass equations, carbon stock and sinks, and then net GHG potential. So this is how higher carbon sequestration rate was found in mango, followed by pomegranate and coconut. So this is all about the conversion of basaltic rocky degraded land with multi-base multi-fruit-based system has a mitigation potential of 164 tons carbon dioxide equivalents per hectare per year, whereas this coconut 68 tons per hectare per year, and then pomegranate. So I'm coming to the awards and fellowship to the students. Like this is the list, and our students got best poster award, best thesis awards. So this and these are the some pictures of our students, and our student got elsewhere Young Scientist Award in Gujarat, 200 euros they get, Naveen Malik and Vinita Madula, and one of our students got Newton Bhabha Atomic Fellowship in Edinburgh. And then awards and recognitions to the faculty, and our faculty, Dr. Manoj Srivastava, get Fulbright Nehru Fellowship in the US, and then Dr. Nimita Jain, she is representing India in the IPCC, and she is also reviewing the UK report, and others are. So these are some of the pictures of our faculty. So the impact factor that we have, our students published 12 papers. The maximum NAS rating was 12.79. Our faculty published 22 papers, and then more than 13 was more than 9 rating, and the highest NAS rating was 20. So and this 2 training program was organized by our division, and I want to thank the students, the faculty, staff, laborers and farmers, and I want to thank IRA authority, the honorable director sir, dean, associate dean.
Thank
you sir. Thank you.
Thank you. Okay, thank you very much. So I invite next Professor Rajiv Kaushik, because I have another appointment at 4:30.
Yes, sir. I will try my level best, sir. Respected Chairman,
Dr. Kaushik,
Sikka.
kindly, skip the last part of it, only just showcase and then.
Okay. So, good afternoon, everybody. I am representing Division of Microbiology, and we have 17 faculty members in our division, out of which only 15 are eligible to guide the students, and out of that only 8 are eligible for guiding the PhD students. So we have on rolls total number of 42 students, out of which 11 are MSc and 31 are PhD students. We have faculty publications 65, out of which more than 10, 19. Similarly, student publication are 17, out of which more than 10 are 3 and 8 to 10 are 7. And our faculty has got awards, like Dr. Papi has been elected as President AMI. Dr. Lavleen Shukla Govindswamy has been awarded Best Young Scientist and Women Researcher Award at Asian PGPR Society. Dr. Meenakshi Grover has done well in Hindi Chetna Mas. And training program, Dr. B. Ramakrishna has organized 2 trainings, and Dr. Lavleen Shukla and myself, we have organized the training to the licenses of Pusa companies who have taken the license of Pusa Decomposer, Pusa Sampoon, and Liquid Boil Potash. Students also have attended different NEHP cast trainings and different training organized by NBM and again by Delhi AMI, Association of Microbiologists of India, and all the students have attended AMI conference. Students have got award in the various platform, different like Best PhD, MS Swaminathan Award, Best Poster, Best MSc Thesis Award, and Young Microbiologist Award at various platforms. And our 4 students have been got placed at very well in the guested officer scale. 2 have been selected as scientists. 1 student who is at the guest faculty at RPCOA has now been selected as DRDO scientist. Sir, I am going to present for the 15 students, out of which 10 are MSc and 5 are PhD students in the broad 3 theme areas: plant growth stimulation, nitrogen nutrient mobilization using beneficial microbes, microbe-mediated abiotic stress management, agri-biomass degradation, and value addition using microorganisms. First student, Mr. Yadesh, under the chairperson, and Dr. Meenakshi Grover, has used the co-inoculation of azospirillum and bacillus for improving the growth and yield of pearl millet under moisture deficient conditions. He used the plant growth promoting bacillus and azospirillum, which were when grown together had a synergistic effect on plant growth promoting ability, and they also improved at different water potential, which was created by different concentration of PEG. And this consortium improved the seed germination at 20% PEG. And when they did pot experiment, they found that the dual inoculation of both the bacteria significantly improved the all plant parameters and under moisture deficit conditions. Another student, Miss Harita, worked under the chairmanship of Dr. Bhaumik. Actually, sir, the mycorrhiza, which is an obligate symbiont, and our division is selling the inoculum to the farmers. The main problem hurdle is because it is an obligate symbiont, we have to design a media in the lab where we can culture it in a pure cut form, so they can grow in the carrot roots which have been transformed with the tDNA. So Dr. Bhaumik has devised a media in which the mycorrhiza can be grown in the lab, and he optimized the media by adding certain components, like when he added streptomycin, the pure culture of mycorrhiza could be obtained, and also in the minimal medium he obtained the maximum root length, and in the presence of phytohormone auxin and cytokinin he obtained the best colonization of the carrot roots. Similarly, in the presence of phenolic compound catechin he again found the best colonization. So in the conclusion, he has devised a media with certain phytohormones and phenolic compound by which he can culture the mycorrhiza in the in vitro condition. Another student, Ms. Rupa Kumari, worked on the production of carotenoid pigments from different cyanobacteria. She collected cultures from cyanobacteria, heterocystus and non-heterocystus cyanobacteria from the CCBGA. She characterized them for carotenoid production, and she found that the nostoc species heterocystus, which is heterocystus, was the maximum carotenoid producer, and spirulina, which is non-heterocystus, was the maximum carotenoid producer. So she optimized the media by modifying the physiological conditions and nutrient condition, and she found that spirulina species grew best by 2.4 fold increase in carotenoid production was obtained when optimized conditions were used. Similarly, nostoc, only the NaCl when concentration was added to that, there was a significant jump in carotenoid production. Another student, Mr. Manoj worked similarly on the similar aspect of the CC of the algal cyanobacteria, but here he used a spirulina culture for developing a probiotic yogurt. Basically, all the spirulina strain which they collected from the cyanobacterial collection, they screened for certain components, and when these cyanobacteria they screened for the best growth and production of different compounds. So they selected CC477 strain, which grew faster and produced all the parameters, the nutraceutical parameters required for the yogurt preparation. They added this spirulina culture in 3 different concentration, and they found that when at 1% concentration, the yogurt there was an increase in 1.19 milligram of calcium, 2.74 microgram of iron, and similarly 0.06 microgram of zinc. They also tested the shelf life, and it was found that 6 days was at which all the taste and the lactobacillus population was maintained for the 6 days. So this is a bio-fortified yogurt they could prepare. Then another student, Jahid Hassan, worked on the phycobilly proteins from Oscillataria species. They collected large number of Oscillataria from the algal collection, and they screened for the phycobilly proteins. And among different strains, they selected one strain, and they optimized the nutritional parameters. And maximum phycobilly protein was observed at pH 8, normal light, and of 1.5 RD and Mg, magnesium 1.5 recommended dose. Another student, Coach Hania, he worked on evaluation of native mesorhizobial population and inoculation effect on growth and symbiotic response of chickpea after rice. Basically, they took 2 treatments, transplanted rice and DSR. And after that, when mung bean, they were testing the chickpea, but what was the impact of the transplanted rice and directed rice on the subsequent crop nodulation. So different treatments were taken, and they found that the microbial biomass was higher in DSR and in the soil than the transplanted soil, and prehydrogenous activity was higher in transplanted soil. The overall nodule number per plant was recorded greater in treatment T6, which was the transplanted soil, was better performing than DSR. So basically, they concluded that the transplanted rice, after transplanted rice, the nodulation efficiency is much better than the DSR. And this I've already told. Then another student, Branda, she worked on the assessment of actinobacteria for plant growth promotion in aerobic rice under water deficit stress. And she selected 35 actinobacterial strains and screened for PGP properties. Then they tested for the seed germination assay. And then they found that when they inoculated these actinobacterial strain to the crop, they found that key physiological parameters related like electrolyte leakage, leaf MDA content, peroxidase activity, etc. Were increased subsequently due to the inoculation, and crop yield was improved. Another student, S. Syam, worked with Dr. Sangeeta Pal, and they studied the interaction of stress tolerant bacteria in wheat crop under the dual stress of acid and drought. So they used they collected soil samples from the Jharkhand, from where they isolated bacteria. They screened for their osmotolerance, acid tolerance, and water potential tolerance. And they selected two strains, ANI4 and NNI2. And they found that both isolates NN12 and AN4 possessed all phytohormone production ability. And also when they inoculated in the pot experiment, Jharkhand soil was used, which was having acidic pH, and inoculation with bacterial isolates improved plant parameters, and Achromobacter inoculation resulted in significant increase in shoot biomass. Bacillus inoculation had significant photosynthetic inoculation, whereas dual inoculation improved all the activities. Another student, Sania, she worked on prospecting diversity and abundance of diastrophic soils in Jharkhand soil. And here she again worked on acidic soils, where she used two bacteria, Brucella species and improved the yield of wheat crop in the acidic soil. Then another student worked in the student Shashank God. He used IA producing bacteria and inoculated into the wheat crop, and they found that this Brevibacillus and Bacillus in the presence of IA which they were producing could significantly improve the growth of wheat crop under acidic soil. Then coming to the PhD student, Bhagwan Singh Dhakar worked on the carbon sequestration by the inoculation of AMF fungi, and they found that in the different treatment in zero tillage conditions by incorporating wheat residue, mung bean residue, and rice residue, they found that better colonization was observed in case of when crop residue and brown manuring was done. They could get better colonization of AMF fungi, and DSR with MBR, that is mung bean residue, followed with zero tillage wheat and rice residue, and followed by zero tillage mung bean were the best treatment in which carbon was sequestered and the plant growth was improved. Another student, Dia Roy, worked on the production of cyanobacterial phynoscinin, and she purified and characterized the phynoscinin present from five different algal species, and she found that four medium species were the best in production of phynoscinin, and she characterized purified using anion exchange chromatography. And she studied the different stress condition on effect of this activity of phynoscinin, and she found that temperature at the 60 degree was lethal. Similarly, pH condition 4 was optimum, but 8 was not optimum, and 1% of H2O2 was the activity was declining. And so two more students are left. Shri Niketan Puranik worked on the degradation of phthalate esters, and two phthalate esters like DBP, bis-2-ethyl hexyl phthalate and N-butyl phthalate was used. Two fungal strains were selected, SDB4 and SDPHP2, which were isolated from landfill soils, and they degraded this phthalate to 99% level by incorporating these two fungus. They designed a media using jaggery, and also they tested into the crop whether there is an effect of any toxic effect. They found that there was no toxic effect.
Sir, Rajiv.
Sir, this is last. Then prospecting potential of cyanobacterial biofilm on the biofortification of maize crops by iron and zinc. This Mr. Nishant developed two different biofilms using Anabina and Trichoderma and Anabina prudentia. And by the dual inoculation of both these biofilm, they found that there was a significant improvement in the zinc and iron content of the crop. And also they found when they tested by qPCR, they found that there was upregulation of genes responsible for zinc and iron uptake. A last student, Kokila, worked on exploration of potential of cyanobacterial inoculation under elevated CO2 condition on the tomato crop. So under elevated CO2 condition, when they grew cyanobacteria, they found that total chlorophyll, phycobilin proteins, total sugar, organic carbon, and total nitrogen increased by 14 to 16% in both the cultures under elevated CO2. Similarly, certain enzymes like PEPCase and CA also increased under elevated CO2, and elevated filament length, heterocyst number, nitrogen fixation, biomass comparison with ambient CO2 grown cultures. Then in the last slide, the conclusion was that overall beneficial effect of cyanobacterial inoculation was plus elevated CO2 condition was observed in terms of cyanobacterial metabolic activities and application of CL to soil coupled with stimulation in plant growth quality.
Dr. Kaushik.
Produce and yield. So these are publication of the students, and thank you.
Yeah. Thank you very much, because we have to keep it to time because they here. So thank you very much, Dr. Kaushik, and we invite soil science and Ahmed. So Dr. Precise, doing a precision nutrient placement.
So how much time can I get?
Respected chairman of this session, Dr. Sika, Dr. Anupama Singh, Dean and Joint Director Research, Dr. Monica Joshi, convener of this session, Dr. Deshmukh and Dr. Tyagi, all my respective divisions, professors, my senior colleagues, junior colleagues, students, ladies and gentlemen. So as I have been told to squeeze my presentation to 15 minutes, so I will just try to. So these are the significant, I will highlight the significant educational achievements in the Division of Soil Science and Agriculture Chemistry for the year 2022-23. And the mandate of the division is to carry out research on fundamental and applied aspects. Yes, sir. Applied aspects of physical, chemical, and biological properties of soil. And also we are involved in postgraduate teaching and extension activities. Total faculty position is 18. Out of that, 2 are on deputation, both in US, 1 in postdoc and 1 for PhD. Total student strength is 96. Currently 25 MSc and 71 PhD students. These are the list of students and their research topics. So 13 students from MSc will get their degree this year and 5 from PhD. And these are the students and their thesis topic and the chairpersons. So these are all the MSc students. Then these are the 5 PhD students who will be awarded in this convocation. So the key research areas, what we have focused is soil organic carbon dynamics and stabilization. 7 MSc students and 1 PhD student worked on it. Assessment of soil quality, 2 MSc students. And then synthesis and evaluation of novel fertilizer products, 1 MSc student, dynamics of soil phosphorus, for enhancing phosphorus supplying capacity. 3 PhD students worked on it. Then dynamics on availability of micronutrients in soil. So 1 MSc and 1 PhD student worked. Soil test-based fertilizer recommendation, 1 student, and recommendation remediation of polluted soil water. So 1 MSc student worked on this. So just I will highlight briefly about the work done by the students on soil organic carbon dynamics and stabilization. As we know that this Gautam Padida, he worked on the chairmanship of Dr. Sheila Das, and in Assam he had taken different land uses. The land uses were inorganic tea, organic tea, rice fallow, sugarcane and forest. And he has taken these soil samples from profile. Actually, he has dug profile up to 150 centimeter depth. Different depths, he has studied the carbon stratification ratio, and it has been found that the carbon stratification ratio was highest in the up to 50 centimeter of the soil depth. And this from this study, another thing, he has calculated the carbon stock in this different land use system, and he has found that the highest carbon stock was found in the forest and rice fallow was the lowest. So what we can see that when actually from forest we are converting to other agricultural system, production system, then actually carbon stock is decreasing. But highly managed your organic, inorganic tea, these are actually maintaining the carbon stock. Another student, Ajata Dibhadarshan, he worked with Dr. Sushanta Kumar Nayak. Here also, he has worked on different agricultural production system. Here actually, he has taken the rice, then plum orchard, then mango, mahogany, anola rice, mango, mahogany, peach, rice, different pure agricultural production system. Here he has seen the carbon management index, and here he has found that the highest carbon management index was recorded in multi-tier production system, whereas lowest was in the rice system. And higher biomass, microbial biomass carbon was recorded in all the multi-tier production system. Now, soil organic carbon pools and their aggregate associated carbon under different orchards. This was evaluated by Deepasri A under chairmanship of Munoj Chaudhary. So here he has observed that the labile pool of carbon, that is the KNO4 carbon, was recorded in soils under guava and bale orchard. So highest was recorded under guava and bale orchard, whereas lowest carbon, this KNO4 carbon, that is the labile pool, was observed in the fallow land. Now total, she again, Deepasri, she estimated the macro aggregate and micro aggregate associated carbon, and it was found that the total organic carbon in macro aggregate was higher under the different orchard system, whereas micro aggregate associated carbon was highest under bale orchard. So this is the findings from the study. Avinash Das, he worked under Dr. T.J. Purokaysa. So here actually we are seeing the carbon distribution in different land use system, but here what he has done, he has seen that how clay minerals and sesco oxides, they help in stabilization of carbon in soil. So for that he has taken soils from 4 soil orders. One is your alphisol, inceptisol, mollisol and vertisol. Here what he has done, he has given different clay doses, and he has in soil he has removed the sesco oxide and he has kept the sesco oxide in the soil. So what he has found that the vertisol soil clay fraction showed the lowest amount of carbon mineralization, whereas inceptisol, alphisol showed the highest. So in vertisol, what is the advantage is because of its smectite mineralogy. Here the carbon sequestration is maximum. Even in mollisol, there is we have elite type of mineralogy. So here also the carbon sequestration is higher, and the increased amount of soil carbon fraction as well as presence of cesium oxide significantly decrease the carbon mineralization. So carbon mineralization rate is decreased in presence of your clay, nature of clay, as well as presence of cesium oxide. This Moedakshi Chandra, she worked under Ruma Das, and she found that when actually a high amount of sludge is applied, that is 300% nitrogen replaced through sludge, it is observed that the mineralization is actually decreased. So it indicates that the adverse effect of higher rate of the sludge application, that is when 300%, so different doses of your nitrogen was applied through sludge. So when we are applying 300% nitrogen through sludge, then we are finding that the mineralization rate is decreasing. So it is adverse effect on soil respiration. This for conserve, again this student Vikramjit Mondal, he has worked under chairmanship of Dr. Sunanda Vishwas, and he has found that in different conservation agricultural system, he has estimated the active and passive pool. So he has found that the active and passive pools, the active and passive pools of carbon in zero tillage plots showed greater values with respect to conventional tillage in all three depths. So active and passive pools actually are increasing in all depths, and the passive pool of carbon dominates over the active pool at a depth of 15 to 30 centimeter. So at the depth of 50 to 30 centimeter, your passive pool is increasing. That means recalcitrance of carbon is increasing in case of your passive pool. This Mr. Reddy, under chairmanship of Dr. Sheila Das, he has, now we are seeing that this what is the happening in the conservation agriculture, but here actually he has worked on the different aggregate associated carbon. So aggregate associated carbon, when he is trying to when he fractionated different aggregate associated carbon, like the macro aggregates and the micro aggregate associated carbon, and the different this aggregate fractions show, these show that the protected carbon was mainly found in this macro aggregate and micro aggregate associated carbon, followed by silt plus clay micro aggregate associated carbon in the zero tillage. And treatment, this when there is a triple zero till with triple residue retention, this is the most effective treatment in carbon sequestration point of view. So this T5 is showing the highest macro aggregate and micro aggregate associated carbon in soil. Soikot Bera, he worked under Dr. Vibhas Chandra Verma, and it was found, he found that when he used different organic amendments on carbon and nitrogen mineralization in acid soil of Jharkhand, it was observed that when this Dhaincha was used, and then the different temperature effect was there, and with and without nitrogen, it was found that the carbon mineralization from organic amendments mainly depends on the carbon content, that is the CN ratio of the amendments. It was positively affected by temperature and nitrogen emission. So here we are finding that actually temperature and nitrogen actually will control your carbon mineralization rate. And among the various organic amendments, Dhaincha amended soils showed relatively higher nitrogen mineral content. Here you can find that these Dhaincha amended soils are showing higher mineral nitrogen content. So this actually is better for your mineral nitrogen potential. Now for assessment of soil quality, he worked with Dr. Manoj Chaudhary. So here what he has found that in different cropping systems, the soil quality in pulse-based cropping system proved to be better than other cropping systems. And pulse-based cropping system showed better soil quality than the maize potato system, which is followed most commonly in this region. So pulse-based cropping system was found to be better than the maize wheat cropping system. For assessment of resistance and resilience of soils under rice-based cropping system, it was found that when we are applying heat stress, then we are finding that under CA, highest resilience was found under 000 tillage management, and under conventional tillage practices, higher resistance after heat stress was obtained in case of where there is 50% nitrogen was replaced with FOM combination. So when there is a 50% replacement of FIM, nitrogen through FOM, then we are getting your better heat stress sensitivity. Then this next was on novel fertilizer products. Ms. Jose, she carried the work on this nanoclay biopolymer composite, and it was found that when the this nanoclay biopolymer composites, there 25% of nitrogen can be substituted by the nanoclay biopolymer composites. So agronomic use efficiency and apparent nitrogen recovery were higher in case of nitrogen supplied through nanoclay biopolymer composite. Then for dynamics of phosphorus availability, silicon was applied for with different sources, and it was found that among the silica sources, corn cob ash maintains significantly higher solution P, and PSM application increased the solution P level, and application of inorganic silicon at the rate of 125 milligram per kg was sufficient to maintain the P availability in soil. So this is one important finding, that a concomitant application of silica along with corn cob ash can help in maintaining the phosphorus availability. Another work by Punita Kumari Meena. She worked under Dr. M.C. Meena.
Please,
and then
your time is up. Just finish.
डॉक्टर नयन, आप टाइटल दे दीजिए और जस्ट उसका नीचे जो कंक्लूजन है वो बता दीजिए, या वो भी इस टाइम पे जस्ट
Sure. So here it was found that in the IPNS system, integrated plant nutrition system, the 100% FOM treatments had significant positive effect on the level fractions of P. So IPNS, that means integrated plant nutrition system, helps in improving the P availability in the soil. Similarly, when we are in conservation agriculture, it was found that when 4 ton per hectare with 50% RDP and PSB plus AM can curtail fertilizer application rate by 50% by increasing the grain yield about 30%. So this 4 ton per hectare residue application along with 50% per RDP plus AM plus PSB is suggested for maintaining the phosphorus availability in soil. Now for micronutrient dynamics, the student Bhagavan Pawar has worked under Dr. R.N. Pandey, and he has identified different sensitive on basis of gene deficiency tolerant index, the different genotypes which are highly tolerant, medium and low sensitive, that means sensitive, and he has found that these different genotypes were screened, and it was found that for your sensitive and tolerant varieties, the IR64 was sensitive and Joya was tolerant. These two varieties were treated in the pot, with different organic acids, and it was found that 2.5 milligram of zinc along with 10 milligram of citric acid was found to be superior over other treatments. Similarly, soil boron dynamics was studied in an insectisol, and it was found that the organic treatment increased the specifically absorbed boron and organic boron and decreased the oxide boron in the soil. So, and this finally, the soil test based fertilizer recommendation was carried out by Mr. Meena under Dr. Pandey, and it was found that these fertilizer prescription equations were developed for Leadstone wheat and maize crops, and it was found that this balanced site-specific nutrient-based management in Leadstone wheat and maize was prescribed. So these are, and then Mr. Monindra Burman worked under Dr. Indu Chopra, and it was found that when actually maize crop is used for treating this contaminated water among the agricultural waste, it was found that the maize crop showed maximum dye removal absorption compared to other absorbents, and biobate treated water was used for irrigating mung crop in pots showed with.
Excuse me, Dr. Nayan.
Yes.
Now simply just read out the titles. Okay,
Yeah.
that's it. Now just,
This is
no,
the
absolutely,
total publications.
absolutely no need to read out, the names and all.
Yeah.
Perfectly fine.
So total, we
Thank
have 30 students publication, and the research paper published from different journals like reputed journals like
okay,
Geothermal,
Dr.
Soil Tillage Research. These are the students explained.
Thank you,
All
Dr.
right, so
Let's
thank
give
you.
some time to the chairman for his remarks because
Thank you.
These are
we
the
are running
training
short of
courses, and
time.
this is,
Thank
I
you.
acknowledge.
Thank you. I'm sorry that we are running short of.
Very
already
sorry,
over
but
time.
time constraint. It's
Time was short.
Okay, so next I
minutes will take.
invite Dr. Sushma. There is Sushma. Sushma, you are a smart scientist. Be smart with the time. Climate smart, time smart.
Now, Sushma, just, be very, very specific. Very, very specific.
Anyway, she has only few.
Good afternoon, all of you. With the permission of the chair, I am going to present significant educational achievement of water science and technology on behalf of Professor Mansingh. As you know, the mandate of Water Solution to conduct basic and applied research on water management in interdisciplinary mode, along with developing human resources and organized training program and disseminate knowledge. And our in water science and technology, total 11 faculty with 33 students. And during this year, 4 MSc student and 1 PhD student are going to take the degree. And under the theme, 3 themes: water conservation and crop productivity to manage the green water, marginal quality water use in agriculture, that is 2 MSc students to manage the grey water, and efficient irrigation technologies for enhancing water and crop productivity, 1 MSc and 1 PhD students to manage the blue water. Coming to first MSc student, that is Abdi Nasir, under the chairmanship of Mrs. Rosin KG. Effect of water conservation, nutrient management. They have studied different water conservation practices and nutrient management with 2 objectives. These are the trenches and microcatchments are the water conservation practices and 4 nutrient management practices. And they have found a higher moisture content and highest available NPK and biomass weight in trench along with 50% RDF, biofertilizer and FYM. Another student, Bhavna Verma, under the chairmanship of Dr. D.S. Gurjar, to find the heavy metal, that is the heavy metal remediation in spiked wastewater by the Calendula with 3 objectives. And they found the translocation, they have taken the cadmium, chromium, nickel and lead. These are the heavy metal spike added to wastewater. And they found that the translocation factor is less than 1 and bioaccumulation factor is more than 1. And but they found the more in the roots compared to soot, that is the accumulation of heavy metals. And ultimately they found the calendula showed a good phytoremediation potential for heavy metals. Another student, that is Sumit Jangra, under the chairmanship of Mrs. Rosin KG, and he has done his work to arrest nickel contamination in soil using the agricultural waste materials under wastewater irrigation with two objectives. And they have taken nickel concentration two types, one ppm and 2 ppm concentrations, and sawdust, rice straw, and rice husk with the rate of application 0.5 and 0.25% in lysimeter study. And they found the nickel arresting potential of rice husk is better compared to other waste. So rice husk would be the promising potential soil amendment for nickel stabilization. The student Arul Selvam under the Professor Mansingh, he has studied the digital tension-based irrigation scheduling with the 3 objectives, and they found they have taken different studies on soil moisture distribution with respect to near to emitter and at the different distance from the emitter with different irrigation regimes using a tensiometer and 3 fertilizer doses. And they found the soil metric potential of -30 kilopascal with 120% RDF in garden pig gave the highest yield and highest water use efficiency. Another student, Mona Lisa Pramani, PhD student under the chairmanship of Dr. Manoj Khanna, she has already presented yesterday for gold medal, and she has worked to under 3 objectives. Generally, we are doing the automation in pressurized irrigation, but she has done the automation in basin irrigation system. That is a new and novel thing she has done. Under this, there are 3 components: sensing unit, sensing unit, communication and controlling unit. She has explained very well yesterday, so I am not going in detail. Due to this system design and validation in field, they have found the irrigation application efficiency increased by 27% from 69 to 88, and water saving of 24.3%, and money saving. This is rupees 2,150 for diesel only, not overall cost. And they have found where should be the placement of soil moisture sensor for opening and closing of check gate. And they have studied. So due to this, the overall conclusion is the developed LoRa-based and GSM-linked solar power sensor-based automatic surface irrigation system was found to be the robust, easy, and effective tool to the farmers for enhancing surface irrigation efficiency. Total number of publication 22. These are the student publication and student placement 2, and all other students are doing their MSc and PhD. 2 students, they have got the first in group dance performance and first in spelling combat or IIRA. These are the participation by the students in different forum. And Professor Mansingh is the member of different advisory and technical committee. And Dr. Ravinder Kaur became the associate editor of Soil Health Journal. Dr. Manoj Khanna, fellow of ISA. And myself became the fellow of 2 society related to soil and water conservation. Dr. Nita Dwivedi and Mrs. Rosin KG got the Achievers Awards from CAPA. Dr. Imanuvis got the second oral presentation award, and Dr. V.K. Pajapati became the member of BOS of Raffles University of Rajasthan. Since 3 years, our WST student and faculty, they are showcasing the water management technologies in watershed during Pusa Krishi Mela. Also this year, last year, they have shown it. This is the complete water management package we are showing there on watershed basis with a different topography and different water management practices from higher slope to lower slope, including water logging and drainage, everything. This is the training activities that we are doing in our farm. This is the only sample, but we are doing so many trainings and exposure visits in water technology center when and wherever they are coming. And at the last, our Jalopchar technology developed by Dr. Ravinder Kaur Madam, that has been recognized by ICR. And due to this outscaling of this Jalopchar technology, that has during this year, it has been installed at 4 places. But in coming year, it is 3 upcoming projects under SHAP. It is going to be installed at Guwahati, Umiam, and Dehradun. Due to this, total revenue generation was 44.75 lakhs. With this, I acknowledge to the faculty of WST students, their chairperson and our advisory committee members, Dean, Associate Dean, JDR, colleagues of EGS, Director and IRI for their cooperation and support. And my special thanks to Professor WST and Project Director WTC for providing me this opportunity to present. Thank you.
Thank you, Sushma, for being time smart. Thank you very much. So you can have your seat. Okay, so I think with this we come to the close of all the presentations by all the professors. I know it is really to put everything in a very concise manner, particularly when you have got, I think, one of the biggest number of the presentations here, 122, and across 7 or 8 disciplines within the NRM. And NRM, as we all know, is a very complex, as you must have seen from the glimpses of all this presentation, that cuts across so many disciplines. It's very, very complex and a comprehensive area of research. And the presentations were definitely of a good quality, I must say that, and though they were kind of in a hurried manner, and I could see that there was a essence which was out of all this presentation, that a good amount of research has gone into the effort which has been taken up. So I definitely congratulate all the professors and all the students who have been working behind all this research, and which is pretty much evident from the amount of the good publication in the impact journals which you have with high impact factors, and also from the fact that so many of them have got the awards, they have got a good placement. So that itself speaks volume about the kind of the achievements which are there, and that's kind of emanating out of what they have been doing. So I think, so this is something that was very, very and informative to I initially, when it started, I'll be doing some kind of a summary, but I have now in no mood to do any summary because already we are running out of time. What I would like to leave you all with a message that in this kind of presentations, I mean, that's my suggestion, don't take me otherwise, because I'm entitled and free to make my own suggestion, is that in this kind of a setting, when you are making these presentations for the students, it's better we don't go into each and every student's thesis by thesis or dissertation by dissertation, because the paucity of time has to be seen. So from that point of view, I would suggest that you make various thematic titles with each and every discipline, and in each thematic title, you give the key highlights which are emanating out of the research work done by the students. You can attach the name of the students by the side of that so that they get acknowledged that who has really done that, rather than going to the methodology and the process, because this is not a regular routine presentation. This is only the presentation of the key highlights and the key achievement. So that's one of my suggestions. And the other thing which I know that there are a number of areas which are cross-cutting, like precision agriculture, precision farming, precision irrigation, and a lot of remote sensing and mod. So some of those things I see that the same thing is being done by various different divisions. So which perhaps could be integrated, and we could have what we call it as a cross-cutting mode of research. So that kind of thing also perhaps would be good idea, so that way we can avoid those kind of things. So one interesting thing which I saw, as I said, I will not go into any specificities, that I was happy to note that the environmental sciences are talking about the PES, Payment for Ecosystem Services. I know that's very important, but it is hardly done, and it's very difficult and very complex, because to monetize it is a humongous task. But I'm happy that you made an attempt, but I think you have a long way to go into that. And when you are looking into those kind of things, you have to see sometimes when we call the word payment for ecosystem services, people, they start running away. So sometimes we also call it as a sharing of benefits. So that's another way of trying to look into, but the point is that that's an area where I think we should be getting tightening our belts, that to try and see that how best we can really look into that, because when particularly the IRI and NRM school is working pretty much on the climate change and carbon sequestration, conservation, agriculture. So it's a high time that we monetize those benefits and try and put it in a perspective of payment for ecosystem services. I think with these words, I thank one and all, and I congratulate all of the professors and their students for doing a really good job. Thank you very much.
Thank you, sir. Now it's time to felicitate chairperson. I request Dean ma'am to do the honor.
Yes, I'm sorry, I'm also in a hurry because I didn't know that it will go last, so I had given time at 4:30.
Thank you, sir. So it's my moral duty to propose a formal vote of thanks. I'll cut it very short.
Oh, say one and all.
Yeah, so that is what.
Thanks, one
so
and all.
thank you
That's
for-
it. Yeah.
Thank you so much for accepting our kind invitation and being here for today.
Yes.
Thank you, everyone, and thank you to all the professors who presented and adjusted with us for the time schedule. Thank you. Okay, now we can break for tea and we'll assemble at 5 p.m. please. हाँ, मैडम आ गया, ले लो
ऐसे ही
ये ले लो, ये भी ले जाओ, इनसे ये ले जाओ। कहाँ है? धर्मपाल किसी को भेजो, ये भी डाल दो ना। ये तुम करो, मैं मैडम के लिए लगा दूँ।
ये तो पता नहीं कहाँ कर रहे हैं, डेस्कटॉप पर देखिए। नहीं, वो ये प्रोफेसर प्रेजेंटेशन ना, ये वहाँ कोई बेसिक साइंस बोलते होगा। नहीं, ये नंबर है ना। अच्छा, नंबर है। हाँ, अरे साहब आपने तो कर ही दिया। हाँ, सब तो भी रहे थे। क्या फर्स्ट को सर ऊपर आए थे। हाँ, मैं देखा कैसे किया। अब यहाँ आए थे। हाँ, अच्छा। देख तो लूं कितना आ रहा है कि नहीं। तो ये है तुम्हारा। अच्छा, ये फंक्शन ये तो ये दिखाई नहीं रहा है ना। हाँ, वो फंक्शन करना पड़ेगा। मेरी साइड से बहुत है सर।
ये है।
हेलो हेलो यहाँ ठीक है हेलो
हाँ
हेलो हेलो
हेलो हाँ
यहाँ हेलो 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 यहाँ ठीक है हेलो हाँ यहाँ थोड़ा होना चाहिए
Very good evening to all after the tea break session. Dear faculty and students, the chairperson for the 4th session on School of Basic Sciences, Professor Akhilesh Kumar Tyagi, is amongst us now. So it's an honor for us, sir, to have you here. May I kindly request Dr. A.K. Singh, sir, to kindly welcome the chairperson with a bouquet of flowers. Thank you, sir. May I now request Dean Madam for her welcome address to the gathering.
Thank you, Dr. Monica. I welcome you all in this 4th session of the professor's presentation of the first day of the 2-day this event. And prior to this session, sir, we had the presentations of the schools of crop improvement, crop protection, and natural resource management. Now, dear audience, you will be glad to see that to chair this session, we have with us Professor Akhilesh Kumar Tyagi ji, the senior professor. I don't really like to say anyone retired. Yeah, officially superannuated, that's all, but still very active. In the Department of Plant Molecular Biology, University of Delhi. And our Director, Dr. A.K. Singh ji, is also with us. I warmly welcome you, sir. And sir, in this session, the professors of the disciplines and discipline under School of Basic Sciences, Biochemistry and Molecular Biology and Biotechnology and Plant Physiology, they will be making their presentations on the significant educational achievements based on a total of 43 theses, comprising of 26 MSc and 17 PhDs. So, sir, although it doesn't, of course, sir, you are aware that it doesn't represent the entire spectrum, but more so it covers the canvas. So with this, sir, thank you very much. And now I request Director, sir, to kindly introduce this, the chair to the audience. Thank you.
Respected Professor Tyagi, Professor Mehta, and distinguished colleague, Dr. Anupma. It's my proud privilege to welcome you all once again for this session on basic sciences, where we are going to have the presentation by respective discipline professor on the significant research finding of the students who couldn't have an opportunity to present yesterday, but their work will be presented today by their professors. So this is a great occasion. We are particularly pleased to have Professor Mehta always, and also Professor Deshmukh, both are here, and they always have their blessings for us and for IRI. Professor Akhilesh Tyagi is very well known to each one of us, and particularly I have had the fortune of working in close association with him for very long. We have many papers together, and one incident that I never forget, always I remember, and something to take lesson from, and also to emulate in our practice, is that in 2004, I think it was 2004, he was director of Center for Plant Molecular Biology and heading that division. And Dr. VP Singh, he had contacted and wanted to have some discussion as to what kind of collaboration South Campus can have with IRI in basic applied research in genomics, particularly on grain development in rice. So I went with Dr. VP Singh to his lab, and we discussed for 2 hours. And in that 2 hours, he took out pen and pencil, jotted down that what is the collaboration going to be, how many research papers are expected from this, what is the tentative titles of those papers, who will be senior author, who will be next author in which paper, and what will be the contribution of whose. 2 hours we spent, and I tell you, exactly 10 years later, we came out with the same number of papers, with no controversy, with no, everybody's contribution duly acknowledged. That kind of meticulous planning, I think, is a lesson to learn for us. And if that happens, then many times the issues comes for, taking credit or sharing credit. And often we find that it has become a culture that we have large number of names on a paper, but if you ask to specify who has contributed what, it is sometimes very difficult to specify. And that culture, I think we have to get away with, where we identify the people and identify them with their work. It is very important. Even our students' paper, if you see, the number of authors has gone very large. Fine, if it is a collaborative project, if everybody has contributed, it's perfectly fine. I mean, it's a culture today when we want to do multidisciplinary research, then number of authors is not a limitation, but it has to be well defined. And that was one thing that I learned from Professor Tyagi in that interaction. It was really very productive. He did his PhD from Delhi University with Professor S.C. Maheshwari, and all of us know Guha and Maheshwari for applied work. And it was Professor Tyagi with whom Dr. Maheshwari started, in fact, exploring the field of molecular biology. And today, molecular biology stands at a new pedestal. He led the rice genome sequencing project along with Dr. N.K. Singh from IRI, chromosome 11 long arm. This was done by these two groups. Also, tomato genome sequencing and chickpea genome sequencing. More than that, if you look at the capacity building and human resource development in field of molecular biology and biotechnology. Today, best of the best manpower, you will find they have training, whether it is Amitabh Mohanty in Corteva doing genome editing, upstream work. He is trained in Professor Tyagi's lab, and likewise many more. And he was professor at Delhi University, and currently he is distinguished Meghnad Saha professor at Delhi University, which is from Nasi. He continues very actively. He also served as director of National Institute of Plant Genome Research. There again, we had very good interaction. In terms of award recognitions, of course, J.C. Bose National Fellowship, National Bioscience Award, Nasi Reliance Industry Platinum Award, GM Modi Award, Racine Award, BP Pal Memorial Award, and many more. He is fellow of all the scientific societies of India, and also the president of the National Academy of Sciences India. He has been in past, and also the World Academy of Sciences. He is fellow. So it's our pleasure, sir, to have you here, and we look forward for your very valuable guidance and suggestion to our program. Thank you very much for sharing your time. Thank you.
Thank you so much, sir. So with the permission of the chair, I would wish to apprise the House regarding the modalities for the presentation. So as since morning we have been doing, for the session 4 under School of Basic Sciences, we will have 3 presentations from the professors of the following disciplines in the following order. First will be biochemistry, then to be followed by molecular biology and biotechnology, and then plant physiology. So for all the 3 disciplines, the time allocation has already been conveyed to the professors, and it is based on the number of thesis abstracts from each discipline, plus an additional 3 minutes we have given. So for the ease of the presenter, a yellow light will blink 2 minutes prior to the allocated time, and a red light at the end of the time. So kindly bear with that. And I welcome you all for the presentation. And now I hand over the session to the respected chairperson to kindly conduct the further proceedings of the session. Thank you.
Good afternoon to everyone. It is always a pleasure to come here. Here, sitting with Dr. Singh, Dr. Anupma Singh, and Dr. Joshi, and in front of me, Professor Mehta and Dr. Chinnuswamy and other leading luminaries of the agriculture sciences. Thank you very much once again for giving me this opportunity. What I would like to say in the beginning in brief is that the presentation of the work of students is the most important thing, because it tells the truth about the activity of an organization. This was the reason at one time I started a student symposium in which a student used to speak only about their work, and all the boundaries of the laboratories will be dissolved in that particular symposium. This is also important because all teachers, barring a few, would like to give their best to the students. And therefore, these efforts ultimately fructify in the form of excellent research. And this is what we are looking forward for all the three divisions and NIPB. I will reserve my few appreciating comments. I have read all these abstracts which have been given to me to the end. And right away, if I am allowed, I will like to invite the first speaker on this, who is from Division of Biochemistry, Professor Anil Dahuja. So Professor Anil Dahuja, please. Podium is yours.
Thank you, sir. Esteemed chair and other distinguished dignitaries on the dais and off the dais. I'll be making the significant educational achievements in respect of discipline of biochemistry. So the mandate of the biochemistry discipline is to undertake the fundamental and applied research, and of course oversees a Center for Advanced Faculty Training. So we are involved in the conducting teaching and training programs regularly. And regarding the strength, we are total 12 faculty members, and the student strength, 10 MSc student and 34 PhD students, they are enrolled at present. And we are running courses both in the trimester and the semester system. Of course, this is overlapping, which is going on. And this year's 5 MSc and the 5 PhD students, they will be receiving their hard-earned degrees in the forthcoming convocation. So these are the two thematic areas in which the students, they have mostly worked. The first one is the characterization and the improvement of the nutritional and nutraceutical characteristics of the plant-based foods. And the second one, it is based on the metabolic pathway genes and the enzymes which are actually contributing towards the higher yield and the better nutrition in the food crops. So these are the number of the students who will be receiving their degrees in each theme area. And with regard to the PhD students, as I said, the 5 students, they will be taking their degree, and these are the names, titles, and the chairpersons who are involved in the guiding of these students. And this is the list of the 5 MSc students along with the thesis titles. So coming to the first thesis, that is which was done by the Durga Lakshmi, who worked under the guidance of Dr. Vinuta T. And the topic, it was enhancement of the protein quality and the functionality. So as you all know that the protein malnutrition, it is a biggest menace which is coming up in a big way, and we need to address this challenge. So Dr. Vinuta and Mr. Durga Lakshmi worked on this particular aspect. So they tried to develop the protein blends using different plant proteins, and they mixed these chickpea, rice, and the peanut flour in a ratio of 30:40, and then they determined the protein quality and what is the extraction of the protein, and what is the level of the different essential amino acids which is present, and of course along with the digestibility, because it is not only the content but the digestibility, it is also very, very important if we really want to derive the health benefit out of it. And since it is a misconception, rather there are apprehensions that the digestibility of the plant proteins, it is lesser than the animal proteins. So they wanted to enhance the digestibility of the protein through the steam infusion process. And after the steam infusion, they have again checked the nutritional quality of this protein and how digestibility it is getting affected, along with the functionality as well. And this is the formulation which was developed, and they have identified that the steam infused protein blends, they have better digestibility, protein content, and solubility. Also, it was enhanced in case of the infused protein blends. And this was the second thesis, which was done by Tejveer Singh, who worked under the guidance of Suneha Goswami, and they worked on the pearl millet crop. And as you know that the shelf life of the pearl millet flour is a problem, and they wanted to identify what is the mechanism underlying regarding the low shelf life of the pearl millet flour. And they targeted the phenolics, how the phenolics they could be contributing, and of course along with that they worked on the phytic acid and the iron and the zinc, and how phytic acid is influencing the bioavailability of the iron and the zinc. And they identified 43 different phenolic compounds in 30 diverse pearl millet genotypes, and they quantified 6 phenolic acids using the HPLC. And of course the bound and the free phenolics, they are also calculated. And regarding the digestibility and the bioaccessibility parts, they use the InfoGest model, and they try to simulate the conditions which are actually present in our gut. And they found that the phenolic composition, if it is present in the perfect ratio, the bioaccessibility of the phenolic compounds can be enhanced. And then the negative correlation they observed in the, as far as the phytic acid content and the bioaccessibility to the micronutrient which is concerned. And the third student, Harish Dhal, who worked under the guidance of Dr. Sneha Narwal, he worked on barley crop. And as you know, the barley is mainly being used in India for the malting purposes. But for the malting purposes, we are importing the genotypes from abroad. And because there are certain biochemical compositions which are not favored as far as the malting quality is concerned, I have listed 3 of them. The starch should be easily hydrolyzed into the fermentable sugars, and then there should be low beta glucan content and the high beta amylase activity. And of course, the starch with low pasting properties, they are preferred because high pasting temperature are destructive for starch hydrolyzing enzymes. So they screen the different Indian and the exotic genotype, and they want to identify what are the various differences as far as the biochemical composition is concerned. And this is the data which they have generated. And as you can see in the salient findings, the Indian genotypes, they have better grain quality, but exotic genotypes have better rheological and malting quality properties because they are having the low glucan content and they are having the high beta amylase activity. And then because of this particular aspect, so there is a great scope to improve the barley genotypes in terms of low beta glucan content and the high grain beta amylase activity, and of course with low pasting temperature. And the next student worked under the guidance of Dr. Shweta Kumari, and they worked on the carrot. As you know, the carrot is a good source of the beta carotene, but the stability of the beta carotene, it is an issue. As we go for the various processing methods, the stability gets compromised. So how we can develop certain methods, certain treatments which can retain the stability of the carotenoids. So this is the aspect they worked on, and these are the treatments which they have tried. And after the data which was generated, and they could find that if you use the citrate buffer with probiotic fermented carrot slices, so it retains the stability of the carotenoids. So this is a major output which has come from this particular thesis. And they also did the functional analysis and the bioaccessibility as well, because as we are going for the pre-treatments or the processing treatments, the bioaccessibility should not be compromised. And they duly checked this particular part, and the redness chromaticity, it has been mentioned here, so which is an indicator the quality of the color or the pigment it is retained. And this was all about the MSc student. Coming to the PhD student, the first student, Anshi Falli, who also worked under the guidance of Dr. Sneha Goswami. And again, because they have specialization in the pearl millet, as far as the shelf life quality is concerned. So they, this PhD student, Anshi Falli, also worked on the shelf life of the pearl millet flow. And again, they wanted to decipher the mechanism which is involved in the development of the residity and the during the storage process, and they could identify the enzymatic as well as the non-enzymatic mechanism which is responsible, and they could identify that the linoleic acid content, it is directly related to the lipoxygenase activity, which is the major culprit for the development of the residity. And the total phenolic acid content, it was found to be directly correlated with the enzymes which are responsible for the browning of the flow. And then they developed a physical processing method, how we can actually enhance the shelf life of the pearl millet flow. And they tried 3 different methods: infrared, microwave, and the hydrothermal treatment. And as you can see that hydrothermal treatment, it is giving the positive result because it is reducing the activity of the peroxidase, polyphenol oxidase. And this is in comparison to the other 2 treatments like the infrared and the microwave treatment. But of course, the extensibility of the hydrothermal treated pearl millet flow was compromised a bit, and we need to modify this method further so that this negative impact does not come. And this is the another part which they worked on, characterizing the effect during the storage also, and they also checked the bioaccessibility because, as I said, if you are going for the pre-treatment method or the processing method, you need to know whether the bioaccessibility has been compromised or not, and they duly took care of that particular aspect. And again, in conclusion, you can see that the HT treatment was found to be more effective. It effectively reduced the starch digestibility, reduced the bioaccessibility of the phenolics of the microelements. The next student who worked under my guidance, Sandeep Kumar, we are working on the plant-based milks, especially the soya milk, and these are the objectives of his study. And basically the health attributes to the soybean, they have been attributed rather to the isoflavones, but these isoflavones, they are present in the conjugated forms, and because of their this particular aspect, their bioavailability gets compromised. So if somehow we can deconjugate these isoflavones, so their bioavailability can be enhanced. So that's the aspect which we worked on, and we have found that the beta glucosidase is the one enzyme which is responsible for this deconjugation process. And we cloned the gene encoding for this beta glucosidase enzymes, not only from the soybean seedlings, but also from the lactobacillus bacteria. Because ultimately we want to develop the fermented soya milk, the probiotic bacteria which are going to be used for the fermentation process. So we wanted to know whether they have the beta glucosidase activity or not. And we also isolated the genes from them as well. And these genes, they were also heterologous expressed, and we could get the purified beta glucosidase. And the purpose was to use this beta glucosidase during the fermentation process so that the deconjugation can be easily carried out. And we also went for the immobilization of the beta glucosidase, but unfortunately, after the immobilization process, the activity was lost. And this is the aspect which we are working now. And the key, I think, lies in the kind of substrate which is being used. And we have found that the phosphorylated substrate, it is taken very easily rather than the free sugars. So another student, Muzaffar Hassan, also worked on the similar aspect. And as I said that these days, the health benefits, they have been attributed to the various fermented products. And in the fermented products, there are claims that these many CFUs of the probiotic bacteria, they are present, but actually in these products they are not. So we wanted to make use of the prebiotics, which will ensure that there are sufficient number of the probiotic bacteria in your product. And of course, the various oligosaccharides, they have been tried earlier, but we put our focus on the anthocyanins, because whether they can be utilized as a source and the carbon for the by the bacteria, because it will give the dual benefits, not only the antioxidant property coming from the anthocyanins, but also the if they promote the growth of the or proliferation of the probiotic bacteria, so it will also help in the maintaining the CFU count. So this data clearly shows that anthocyanins, they can be metabolized by the probiotic bacteria, and as can be indicated by the presence of the methyl propionate and the methyl butyrate, which are the short chain fatty acids which are formed after the fermentation process. So the presence of these fatty acids, it confirmed the prebiotic potential of the anthocyanins. And we also did the nutrition and the functional characterization of the anthocyanin fortified fermented soy milk. As you can see, that antioxidant potential, it was enhanced tremendously. And of course, along with that, there was a reduction of the anti-nutritional factors as well, because as the counts of the probiotic bacteria, it increases, so there will be increase in the activity of the various hydrolytic enzymes, which can degrade this anti-nutritional factor. So this is the dual benefit which we are getting if we are making use of the anthocyanins as the prebiotics. And then effective storage, it was also studied, and as you can see, the viability count of the probiotics, it was enhanced tremendously after the use of the anthocyanins as the prebiotics. And this is the, in nutshell, the outcome of the study. The anthocyanin fortified fermented soy milk showed better growth and proliferation of the probiotics, improvement of the sensory as well as the nutritional quality. And of course, we identified Lactobacillus acidophilus and the Lactobacillus rhamnosus, which are having the better enzymatic properties, enzymatic wherewithal as compared to the other bacteria which was used, that is the Vizziella confusa. So next student worked on Jyoti Singh, who worked under the guidance of Dr. Ranjit Ranjan Kumar. So as you can see from the topic itself, that they developed and characterized the thermostable wheat rubisco activase through protein engineering. So as you know, the rubisco activase, it is the protector for the rubisco, which ultimately is determining the yield, and we need to protect this protector. So this is what they have targeted on. And this is the through transcriptome analysis, they could identify 12 mutated RCA transcripts, and they cloned this gene and did the heterologous expression of this cloned RCA gene, purified the recombinant RCA protein, and then they developed the 3 mutant proteins using the protein engineering by comparing the sequence of the different RCA available, and one mutant, RCA mutant, it showed really maximum thermal stability as compared to the other mutants. And then the functional validation of the thermostable RCA was also done in the Arabidopsis. They generated the transgenic plants, and they could functionally validate this particular mutant. Next student Sunil worked under the guidance of Dr. Archana Singh, and they worked on the what are the genes and the enzymes which are involved in the accumulation of the starch, because as you know, these days the trend is that whatever food crop it is containing the starch, people they are looking for having the enhanced amount of the resistant starch in the existing starch proportion. So they worked on these particular aspects. They did the expression analysis of the different genes, and they tried to correlate it with the metabolites analysis, and they could develop the network, how the metabolites they are actually related with the expression of the genes. And this is the conclusion coming from their study. And the next student, Rashid Sanyal, who got the gold medal yesterday, he worked under the guidance of Dr. Sajid Bishi. He worked on the raffinose family oligosaccharides, which again are very good prebiotics, and how we can, of course, they are considered as anti-nutrition factor also, and they are also involved in the flatulence. So that's why they targeted this particular what are the enzymes and genes which are involved in the synthesis of this raffinose family oligosaccharides, so that through use of the modern techniques, we can go for the down regulation of those genes, so that we can have the less accumulation of the raffinose family oligosaccharides in groundnut. So this is the result of their study. So they identified the raffinose synthase gene, rather the isoform of that particular gene, which is actually responsible. So this is the list of the genes which they have identified, which will be targeted in the future. And this is about the research papers which have been published. So total 42 papers they were published, and out of that, the 19 research papers they were having the NAS rating more than 10. So this is the placement record of the students who received the degree last year. And this is about the awards and honors by the faculty, awards and honors by the students. And these are the extension and training activities which our division was involved in. And my sincere acknowledgements to Director IRI, Dean and Joint Director, Associate Dean, Joint Director Research, Joint Director Extension, and thank you all for patient listening. Thank you very much.
Thank you very much. I think you are well within the time. We will move continuously.
Yes.
So we will move once again. Thank you very much. And we will move now to the National Institute of Plant Biotechnology, from where Professor Debashish Patnaik would make the presentation. Dr. Patnaik.
Reverend Chairman of this session, respected teachers and the august audience, very good evening. Let me present significant research achievements of the Division of Molecular Biology and Biotechnology. The mandate of the division is to conduct basic and strategic research on plant molecular biology and biotechnology and disseminate knowledge to students and trainees as part of human resource development. The division offers 27 courses in predominantly semester mode by 34 faculty members. At present, we have 77 students, 20 MSc and 57 PhD. And this year, 20 students are receiving degree, 14 MSc and 6 PhD. The 14 MSc students, they conducted their research either at NIPB, NIPSM or IIAB Ranchi. For the sake of presentation, the research conducted by the students are arranged in three theme areas: abiotic stress management, biotic stress management, and basic study. Research work of 1 MSc students and 2 PhD students are aimed at to improve abiotic stress tolerance in crops. As heat stress severely affects wheat production, and brassinosteroids has its role in imparting heat stress tolerance, MSc student Mr. Adil analyzed the brassinosteroid mediated modulation of gene expression in 2 contrasting wheat cultivars during heat stress under the guidance of Dr. Sarmishta Borthakur. His study revealed that when seeds are primed with 24 epibrassinolide, it has profound effect on root architecture. On an average, root volume, root length, and all morphological parameters of root increased when plants are treated with brassinosteroid than that of control. Transcript profiling revealed that 2 genes has higher expression. One gene, this is BRI1 encoding a brachiosaurid receptor, and another gene BZR1 involved in as a positive regulator of brachiosauroids. They are showing higher expression in brachiosaurid treated plants. The full-length cDNA of BZR1 was cloned and analyzed. This gene is present in wheat chromosome 3A, and his work paved the way for functional characterization of this gene BZR1 to understand more on brachiosaurid mediated regulation of gene expression in wheat during heat stress. PhD student Ms. Shruti did molecular characterization of different pigeon pea genotypes for chilling response. It was known that pigeon pea, specifically the long duration crop of pigeon pea, faces chilling stress, specifically central and North Indian states. So she screened a number of pigeon pea genotypes and identified two genotypes, and one is UP, that is tolerant, and one is susceptible, called CP, based on different morphological and biochemical screening test. And her transcript profiling study revealed that the sensitive genotype CB has very high number of reprogramming or transcript during chilling stress. Through her study, she identified 4 cold specific expression of genes, that is cold subdomain protein genes, during this chilling treatment, and these genes were involved in probably reactive oxygen species mediated metabolism of carbohydrate and during homeostasis establishment. The overseas student from Ghana, Mr. Maoli, did characterization of heat stress responsive genes in a contrasting wheat cultivar under the guidance of Dr. Jasti Padaria. Now he analyzed the transcript of resistant wheat genotype 3764 and found that 7,990 genes show differential expression. So from this differential expression, he identified a total 6 genes and validated those 6 genes in a panel of 16 different wheat genotypes. And from these 9 differentially expressed genes, he pointed out one particular gene called short stress root protein, or RS1, and characterized its functional validation under the transgenic tobacco background. The transgenicity of the tobacco transformants were confirmed based on PCR screening and Southern analysis. And based on that screening, he selected a number of tobacco promising transgenic lines and exposed them under heat stress. And it was observed that total chlorophyll content, membrane stability index, and relative water content was much higher in those tobacco transgenic lines expressing this gene RS1. So it was concluded that this gene, probably RS1, imparts tolerance, heat tolerance to these transgenic lines and could be employed for heat stress tolerance in wheat. Research work of 3 MSc students and 4 PhD students were aimed at improving biotic stress tolerance in crops. Ms. Rishika, she screened different BT isolates to find a number of insecticidal protein genes of BT, and also evaluated toxicity of different vegetative insecticidal proteins under the guidance of Dr. Sarabjit Kaur. She screened a number of 63 native BT isolates with different primer combinations and found presence of 1 or more CRY1 type genes in 16 BT isolates. She also evaluated the toxicity potential of different VIP3A proteins against Spodoptera luteum and determined the LC50 value. Her study showed the acute toxicity potential of VIP3 proteins against this insect pest, and these VIP3 proteins showed when treated with this larvae, they showed decrease in pupation and also adult formation in presence of these VIP3 proteins. Ms. Bhuvaneshwari worked on development of prime edit CRISPR-Cas gene construct for targeted editing of the bacterial bite susceptible genes XA5 encoding a transcriptional factor called TF2A gamma 5 protein by very targeted editing of single base substitution. She multiplexed 2 guide RNA sequences and then mobilized the guide RNA expression assembly, the nickage and reverse transcriptase in the final binary vector construct for development or transformation of rice. Then this binary vector construct was introduced into the rice protoplast for transient expression of the case per case for very predominantly editing of single base substitution. Mr. Onik, he isolated a number of bacterial endophytes and characterized these bacterial endophytes for its antimicrobial activity against Fusarium wilt chickpea under the guidance of Dr. Binoy Kumar. He isolated 109 bacterial isolates from different leaf tissues of chickpea, tested them based on the number of biochemical tests, and shortlisted a number of bacterial endophytes having antifungal activity, and 21 bacterial endophytes were analyzed based on sequencing of 16S rRNA genes. He found that 16 bacterial isolates have antifungal activity or antagonistic activity against Fusarium oxysporum. This bacterial endophyte treated plants showed better germination, growth, and stability, and also survival upon infection. And his study showed that based on transcript analysis, he found that 5 genes showed differential expression. These genes were involved in hormone pathway and signaling. Ms. Sarani worked on identification of genes, resistance genes that were present in the introgressed lines. These introgressed lines were developed earlier based on the interspecific hybridization of Brassica jancea and wild species Diplotaxis erucoides. She studied on characterization of these promising interspecific lines for presence of genes and pathways that governs that resistance against alternaria disease under Dr. R.C. Bhattacharya. Her study showed that the resistant introgressed lines showed resist or pathogenesis directly at the entry of the pathogen. Her GBS genotype by sequencing experiment showed that presence of a very significant QTL that harbors a number of potential resistance genes that modulate resistance against this disease. The overseas PhD student, Mr. Dona Gaya, Ms. Dona, she characterized a number of defensin genes from Brassica jancea and Camelina sativa, did their transcript analysis in presence of alternaria infection, and then did cloning and sequencing of a number of defensin genes, and it showed that the defensin genes has 234 nucleotides. And she also did the antifungal activity of defensin proteins. It was observed that the mycelial inhibition was dependent on the nature of the defensin proteins and also the concentration of the defensin proteins. As such, the defensins from Camelina showed better activity and better stability in inhibiting the disease. Mr. Suhas did cloning and characterization of a gene encoding a novel von Wilbur factor type A domain gene in imparting resistance under the guidance of Dr. Amol Kumar Solanki. What he did, he identified a 40 von Wilbur brand genes or VWA genes by analyzing the genome sequence of rice. The chromosome map was prepared and location of those genes was predominantly located on each chromosome. It was observed that these VWA genes from the resistant cultivar T-tape harbors more SNP than the VWA genes from the sensitive cultivar Nippon bear or HP2216. Based on the transcript analysis, it was observed that 3 VWA genes showed higher expression or differential expression. He has pinpointed one VWA gene called VWA36, put this VWA gene under different promoters and introduced into a sensitive rice cultivar for its characterization. The transgenic lines harbor 1 to multiple copies of the transgene, and the VWA gene showed high expression in the selected transgenic lines. Based on the GFP localization study in onion peel, it was observed that this gene was predominantly expressed in the cytosol. The gene, when expressed in the transgenic lines, confers a high level of resistance compared to that of control. And based on his study, he has already filed a complete patent, Indian patent. Mr. Manjesh Sakre, he has devised a novel strategy against the polyphagous insect pest Helicoverpa arpigera. In today's date, only the viable strategy is VT or chemical management. But VT has that stability concern, so he has combined RNAi VT combinatorial to give durability to the transgenic strategy under my guidance. So he has developed 3 different constructs, one harboring a chimeric BT gene called Cry1ACF, another vector construct harbors 2 multiplexed artificial microRNA in a single transcript under the NOS promoter and terminator, targeting 2 Helicoverpa genes, acetylcholine esterase and 20-hydroxyecdizone receptor. And these 2 strategies, VT and RNAi, was combined in another construct, VT RNAi combinatorial, and these 3 gene constructs were introduced into transgenic tobacco and then evaluated its potential by detached leaf insect biopsy. The detached leaf insect biopsy showed that the VT RNA combinatorial has highest resistance against the polyphagous insect pest Helicoverpa armigera, as evidenced by the minimum leaf damage. When the larvae reared on the leaves of BT RNAi combinatorial transgenic leaves showed 100% mortality within 4 days. Whereas when the larvae reared on only BT transgenic leaves, it showed 80 to 90% mortality in 4 days, and it took 8 to 9 days to obtain complete larval mortality. Whereas in case of the RNAi leaves, the larvae showed 40 to 50% mortality in 4 days, and it took 12 to 13 days to get 100% larval mortality. The transgenic lines selected promising transgenic lines carried single to multiple copies of the transgene, and the BT RNAi combinatorial transgenic leaves and also the multiplexed RNAi transgenic leaves showed maturation and expression of both the AMI RNA targeted against artificial choline esterase and also 20-hydroxyecdizone receptors. The research work of 14 MSc students were grouped under basic research. Mr. Sohanlal made an attempt to understand the low light perception by the cultivar rice Swarnabhava, and for that he constructed a subgenome library from Swarnabhava under the guidance of Dr. Prashant Das. He screened 2 contrasting rice cultivars Swarnabhava and IR64 under artificial low light condition and found that the growth of Swarnabhava is better than that of sensitive rice cultivar IR64. He isolated DNA from the Swarnabhava, restricted with the genomic restriction enzyme, and then a size fragment of 4 to 9 kb was eluted and constructed the subgenomic library, which could be a valuable research in understanding low light perception. Now, maize is a very good source of carotenoid, but unfortunately, the maize carotenoids get degraded very easily upon storage. And the enzyme responsible called CCD1, called cleavage dioxygenase 1, its expression was studied by Mr. Sagnik. And also he developed a CRISPR-Cas vector construct for targeted editing of the gene under the guidance of Dr. Jayanti. He studied different inbred lines of expression of this gene, and it was found that the CCD1 gene highest expression was observed only during reproductive stage and more specifically during the milking stage of the grain. He has constructed the CRISPR-Cas vector cassets by multiplexing two guide RNA sequence, and these two guide RNA sequences are multiplexed by tRNA processing system, and made an attempt for transient expression of this CRISPR-Cas expression cassettes. Mr. Gotham, his work was aimed to understand the low light perception of light by cloning and characterization of hinge region of the phytochrome A from the resistant cultivar Swarnaprabha and its allele mining under the guidance of Dr. Prashant Das. He has PCR amplified the hinge region, cloned and sequenced, and did allele mining, but the allele mining showed that there is no polymorphism in the hinge region of the phytochrome A gene, and it was concluded that this hinge region might not be responsible in low light perception in Swarnaprabha. Ms. Nujjot evaluated different maize genotypes, its regeneration and transformation efficiency under the guidance of Dr. Jayanti. She took different maize genotypes and evaluated its regeneration performance and found that the genotypic difference in regeneration efficiency, and she also optimized a protocol of using direct embryogenesis from different maize genotypes, and found that the maize genotype HKI161 showed highest regeneration efficiency. She also made an attempt for implantar transformation of different maize genotypes and evaluated their efficiency, and she found that there is genotypic difference in transformation efficiency of different maize genotypes, and the maize inbred line HKI161 showed highest regeneration efficiency based on implantar transformation with the GAS activity or positivity of 27%. Mr. Yogesh, he cloned the phytochrome A gene as analyzed by allele mining from the low light tolerant cultivar Swarnaprabha under the guidance of Dr. Prashant Das. Primer sets were designed for amplification of 4 amplicons of the phytochrome NG, and these were PCR amplified and cloned and then sequenced. The sequence analysis of phytochrome A gene showed that the genic region, including 3' UTR, did not have any polymorphism, but the 5' UTR has 5 inversions and 4 transversions and has lots of polymorphism. So it was concluded that the 5' UTR of the phytochrome A gene might be responsible for low light perception and signaling in Swarnaprabha. Mr. Ramesh characterized a QTL called GN4 in a near isogenic lines of Samba Masuri, and showed that it governs the high panicle grain number in the panicle. These near isogenic lines were screened and found that they have less number of pillar, but very high grain number in the panicle was responsible for 25 to 35% grain yield. The analysis of this QTL showed that out of 48 genes present in this QTL, 2 genes showed differential expression. One gene encoding end-chiron synthase that showed down regulation, and another gene encoding protein or protein kinase domain, it shows higher expression. So these 2 genes might be responsible for this high grain number for this QTL present in these near isogenic lines. Now, isoflavone synthase enzyme is involved in biotic stress response. Ms. Larissa, she characterized the promoter of this isoflavone synthase gene from green legumes under the guidance of Dr. Sivalingam. Based on different primer sets, she amplified promoters from different green legumes, chickpea, soybean, and also cowpea. Those promoters were cloned and sequenced, and those promoter strengths were analyzed based on the GAS expression. It was observed that the IFS promoter from chickpea drives GAS expression upon infection by cholera disease caused by Sclerotium rolsi and Fusarium oxysporium. And another promoter, IFS soybean, drives GAS expression under yellow mosaic virus of mungmin. Mr. Sampati Rao Dilip identified and characterized seed storage protein genes from winged bean, specifically the expression of allergenicity related proteins under the guidance of Binoy Kumar Singh. His transcript profiling showed different expression of 9 gene encoding allergenicity related proteins, and those allergenicity related proteins showed maximal expression during 10 days after anthesis in pods and seeds. Sriharsha, he worked on expression of ABA mediated signaling pathway genes during salt stress and shock under Dr. Binoy Kumar Singh. He grew plants for cold that salt shock and stress, and analyzed differential expression of genes and found that the MAPK signaling pathway genes were shown differential expression during salt stress and shock. Ms. Pragati developed calcium oxide nanoparticle synthesized and studied this nanoparticle mediated gene expression in chickpea under the guidance of Dr. Biplav Sarkar. The calcium oxide nanoparticle was synthesized and sprayed under different doses, and found that the spraying of calcium oxide nanoparticle give higher germination, growth, and early flowering. And a number of genes upon the nanoparticle application showed differential expression. The students published 13 papers of different NAS rating journal, and faculty published 101 research papers. And based on student research, 1 complete patent and 2 provisional patents were filed. All the students are very well placed. The PhD students are engaged in jobs, basically the overseas students. All the majority of MSc students are pursuing further study either in IRI or in IIT Kanpur or elsewhere. They have received a number of honors and awards, specifically the best poster awards in different seminars and symposiums. And apart from their teacher, they are also engaged in extracurricular activities with their artistic talents by organizing freshers welcome and also farewell party. They also showed their respect to teachers by organizing a Teachers' Day and by conducting a cultural program. And apart from research and study, they also conducted a cricket match, engaging both faculty, students, and staff. With humility, I thank all for their support and patient hearing. Thank you all.
Thank you very much, Professor Debashish Patnaik. Right. So we will, I think, move and keep the same pace. And I would like to invite Professor Renu Pandey, who will be presenting the work from Division of Plant Physiology. Professor Pandey.
Thank you. Respected dignitaries on the dais and off the dais, distinguished scientists and dear students. So today I'll be presenting the achievements of our division. So this is the mandate of the division: enhancing the physiological efficiencies for improving the productivity, and to conduct basic and strategic research, training the students and imparting training in physiological tools. This is the strength of faculty in our division. There is 16 from IRI plus 2 from Niazam Baramati. Out of that also, Dr. Gurumurthy is not a faculty now, but Dr. Jagdish Rane, who was a faculty, but he is selected as a director CAEH. So currently we do not have any faculty at Baramati. And total student strength is 51 on roll, including the newly admitted students, 21 MSc and 30 PhD. The awards and honors or deputation report from the for the faculty, it goes like this. Like, I'll not read all the names, but Pranjal Yadav, Dr. Lakshmi, Dr. Shivani, Dr. Vijay Paul, Dr. Rakesh Pandey, Vijay Paul, they have all got this honors and awards and deputation also from the division. The awards, recognition and employment of our students. So I would like to mention here that Ms. Neha Anand from our division, she has backed the SERB Overseas Doctoral Visiting Fellowship for Purdue University. And this is, I would like to mention here that this first time that any agricultural student has got this. Otherwise, this is always from IIT and ICER and ISC. Besides this, Sintu Antu, he has got IRA Merit Medal last year. And also he has been getting training under Naib caste. Milan Kumar, he got RDSana gold medal, and also he is already a scientist at CPRI, Divya Bharati, and besides this, they have our students have also placed very well in other universities, and out of 4 students, 4 positions in ARS, all 4 are backed by our students in ARS. This is the publication for the year 2022. So we have 30 publications, 22 from faculty and 8 from the students. And I would like to mention here that the NAS score, more than 8 to 10, we have 14, and more than 10 NAS score, we have 6 publications, and all are in this reputed journals. So this year, students, they're receiving degree are 12, 7 are from masters, and 5 are from PhD. Ali Ashur, who is from Egypt, he'll be receiving degree this year. So now I'll go on one by one, very briefly. So Ali Ashur, he worked on interactive effects of electric carbon dioxide, high temperature, and phosphorus on plant growth, grain yield and nutrition quality in wheat, under the guidance of Dr. Madan Pal. Here they took 2 contrasting wheat genotypes with optimum level of phosphorus and low phosphorus with 2 levels of carbon dioxide and temperature. And here they found that under high temperature and elevated carbon dioxide, optimum supply of phosphorus maintains a lower canopy temperature than the low phosphorus. And they also found a significant positive correlation between grain yield and canopy temperature depression. Further, he studied the effect of carbon dioxide and the phosphorus reduction of growth and phosphorus efficiency in wheat, and also on the grain nutrient content he studied in wheat. So they found that a significant positive correlation between mineral elements, protein, and grain yield, except for potassium and calcium. So they concluded that heat stress alone or in combination with low phosphorus nutrition had a serious adverse effect on grain yield and mineral nutrient composition in wheat. And elevated carbon dioxide aggravate these negative effects mainly due to the reduction in storage protein or because of a dilution effect. Gajendra Ramesh, he worked on physiological analysis of root traits and architecture for adaptability and yield under water deficit stress in wheat under the supervision of Dr. Rakesh Pandey. And here they studied the root system architecture in relation to deeper rooting in different methods. So initially they screened 78 wheat genotypes for root angle between the seminal roots using agar-gel method. And from there they screened 23 genotypes, and that were evaluated in the PVC pipes for deeper rooting. So from that also, whatever they got, like 6 contrasting genotypes they got, and that was put in the field, and they studied the root profile by trench method. So they concluded that the genotypes with the deeper rooting system like C306, Raj 3765, and HI 1500. So these genotypes with deeper rooting system and root size showed relatively higher photosynthesis, stomatal conductance, and transpiration under drought stress, thus resulting in lesser decline in yield in wheat under terminal drought stress. So they they also made a comparison between the deeper and shallow rooting genotypes in wheat also at gene level. So they finally concluded that the response of deeper rooting wheat genotypes under water stress with respect to morphological changes, photosynthesis, yield, and sugar availability for root growth. And these insights obtained will be useful for developing water deficient tolerant varieties. Sagar, he worked on evaluation of pod pedicel as a component trait to facilitate photosynthetic supply to developing seeds during soil moisture depletion in chickpea under Dr. Mahesh Kumar. So this work was initiated initially at Niazam, Baramati, but then Dr. Mahesh Kumar, he was transferred to IRA, so he completed this in IRA. So here they took 31 chickpea genotypes, and they took one set was defoliated and one set was not defoliated. They took the grain yield, seed yield, and also the surface area of pedicel, and did the imaging using MHA, did analysis using MHA, and then they found that the removal of leaves reduced the grain yield mainly due to a reduction in pod number rather than the seed weight per pod. And also they studied the association between seed size and pedicel area under water stress condition, and they found that the shorter the pedicel, better would be the size of seeds under both the irrigated and drought stress conditions. So the genotypes with shorter pedicel in chickpea can lead to a higher seed yield. Ms. Malini, she worked on physiological analysis of photosynthesis and its component traits under high temperature stress in rice under the supervision of Dr. Madan Pal Singh. So here she took 144 accessions which were selected from the 3000 rhizome panel, and they were grown under ambient temperature till booting stage, and then they were transferred to high temperature around 38 degrees for 5 days, and after that the phenotyping of for gas exchange traits was done in the flag leaf. From this study they ranked this genotypes rice accessions, and they selected 4 contrasting accessions with NL44 as a check, and they studied the component traits of high temperature. So this among the first 2 are the high temperature tolerant accessions, and next 2 are the sensitive accessions. So in this they demonstrated that higher gas exchange, cooler canopy, and optimal AVA concentration, these were related with high temperature stress tolerance in rice accessions. So they also did GWAS for photosynthesis related traits under high temperature stress, and using this MLM model, they detected 6 and 24 significant marker trait associations for transpiration rate and should dry weight respectively. And so they concluded that most of the candidate genes identified from GWAS were involved in stress responses, making them valuable for marker assisted selection to improve heat stress tolerance at the reproductive stage. But this needs further validation. Shamima Parveen, she worked on the effect of post-synthesis high night temperature on starch and protein metabolism in developing grains of late sown wheat under the chairmanship of Dr. Anjali Anand. And here they screened, because high night temperature is another factor which is very less studied actually, but it affects the yield or grain filling. So they screened initially 20 genotypes of wheat, including a check based on the heat susceptibility index. So they grew it in the control chamber, and one chamber was exposed to high night temperature. So that was ambient plus more than 2.5 degrees from putting to physiological maturity stage. So they found that the reduction in grain yield under high night temperature was due to reduced biomass and grain number, and also the water soluble carbohydrates. This was negatively correlated with grain yield under high night temperature. So from this study, they selected 4 contrasting genotypes on the basis of heat susceptibility index and crop duration. And then they studied in depth these genotypes on the spike, and they divided the spike into inferior grains and superior grains, and all the studies were confined to this inferior and superior grains. So here they found that increased activity of sucrose synthase at early phase and cell wall invertase at later stages of grain development provides substrate for grain filling. And the higher activity in superior grains under higher temperature showed its higher zinc strength compared to the inferior ones. Further, they studied the biochemical changes in protein composition in developing wheat grains. So they found that the stability in alpha and beta gliadin across both the environments, that is control and higher temperature, and both in superior and inferior grains, in all these genotypes showed the alteration caused, so that the alteration caused protein fractions may affect the dough extensibility and loaf volume. Purvali Mukherjee, who also presented very nicely yesterday for gold medal, she worked on interactive role of sucrose, ROS, and auxine during development of female gametophyte of wheat under high net temperature under the guidance of Dr. Anjali Anand. So here they took they collected ears at the heading stage, and they put it in an incubator and exposed high net temperature from 6 p.m. to 6 a.m. for 12 hours consecutively for 5 days, up to the antithesis stage. And the temperature they increased was +5 degrees and +8 degrees with and without dimethylthiourea. And this study was confined to the high temperature tolerant Raj 3765 and susceptible one 2329. So here they showed that there is a significant accumulation of raffinose in both the genotypes with the increment in night temperature, and the significant accumulation of glucose was also found in sensitive and fructose intolerant genotypes at +8 degrees. Similarly, the expression of raffinose synthase gene, this was increased at higher temperature, and it was particularly more in the Raj 3765 at +8 degrees. Then they also studied the effect of higher temperature on antioxidant defense system in pistil, and here they detected the H2O2 synthesis. So they found a significant increase in H2O2 production under higher temperature in both genotypes, and it was higher in HD 2329. So that is also seen with the pistil having a very dark, more staining compared to the tolerant one. And so this trend of increase in peroxidase activity was also similar to the H2O2, indicating its involvement in scavenging of H2O2. Then the effect of HNT on ABA and GA3 concentration in pistil was studied, and they found that the hormonal control of tolerance was evident by lower levels of GA3 in the tolerant genotype. That is, the ratio of ABA and GA3 is important for the pistil to tolerate the high night temperature. So this study filled the critical knowledge gap on the impact of HNT high night temperature on pistils alone, which are equally responsible for seed set in wheat. Monish Kumar worked on dissecting the photoprotective role of photosynthetic pigments for heat tolerance in developing green grain of wheat under the supervision of Dr. Pramod Kumar. So they initially screened 32 genotypes of wheat for photosynthetic pigments and chlorophyll fluorescence parameters in green grain under normal and late sown conditions. And out of 32 genotypes, they identified 2 best performing ones, the heat tolerance. So the further analysis was restricted to these contrasting ones. So then they analyzed the photosynthetic pigments in this, and they found that the catenoid composition, they found the reduced volatility of these pigments and in the susceptible ones, and there was upregulation of PEP carboxylase and Rubisco genes under heat stress intolerant genotypes, and this indicated the existence of C4 photosynthesis in developing green grains. Pavitra worked the evaluation of wheat germplasm for stay green traits under drought and heat and combined stress under Dr. Ajay Arora. They screened 298 lines with 4 checks under 4 different conditions: control, drought, heat, and heat plus drought. And they identified, classified these into 3 groups, these genotypes. And they finally found that the HT3086 performed better under control condition, while DBW187 performed well under drought stress, while ST2967 and VL829 performed well under both the heat and combined stress. Dinesh Kumar studied the physiological characterization of mutant for vaccination in wheat under Dr. Vijay Paul. And this HT3086 was mutated, and he used 12 mutant lines of this variety. And they found that the lower temperature of main ear, peduncle, and flag leaf of main shoot during anthesis and post-anthesis periods facilitated grain of main ear. They were resulting in higher harvest index of main ear. They also studied the epicuticular wax content and yield-related traits in selected mutant lines. And they found that the better pigment profile in waxy mutant lines was due to higher epicuticular wax content on flag leaves, which resulted in slower desiccation and higher chlorophyll B content under normal and heat stress conditions. Rohit Hegde, under high throughput phenotyping theme, he did precise phenotyping of Brassica junctia for drought tolerance under Dr. Sudhir Kumar's guidance. So they did phenotyping using the non-destructive method in Plant Phenomics Center, and they took 40 genotypes, and also they collected the manual data, destructive sampling, and they generated a big phenom data, image data of 691 GB on drought tolerance in Brassica junctia, identified some drought-tolerant genotypes, 6 brassica, and so they have also found the prospects of combining high-throughput phenotyping and genomic selection to dissect the genetic architecture of complex traits. Another thesis on high-throughput phenotyping was done by Tamil Selvan. So assessment of efficacy of image-based tools to differentiate drought responses of pulse crops at seedling stage under the guidance of Dr. Jagdish Rane. So here they compared the plant genomics facility and affordable phenotyping using the smartphone, and they used 4 grain legumes under water deficit and well-watered condition. And they found that digital volume can differentiate treatment and genotype effects in pulse crops very efficiently. So they found that the smartphone-based protocols with open source image analysis software can serve as an affordable phenotyping tool for water stress response pulse crop. This is the last one. Milan Kumar, who also bagged gold medal yesterday. So he studied the analysis of glycemic index and associated attributes in potato tubers under the guidance of Dr. Madan Pal. So he screened initially 73 potato varieties for amylose, amylopectin, starch, and other carbohydrates. And he found that the resistant starch and amylose, they are negatively correlated with glycemic index and glycemic load. He also studied the effect of processing on glycemic response parameters in potato pulse using potato pulse, potato oil, and potato vegetable combination. And he found the reduction in starch hydrolysis index, glycemic index, and glycemic load, whereas there was increased resistant starch and decreased available carbohydrate. Thank you. I acknowledge the Director IRI, Joint Director Education and Dean, Joint Director Research, our Head Division of Plant Physiology, students and staff of the Division of Plant Physiology, and definitely more important thing, funding from DBT, DST, CSIR, NAHEP, and NASF. Thank you.
Very fine. Thank you very much, Professor Pandey. As I see in the program, it is written here, chairperson's remark. So on this occasion, the very first thing I would like to say is that all 3 organizations are doing wonderful work, and we are very happy to see the presentation and illustration of the work which has been done by students and also by input from scientists. You have presented that there are about 173 publications jointly from all 3 organizations, and out of them about 20% are from the students who have completed their degree this year, which is a significant number because the department includes several other students also. So congratulations for that. If I could briefly say, the biochemistry division emphasized quality measurements and processing, shelf life of millets, and product development. Also, they worked on nutrition and yield-related pathways and the genes. And we saw that in relation to yield, there is work on Rubisco activates, which is very interesting, and particularly in the light of the fact that this is turning out to be thermostable. Molecular biology and biotechnology are working on abiotic stress tolerance, and basic research. I could speak for them for an hour or so because recently I had the privilege of completing URT of that particular institute. But to be in brief, I realized that they are working on temperature stress, salt stress, and root architecture in relation to this. Several of the genes related to biotic stress have also been worked out. There is a specific emphasis on alternaria resistance in brassica. To basic research, besides the genomics work that the institute has been doing, there are basic questions being asked about the biology of the systems, and there is work on phytochrome from the low light tolerance varieties. There is also isolation of the gene for carotenoid cleavage. High grain number is a very promising genotype, and its molecular basis is being uncovered. I am hopeful that within a year or so, you will be able to pinpoint that this is the gene responsible for this. There is work on isoflavone synthase, seed proteins, and some interesting work on calcium oxide nanoparticles as well. So there is some diversification on that. Plant physiology has been doing very interesting work on plant nutrition. And there is a legacy of elevated CO2 input work from this division, high temperature work, and also phosphate utilization. Dot and heat are being combined together, which is the need of the hour, because these two traits are affecting several of the parameters of the plant, be it root development, photosynthetic transport, or female gametophyte development, the work which has been shown here. High throughput phenotyping is a specific domain for the division. A lot of work has been done on this, and this reminds me that I could say something to the director. I am really very happy that this facility has come out very nicely, but I was missing in the field a field phenomics facility. And I think these people who are working on basic and applied work would be able to. I can take those liberties. You see, that's the problem of calling a person from outside. Okay. But anyway, this is not intrusion into the boundary. I just saw, I saw a wonderful pond structure being developed, and also a pyramid where everyone would like to visit. But then I could also see in my mind that there is something which I could say. So these are specific. What I am missing. What I'm missing is that nobody told me that out of these three divisions which are doing a lot of basic work together, how much is the component which is joint work? There is enough room for joint work, and there are specialty. No single person can achieve everything. So I am sure there is some joint work. I am aware of it. But that should also be highlighted so that more students, more scientists are encouraged to come together and work at the cross-boundary areas and give their input. Finally, I would like to just say three things. I hope I have 3 minutes. You can have your red light. Okay. So the first thing is we have so far emphasized creativity of the young people. Now, creativity gives wings to the ideas. But new thing has been emerging, and that is innovation. And we have to train students into innovation for the reason that while creativity gives wings to the ideas, innovation helps us realize that in the form of products and the processes. And therefore, there has to be a regular effort to bring. Government is also emphasizing. We will be constrained at one or other point to go more into innovation. But creativity, of course, is innate. And even if you don't nurture it too much, it will show up one day. But innovation can be taught, and people can be trained into innovation. The next thing is, which I already emphasized, networking. Because individually things are not possible. Every time I come to IRI, I learn new things, let me tell you, without any hesitation. I don't hesitate to pick the phone and call any one of my colleagues here and ask what, I will even ask what is LOD, Ashok. And then he will explain me in detail. Such simple things, why not we ask them? So we should cooperate, we should coordinate, and we should coexist. Our success lies in that. The success of human race has also been because of this networking. We are the people who can network much better than any other living organism on this earth. Why not use that potential? And the last thing is that we have to have the mechanisms and processes of continuous learning. Otherwise, we will be outdated. Students know very well about the ChatGPT. Many of them must have already used it and also must have found what are the drawbacks But fact remains that these technologies will be perfected, and those things which can be worked out by artificial intelligence will be relegated to computers. And therefore, the opportunity and jobs will be for those who are going beyond this. And therefore, this learning process for scientists as well as for students need to continue. We have to find some specialization, we have to find some direction, and we have to remember that there is enough space on the top. Only thing, you have to reach the top. Thank you very much. That is all what I want to say. He has a question. माइक दे दीजिए अगर है तो.
Good evening to all. I am extremely happy to be part of this. Means IRI is a family. It's homecoming for me. Then second one, all the basic sciences professors I congratulate. And I agree, Professor Tyagi, that boundaries within the subject, I mean, there should be coordination between the subjects. Sir, I'll tell you that maximum paper I have published with all breeders in genetics division. Maybe chickpea, wheat and all crops, rice also I have published 3-4 papers. So I mean, since then I was advocating that things should come more vibrantly and more understanding should be among the colleagues. So I congratulate everyone because I am extremely happy that whatever limitations what we were having at our time, so all those limitations are over. And congratulations to all 3 professors for excellent presentation. Thank you very much for the opportunity.
Thank you. Thank you.
Thank you all. So now it's time to felicitate the chairperson for today. May I request Director Sir and Dean Madam to kindly do the honors. So thank you. As the convener for the professor's presentation, it's my moral duty to propose a formal vote of thanks. So on behalf of the director and dean, we all are thankful to the chairperson, Professor Tyagi, for his acceptance of the invitation and conducting the proceedings of the session in a very comprehensive manner. Thank you so much, sir. The suggestions given by you, sir, will be duly taken care of, and we will try to seek your guidance in all the years to come. Thanks are also due to JDR sir and other dignitaries who have joined us for the session. And my sincere thanks due to our director and dean ma'am for their constant support and guidance throughout, taking care of every minute detail, for which Dean Madam we have seen is personally awake till midnight, sending WhatsApp and getting the rehearsals done for the next day. Thank you so much, ma'am. So this has ensured the success of the program. I wish to place on record my gratitude also to all the convocation committees for their kind cooperation and coordination throughout. Special thanks to the team members from the School of NRM, Dr. Renu, Principal Scientist, and from the School of Basic Sciences, Dr. Archana Singh, Principal Scientist, for all their support in editing the booklet and coordination of the sessions. I also thank the Associate Dean along with his dedicated team from PG School and Dean and Director Office for providing all the logistics support. Last but not the least, thanks are due to the professors of the School of Basic Sciences for doing their presentation within the time frame, which has enabled us to wind up the today's session in time. Thank you one and all. And we'll meet tomorrow at 9 a.m. for the next session tomorrow on horticultural sciences. Thank you. Thank you, sir.